Cazzo fai? Non me lo dici già. Andata? Ha gridato per giusto su YouTube. Ha gridato per giusto su YouTube. Ok, vai. Se vuoi puoi mutare poi. Ok, for the presentation.
Yes.
Ok. Which one?
Okay, hold on a moment. Bari Granada. Bari. And now, wait, no. Okay, go back.
Okay.
Here.
Oh, okay.
Okay. This one, I think. No, this is PowerPoint. Should be this one.
September 22.
Yes, but this one is a power. This is this one.
Okay, but today is 23. It's just a
That's,
mistake. Okay.
yeah, that was a mistake. I have it on the.
So
Maybe you can open it. This in template. Yeah, it's this one. It's
okay.
that one. It's that one.
Okay. Now I just copy.
Today is 23. Yes, I'm.
Okay. No.
At 11.
Mm-hmm.
You can change it.
At 11. So here, just rename September 23. No space. Okay. If you want, you can check.
Okay. Well, okay.
If you want to put a presentation mode.
No funciona el este, no.
Ayer no lo estaba usando la gente.
What is about
What do you
doing
want to try?
to go page by page?
To
This
go
one.
page by page. This one.
This one.
Yeah.
What?
Oh, sorry. I don't know why.
Okay. Más o menos.
I don't know if you want to spoil, but we can put on presentation mode just to check.
No, but you mean here or
Yes.
just? No, it's okay.
Okay.
Okay. I think it's okay.
Perez Gato.
Yes. Okay, it's okay. I can get it. The pointer works.
The
Does
pointer
it work?
works now. Is not.
No, but usually, I mean, normally.
This button for the pointer.
Yes.
And if you want to go to the next slide, you can use this button.
Yeah, but maybe I prefer this one.
You can
So
choose.
that one, yeah, with this one will be
These
the
two
arrow
arrows.
here. These two arrows.
These are
okay.
to go to the next.
But maybe if I use the pointer, I will.
I don't know. Yesterday, a lot of people just
Yes.
preferred this.
Pero ¿cuándo me les iba? Porque ayer usaban este, o sea, intentaban con este y como no iba así, iba.
No lo sé.
There are two.
Okay. So this one is.
Yeah. This is the afternoon.
Okay, this is the morning. The next one. Okay.
Yeah, this is the winner, the first one.
So we have to rename it in a second. So this is the first one. Then we have this one. Okay. You want to check?
Yes, please. Okay.
Okay, now because we are mirroring another presentation, another slide. But okay, if we do this. Okay.
Okay. It's fine. Okay. Yep. If I can take the other one just to see that the
This was.
animations. To see is here, right? I got
No, no.
okay.
This.
Okay. Okay. Okay, it's fine.
Perfect. Thank you.
Thank you.
Morning. Presentation. Yes. Okay. You can I believe. Maybe I
It's
can
on the IHS paper. And if you scroll down, it's this one.
this one.
Yes,
Oh, okay.
yes.
So.
I'm the last presenter.
Last presentation.
Yes.
6 p.m. Just to rename it.
Okay.
You want to check?
Yeah. Yeah, that's it.
I don't know if you want to put in presentation mode or you don't want to spoil something.
I want to see if. Oh yeah. I can get the notes here on the side. So
But,
that would be
okay.
perfect, yeah.
Okay.
Thank you very much.
Thank you. Ciao. Sì.
Dentro gli speedrun si chiama di guardo.
Quindi lei è precisamente
Questa mattina verso le 11
10:45. Quanto vorrei nominarlo?
grazie.
10:45, giusto? Ok? Ah sì, perché ha detto giustamente. Ok,
Grazie.
niente. Morning.
Morning.
Presentation, right?
How I think my time is at 12.
12. Okay. Twelve.
Yeah.
Which folder? This one. Okay.
Body, presentations, PC. And put this one here.
Okay. I have to rename it a second.
Sure, sure.
Okay. If you want to check.
Oh, seems fine, no?
No, it's fine. It's fine.
Looks great.
Perfect. Okay, you can
Yeah.
take it.
Thank you.
Thank you. Assolutamente no. Manca un sacco. Oggi è molto nutrito il programma. Manca. Giuseppe
Poi una cosa che devo fare è segnare i post che non sono stati presentati. Poi riceveremo le richieste, facciamo lo stesso lavoro. Questo dovremmo essere...
Chi non l'ha presentato non dobbiamo ancora pensare. Vabbè, e adesso non chiedi. Aspettiamo fino a domani ancora qualcuno.
Allora, la prima sessione dovrebbe esserci tutta.
Ok.
La prima sessione, la parte 1, poi c'è il pranzo e poi c'è la seconda sessione sempre post harvest e product quality. Ok. Ce ne sono soltanto 3 per ora.
Quindi quella di stamattina fino
Dovrebbero essere 5,
fino alle 11:30 ci sono tutti.
fino alle
Poi dopo
no,
c'è quella dalle 12 all'1 ci sono?
dalle 10 alle 13. Dalle 10 alle 13 perché c'è post harvest and product quality.
Sì,
Ci sono tutte. Poi la seconda, che è la parte 2, sempre di post harvest and product quality,
nel pomeriggio.
ce ne sono 3 su 5.
Vabbè, penso che le porteranno.
Poi continua, che vabbè c'è anche, ricordatevi che c'è la presentazione degli sponsor di Unitec,
Sì,
che è
sì,
un video,
dopo.
quindi loro non ci sono.
Ah, non
No, è
sono
un video
presenti.
di non so quanto.
E la stessa cosa ci sarà domani per quello di la poesia Lucia manderà alla registrazione.
Ah, ok. Una presentazione.
Per una presentazione.
Poi
Poi c'è quella
la
Genetics
Genetics
dalle
sono 1,
4,
2,
5 sono.
5, ce ne sono soltanto 2.
Vabbè.
E poi c'è Field Management, 2 sono Field Management,
Sì.
cioè Quartuale, ci sono. Quelle che-
Vabbè, quelle che mancano, penso che porteranno.
Poi uno che è venuto a caricare la presentazione vi ha anche dato un video per gli SFS Business Meeting.
Quello di stasera,
Quindi-
che è quello di Cipro, della candidatura?
Può
Di
essere,
Cipro,
sì.
sì.
Poi bisognerà mettere questo video.
Vabbè, quello è il business meeting che facciamo vedere, quello dopo, sì.
Ma ho visto che c'è in contemporanea o ce la facciamo per c'è messo in programma pure a piedi di fare.
E chi, quello là, sì, alcuni che siano registrati. Ah
ci
sì,
sono.
vedo che incomincia prima.
Perché anche voi dovrete esserci.
No, io non ci vado, tu devi andare a un punto.
No, non lo so, una passeggiata me la farei pure. Vabbè, poi facciamo
Si è cavato.
dove
Sì.
sono. Cioè, c'è una guida.
Sì, forse doveva incominciare alle 7, hanno organizzato così.
Forse per non fare troppo tardi.
No, vabbè. Good morning to everyone. I hope you rested very well yesterday. Today we have a full day of presentation. We now will start with another important keynote lecture from South Africa, Professor Rani Kauri, with the title Congruent Research for Capacity Building, Insights from a value chain based approach in South Africa. Please welcome Rani Kauri.
Greetings, everyone. It's good to be back on the podium. And greetings to my South African colleagues. I think we have a very good, strong representation from South Africa. And today I'll be sharing some of the work we have done so far for the past 15 years in South Africa. Even starting from me being a PhD student, the very first PhD student working on pomegranate when the industry was still very, very small and very, very young. It's still a very, very young industry, but there's a whole lot of opportunities for research, and that's what I want to share with you today. And over the past 15 years, we've been able to train over 50 postgraduate students, and communicated our research in both international and local conferences. So I'm previously from Stellenbosch University, but now I'm at the University of Johannesburg, and I have a research center I established in 2003, 2023, called PAC, the Post-Harvest and Agro-Processing Research Center. And I also have a national research chair, focusing on sustainable post-harvest preservation at the University of Johannesburg. Like I said, I still work very closely with Stellenbosch University, even as an extraordinary full professor there. I'm not going to go too much into my group, but you can read more about our group. We are now very big. We have 48 people in the group, and we are actually doing very great. So the core mandate is about capacity building through, student training, infrastructure building. We also building knowledge systems, and I will share some of these approaches, how we were able to train so many students just focusing on one small crop, pomegranate, and that's what I'm going to talk about. And my story will not be complete without mentioning my PhD supervisor. You all probably know him. He's very popular, Professor Linus Opara. He was my first, he was the person that actually introduced me to pomegranate. And together, we've been able to build human capital, institutional capital, like I said, and also infrastructure capacity around pomegranate. And we work very closely with Pomasa. We have our representative here. They funded our research in the past. They're still supporting us. And through that, we've been able to leverage this funding for other funding opportunities. So, like I said, I think I mentioned this yesterday. I hosted the last pomegranate conference 2 years ago. And there's one thing that stood out for me at that conference. We spoke about pomegranates having, phenolics, L-argitannins and all that, but it goes beyond that because these phenolics actually, the gut microbiome transform, metabolite changes them actually into urolithin, and this urolithin is what actually causes the functional properties that we observe through metabolism of these phenolic compound. And this happen in the gut, So when we talk about pomegranate is rich in polyphenols, it's not only about the polyphenol, it's the conversion, metabolism of those polyphenols into, uronithin A and B. And we have literature, to back this up in terms of how pomegranate can actually be used as an anti-cancer, for cancer management. It all boils down to the metabolism of polyphenol in pomegranate into uronithin. We showed this. I mean, my colleague showed this. I think it was Ali that showed this yesterday. Like we said, also there's no consensus with regards to global population, production of pomegranate worldwide. But we know the key players in the industry. Globally for pomegranate. We also know something for a fact that South Africa is well positioned when it comes to entering the market, the global market. You can see here, this window, we're able to actually supply most countries during this during our window period. The only backdrop of this is our markets are very far from us. So that means storage is very, very important. Unlike some other countries that they are very close to their market. I'm sure Italy is close to the market because you probably send you sell to neighbors. Post service is very, very, very important to us. And if we go into, I was just going to mention that we actually captured, there's a book chapter that will be published soon together with Giuseppe that we work together. This was well captured in this, and we will be sure to share with you some of especially the global production of pomegranate and up-to-date information on pomegranate production. But let me take you through the overview of pomegranate in South Africa. I got this data from Pomasa, a very well organized association, and they update their information. Still very, very young. You cannot compare pomegranate with your apples and your stone fruit and all that. It's still 1,000, just above 1,000 hectares, and you can see the cultivars, different cultivars, and you can see here the wonderful is actually predominant. When I started working on pomegranate, it was about 9, 10. You work on one cultivar this year, the following year they uproot, they plant another cultivar. So it's really very, very dynamic, trying new things all the time, and it seems to be stable now. But you see that all the small cultivars over time can actually fade away. You see new cultivars coming through. But wonderful has been very, very consistent in terms of being the most produced cultivated cultivars. In terms of the exports, exports in terms of the tonnage of fruit that we export, it's also very consistent in terms of the growth is increasing. And you can also see that the export dynamics based on policy changes depending on the export market. As you can see here, you see now US and Canada destination is now last year versus 2023 and 2024 from 2% to 0%. I'm sure this has a lot to do with the policy that is in place in that market destination. And what this means is that we have to change, reposition our, focus on another destination, which might require another post-service in terms of quality standard or post-service management practices. So there's always something to do in terms of improving our protocol or handling system, or also meeting the requirement in the export destination. So what we know, there are quite a lot. In fact, if you ask a farmer, there's 100 items that they will tell you about the challenges with pomegranate, from production all the way to to even the market. But we know major issues, from even when I started working on pomegranate, starting from when do you harvest? Harvest maturity was a big problem then. Well, because we have all this cultivar, we don't know when is the optimal harvest time for each of these cultivars. And I will show you some of the work we have done in terms of how we were able to develop harvest maturity or harvest index for each of the cultivar. We have the issue of moisture loss, which my colleague mentioned yesterday, gave a talk on yesterday. And we have, of course, decay. I have my colleagues here working on pathology aspect of in terms of decay control for pomegranate. So I will share some case studies with you, how our research approach has developed maturity index for pomegranate, and how this approach can actually be used for new cultivars that are being introduced to the country. We also work on storage and packaging systems, how things have changed over time, starting from, the different packaging liners, poly liners, and now zooming on to the unintended consequences of packaging liners, and even now the shift into sustainability, because now export destination, they don't want plastic. Some don't want plastic, and they want us to reduce the carbon footprint. And our research has now shifted into coating, edible coating, forming nanocomposites for post-harvest preservation. So I'm going to quickly go through this. This was based on my PhD in 2010. And, when we were able to, the question is, pomegranate is non-climateric. We know the color of the peel is not the same as the color of the area. You cannot actually use the external to predict the internal. Some already at the very young stage are red, so you don't know what is on the inside. It's not a good predictor. And we know that can ripen depending on the maturity period. It can range between 5 to 8 months based on literature. So what we were able to do here was to develop stages, developmental stages, 5 developmental stages, and group into 3 categories: immature, half ripe, and full ripe. And this was based on the chemical composition, and multi-parameters that we're able to, to put together to categorize this. By the way, this was Bagua, which was no longer in production then. And so from then, we took before we actually did that, the first approach was, to even understand the growth studies, how the fruit actually grow, the growth and developmental stages of the fruit. Then we will look at, okay, what is the pattern of the quality attribute during the growth and developmental stages, before we now went into the last step, which was now focusing on harvesting at different maturity stages. And now, depending on the export destination, we're able to develop a particular index for each cultivar. So this work is based on publication that you can capture and read up on that we published a while back. So focusing on harvest index, this is what I want to talk about, dwell about a little bit more. We know that because South Africa is far from the export destination, our harvest must be informed or must factor in the time it will take for the fruit to enter the export market. And that's what we considered here. We know that, the time it will take on the orchard, orchard to pack house, the time it will take, all the way to get to the market and also to get onto the shelf, we need to factor this in for us to develop a reliable index, and that's what we did here. And now putting everything together with sensory measurement, instrument statistical analysis, and also instrumental analysis. Putting this together, we're able to actually develop a reliable index. And this is just showing how we were able to put all these different attributes together to come up with which harvest maturity or harvest stage should be used for harvesting. For this particular cultivar, I think this might be Ruby or Bagua. Then we were able to have a predictor of TSS greater than 16, greater or equal to 16 to harvest it based on the export destination, the storage temperature then, and also the time it would take in terms of the handling of the cultivar then. And this work was published there. So, and at the end of the day, this was found to be between harvest 2 and harvest 3, which also coincides to a day after full bloom that we were able to, to develop then. And this approach has now been used for many other cultivars. This one, another cultivar, Wonderful. For Wonderful, it's the harvest in terms of the highly ranked indicator is actually TSS and TA ratio for Wonderful, as opposed to your TA separately or TSS separately. The most reliable index for your Wonderful is that ratio, fine balance between the two, to actually have a good predictor or reliable harvest index for Wonderful. So the second one is, okay, now you'll be able to harvest. You probably now harvest at the optimum condition. Storage becomes storage temperature becomes an issue now. Initially we know that chill injury became a big issue especially if it's below 5 degrees. And what we were able to determine, to establish during the study it's actually not only about the storage temperature. It's about how long the fruit stays in that storage temperature when you are dealing with chilling injury. It's about how long. And this work actually was published, and it informed us through the work, the statistics, the outcome of the work, that duration actually plays a significant role. And again, this is for these two cultivars, which can also translate, can be used for other cultivars. And this information actually was used to develop a guideline by the Export Council, then PPECB, South Africa, that actually regulate the exporting of pomegranate. So then the other aspect, my colleague, that the second person that gave the keynote, spoke a lot about relative humidity, which our work actually also agrees with to a large extent. We look into the influence of relative humidity on fruit quality, of course, weight loss. And as you can see here, we looked at low relative humidity and high relative humidity, low being 65% and high being 95%. Of course, you can see how rapid the weight loss could be for pomegranate. Of course, we sell by weight, so this is a lot of money. So we were able to actually see how much this can result into. If you lose so much, even a unit fruit, it can be so big when you have a carton or when you have a whole consignment. It can be so significant. Of course, we now know we know, and also corroborated yesterday, that weight loss had a lot to do with quality. Of course, the fruit now start disintegrating because of it's a stress factor. So what we did here was to now know, okay, what then is the effect of weight loss on, we know that scald will start happening when fruit loses weight. We know that it will also start having patches, shrivel and start having those patches, it will start being deformed. It will start losing the shape. So we did an experiment where we observed that at a very low relative humidity, you lose 5% already in 6 days. But at a high relative humidity, the same amount is lost at on 24 day of storage, on the 24 day of storage, which was still even lower a little bit then. And the same effect that we observed on 6 day was observed only on the 24th day, at high relative humidity. And note that this fruit was actually still at room temperature. We regulated the relative humidity, just to know the impact of relative humidity on fruit quality. So I did some experiment. I said, okay, why is this happening? I hope this video will play. So I did a small lab experiment to show you that pomegranate actually has micropores. It's full of pores, and when you talk about vapor pressure deficit, you can really see that it's so easy for water to be lost from pomegranate if you don't control the relative humidity in the storage. So then we move further to check using X-ray CT, to actually check now, to visualize using X-ray. Of course, X-ray will tell us all these white dots that you see, they are full of pores. Pores. The fruit is full of pores, micropores that you cannot even see. And this what when you see a fruit, and that's why packaging is very, very important. Or some form of coating, you need to ensure that the fruit surface is protected. Otherwise, it's just an open door for water to get into. To get out. In fact, it could be even an opening for any pathogen to go into as well. This work was also published. So we went further in terms of, okay, we know that relative humidity can affect the external features. What is the impact of relative humidity on the chemical attributes, and focusing more on TSS and TA of the fruit? Of course, it was interesting to know that the fruit at low relative humidity, the fruit that is rather looking ugly, actually tasted better because of concentration effect. It's sweeter, it tasted better, but then it's deformed, it's shriveled. So what this means is that if you're going into aerial processing, you don't need to worry about really the external features. Your fruit will be better, of course, if you don't get decay and all that. But fruits that is deformed and stored at low relative humidity actually lost a lot of weight, but then the areas became sweeter and all that. Of course, it has a limit at which after that you start actually entering senescence and the fruit will go bad. So, okay, from our understanding of fruit now, post-harvest management, we know that relative post-harvest life of a fruit is based on so many interventions. If you abuse your fruit, maybe you abuse the relative humidity and the temperature is not optimal, you can get so much of relative post-harvest life. If you supplement this abusive conditions, say temperature and relative humidity, you supplement, maybe you apply some chemicals or you apply any other intervention, you can gain some more, days or steps or months, depending on the fruit that you are dealing with. If you are able to find an optimal temperature and a optimal relative humidity range for your commodity, then you extend it. Each time you have an intervention or a huddle technology, you actually extend it. In this case, if you had now supplemental treatment, then you can actually gain more. So this is what shelf life extension and post-harvest life is all about. So this what now inform our philosophy, our research approach, that okay, we know the relative humidity, the impact of relative humidity, impact of temperature. Now let's look into packaging systems. And this work, we actually trained a lot of PhD on this packaging system. We know how it regulates income of inflow and outflow of gases, talking about oxygen and carbon dioxide and also water. And in this experiment, we looked into, we asked ourselves, what type of modified atmosphere packaging, can actually make a difference. Then then there were so many polyliners in the market. So we came up with a mechanism where we actually shrink wrap the fruit. And unfortunately, the shrink wrap actually induced a lot of decay in the fruit, even though it performed better than control, and even relatively the same as reducing the weight loss. But because moisture is trapped, it's not perforated, it induced decay in the fruit. It is individual packaging system, unlike the big one. So you're actually able to concentrate the microbial load, in the system, the packaging system. Otherwise, it actually also, perform in terms of keeping the quality, in terms of weight loss and other attributes. But if decay is the problem, then you have a big issue with it. So this led us to looking at so many polyliners that were in the market then. Then we had all these polyliners. We have the Deco, Zoe, Extend, and we have them at different perforations. We have the 2mm and 4mm. And you can see they perform quite well, of course, compared to your no liner, the control. And of course, this has an effect in terms of financial implication. But then one must also consider that this an input cost. There's input cost, so you must weigh there must be a fine balance between how much fruit you will rescue versus how much you are spending on the packaging. So it's something that the industry is really battling. We now have, I think, another packaging now that's so expensive, but it's very important to know, to have a trade-off in terms of how much you are losing or you are going to actually reduce, and the cost of your packaging liners. Here we look at unintended consequences, because you see, when you have your polyliners, because they have different perforation, if you don't do forced air cooling, you have water retaining through transpiration inside the packaging system. So we look at the condensate score in terms of how much water is retained in each of these polyliners, which can end up inducing decay, because this can actually result in unintended consequences. And this is very, very important. It's not only about quality preservation, but also what can be the unintended consequences if you don't do air force cooling, or you have abusive temperature in between. Then you're going to get condensate, which can induce decay in your fruit. So then there's this issue of sustainability came by the EU Green Deal that speaks about reducing chemicals, reducing your carbon footprint through plastic use, reuse and all that, and being conscious about the environment. So this shifted our attention to edible coating, and we've been doing this for the past 7 to 8 years now. We know edible coating, what it can do. It's actually something that edible coating technology is difficult on one hand, but also very interesting, because you can customize it for a particular problem, and you also customize it for a particular commodity. But then it cannot be one size fits all. You cannot have one edible coating and you think you can apply that edible coating to all the commodities. No, it's never like that. And what that gives, the privilege that we have with edible coating is that you can now work around the active in that composite. So you can include nanomaterials, you can include your essential oil, you can include whatever, depending on the problem you are trying to address. I have a PhD that finished last year. He focused on phytohormone enriched edible coating. We know that there's so many phytohormones that are actually exogenous applications that are used, to improve the defense mechanism of fruits, and they've been used to actually delay senescence and extend shelf life of commodities. So we developed a composite coating based on this brasinosteroid. It's 2,4-epibrasinolide that we use to delay senescence and also to enhance the quality or retain the quality of pomegranate. And I will show you very quickly before I go into the next one. Of course, the idea here is when you have a composite coating, the coating, the polymer, will help in preventing moisture loss, and there will be a migration of your bioactive into the fruit, which will then enhance the integrity of the fruit, and whether it's converting ROS, reactive oxidants, into the least reactive form, or it's actually enhancing the cell integrity of the commodity. So of course, weight loss, because we had now a protection around the fruit. Weight loss was controlled or was lower in your in coated fruit versus control. But what caught our attention that made us look into the mode of action was electrolyte leakage, which also relate to what the talk we had yesterday. Electrolyte leakage in fruits coated with coating that contain different concentrations of the epibrasinolide that we put in there. So we observe a form of delayed senescence. So which led us to an experiment. This is just a picture of the fruit. And by the way, we also store at cold storage, and then we move to shelf life. So this led us to, of course, we looked at the other parameters to see what other attributes were maintained using this composite coating that contained the bioactive. So, like I was saying, this work has been published. This led us to now understanding the mode of action, how this actually worked, and this is the experimental setup. But in this case, we focus on just the control and the one that contained 10%, 10 micromolar of the phytohormone that we included. So looking at the mode of action and understanding the mode of action, we observe that the accumulation of peroxide in the fruit actually resulted in leakage of electrolyte leakage in the fruit. You can see here where we had more than 60% accumulation of the peroxide, hydrogen peroxide in the fruit, led to 45% electrolyte leakage. And of course, we know the understanding of how this relates to ROS, reactive oxygen species, and how it induces, enhanced lipid peroxidation, and how that can now result in reduced membrane integrity. And of course, when we're looking at the issue of, for example, chilling injury, which has a lot to do with the lipid, the fatty acid in the pill, we know that when there's increase in ROS, in your reactive oxygen species, the unsaturated fatty acid are converted to saturated. You have more increase in them, and then fruit that has higher ratio of unsaturated fatty acid to saturated fatty acid actually can resist chilling injury. And this is what we observed here from our experiment, that those that were treated with the phytohormone, we had higher, higher ratio of unsaturated fatty acid to saturated fatty acid, which means that the treatment can actually help reduce or build resistance to chilling injury. Of course, there's so many things that we can look into when it comes to chilling. When it comes to now limiting the oxidative stress, and this is just a simple concept of what a normal cell would look like. And when you have free radicals, it's just this fragment, the cell. It actually kills the cell, and then you get the cell death. Through oxidative stress. And we look at so many other, enzymes that are now can help limit the impact of ROS, reactive oxygen species in the fruit. And if we look at, there's so many of them actually. If you look at many things, you can look in terms of your oxidative defense mechanism in plants or enzymes in plants. And some of them are enzymatic, some of them are non-enzymatic. So it's important to know the regulation of this in managing the oxidative stress in your during post-harvest management. And this is how we understood the mode of action in this composite coating. Of course, we did the non-enzymatic profiling, non-targeted profiling of the phenolics on the peel to actually better understand and knowing what are the indicators in terms of the biomarkers, polyphenols that can help in regulating this impact. So we propose a mode of action. I need to run fast now. We propose a mode of action for this purpose. So the last one I'm going to present to you is I spoke about that we focus on that fruit, on your fruit, but there's so much you can do with the peel. In fact, when you have your peel, the peel contains threefold of what we eat from the fruit. It contains threefold. And of course, we cannot go about now eating the fruit, the peel. But it contains threefold, and it's about 50% of the whole fruit when you peel it. So the question is, what can we do? What can we do with the peel? We were fortunate to be working with a company, the only organic pomegranate company farm in South Africa. It's called Garnico in Gauteng. They focus on aerials, just aerial packaging. And they were having issues with post-harvest management. In fact, I was at the job market, saw their commodities thrown by the side because they're all disintegrated. And I took this picture, I showed them, you see, what can we do? They said, no, they're struggling. The good thing here is they don't spray chemicals on their fruit. Their peel is considered organic raw material. So we were able to use the peel as an agent for post-harvest preservation, which I will quickly go through with you here. And when you have your peel, you dry it, you cannot apply it directly. It's not soluble, it's got fiber. So the first approach here is to upcycle it through encapsulation technology. So we extracted the peel and we turned the peel, the extract into powder. A powder that when you put in water like this, it dissolves. It's readily soluble in water, and we're able to standardize the powder. And of course, we also tested the powder against pomegranate pathogens. Showed very good activity. Then we were able to, form composites and characterize the film to actually see the barrier property that the film or the coating will possess in terms of protecting the commodity we are trying to preserve. And then this led us to the third step of now applying. In fact, after doing this, we also did in their company, to actually go through this process. And then we experienced significant extension of shelf life from 3 days, from 6 days to 9 days, because they are not allowed to apply any chemicals. The work was published this year in International Journal of Biological Micromolecules. But one thing that I wanted to show you is that, we were able with our composite coating, we were actually able to make their commodity more marketable for a longer period of time, from 9 days. I beg your pardon, from 9 days to 15 days. So we gained 6 days of extended of shelf life. And one thing that was interesting that actually baffled me, that we need to start thinking about patenting this technology. The way we went about this is because the student forgot to discard the experiment, and she went home on holiday. And after 6 weeks, I went into the code room and saw this. So the one that contained 0.5% of the encapsulated powder, still nothing grows. See the other one. So it was quite interesting that then what's going on here? What's going on here? This is something that is worth looking into further. And now we've been able to work with the farmer, not only about preserving, but also working with a company in the state on how they can use pomegranate peel for gut health purposes, where we can now package the powder. The powder, of course, we can use for edible coating, and now we can use for film packaging for meat, preservation, because now it's a functional film that can be used for preserving meat for extended shelf life. Like I said, we've been able to market, to formulate for other using encapsulated pomegranate extract for other commodities. This work was published recently in Food Packaging and Shelf Life. This was another PhD, but we optimized our coating using the encapsulated pomegranate peel extract, and it extended shelf life of banana. So you can see they're using circular economy, how we can use even the peel, the waste, to extend shelf life, not only of pomegranate, but other commodities. And this work, like I said, I've been working on edible coating for quite a while, and we've been able to work with pack houses. And, this trial, we actually coated plums and shipped to the EU, and we got some feedback, in terms of what happened on the other side. I think we used gum arabic on this one. We worked with a company called Stent. Then I think it was in 2018. So we've been on this for quite a while, and it's a way of building capacity, not only in the infrastructure, but also building human capital in terms of students through solving or addressing issues in the industry. The current project we are working on was on pomegranate. That was awarded last year at £250,000 on again edible coating, but now pre-harvest and post-harvest. We apply pre-harvest to address the issue of sunburn, and then we also apply in combination with some other bioactive to address the issue of post-harvest issues. I'm just showing you some pictures that we captured, that we are trying to. It's a big trial that we're working on now. So I think this more like the end of it. We're well, I'm excited to share stories. I mean, outcome of the result on this a very big trial. On that note, I would like to thank my my sponsors and collaborators, and of course my students that worked with me on this journey. And I'm using this opportunity to welcome you back to South Africa. I'm hosting the fourth International Symposium on Brevet Crops next year from 15 to 20 March at the Kruger National Park. If you haven't been to South Africa, well, it's one thing to visit South Africa, it's another thing to have a conference at the Kruger National Park. You really don't want to miss it. So please try your best to get on board. In fact, pomegranate work can also fit in this, can be a good fit on this work, on this symposium. So on that note, I thank you very much.
Thank you, Lenny, for the nice presentation. It's confirmed that the post-harvest aspect are very important, paramount importance for this crop. I'm sure there are a lot of questions. We cannot meet all the few questions since we are already late this morning. So please ask the audience for the question to please.
Morning, I'll try and speak loudly. Since
Is that better?
Yes.
In the last few years, it has become standard practice in South Africa for us to actually coat the pomegranates with the same wax that we use on citrus. So my question is, with reference to the external fruit coating, have you overlaid that or vetted against the certifications that we need to comply with in terms of market access? And the second question is, has any of those coatings been commercialized, or are you looking to do any trials in this coming season?
Thank you for that. So, and that's why we don't call it wax. We don't call it wax, we call it edible coating, because we know the code. There's a list of ingredients that is published that you have to comply with for you to call it edible coating. So we formulate based on the ingredients that are on the list. In terms of certification, if your ingredients for formulate, your composite coating is on the list, then it's actually certified as you can refer to it as. In terms of commercialization, we're working with what's the Citrashine. Citrashine. I think it's bought now by what's it called? Deco International. But now I think the idea here is the limitation, unlike the US, the limitation with South Africa is we don't have startup companies that can actually take care of the same for packaging, biodegradable packaging, systems. But in terms of commercialization, I think it's something we should look into, and it's something that the industry should actually get on board. And like I said, you have to customize your coating for a particular issue and a particular commodity.
Other question?
Thank you for this very nice comprehensive presentation. Now the edible coatings, so you actually showed one slide very quickly, but it was, the actual coatings were very good to prevent senescence and other things, decay, but it doesn't prevent weight loss so much. The weight loss is one of the most important issues for coatings. So what do you suggest to do? Because it doesn't reduce weight loss.
Then that was the dilemma, because we had an experiment where we combined edible coating with packaging liner, and then we were able to get significant. But we don't want to we are trying to reduce the plastic. So now hardening edible coat is another input cost, and that's why I was saying that you have to have a trade-off now. If your market doesn't like plastic, want plastic, you have to deal with what the minimum the edible coating can do. Of course, when we start increasing the concentration of the polymer, now the base coating, two challenges. Of course, the cost goes up. The second one, it becomes difficult to apply because we applied on the pack house. It clogs it clogs it and it becomes impractical. And dipping is not advisable because it wastes so much water and all that. Yeah. Yes, it's good to have more work to do.
You have time for another question? Any? Please.
Morning, Prof. Just one question. In terms of weight loss, you showed that the peel is about 50% of the weight of the fruit.
When you open your fruit, yeah, depending on the cultivar, it's between 48% to 50%.
Is the peel of the fruit?
Oh, yes, the
and
peel.
the rest is the arrows.
Yes.
So when you talk about a 5% weight loss or 10% weight loss during storage,
yes,
is that weight loss on the peel or on the arrows?
Good question. Good question.
Because if you don't eat the peel, you eat the arrows. So if the weight loss is out of the peel, why do you worry about weight loss?
No, but then it disfigures the fruit. Not only that, you are not talking about weight loss. When you start losing weight, then the fruit starts shriveling, and then it starts also the cosmetic aspect. So that means that you might not be able to meet the quality standard in the export destination. However, if your fruit is for area production, you don't need to worry about it.
So is the answer the weight loss is in the peel and not in the arrows?
Okay, so let me answer that question. By the time, I think when you get to, 8% of moisture loss, you start drawing, the peel is already start drawing moisture from the iris because it's connected.
More than 8% you would lose weight
from the iris, yeah. And that's why you get concentration effects. Yeah, and they become sweeter. Yeah.
Last question. Any? Otherwise, we leave the floor to Polani. So thank you so much for the presentation. Polani is now asked to chair the post-harvest and the product quality section. We have 6 presentations. We ask Polani to skip the time because we are already late. Okay. The floor is yours.
Okay, good. So we'll go straight to the next presentation, and may I invite Idris? Yes. Pardon me if I did not pronounce it well. From Volcanic Institute in Israel. You've got 10 minutes.
Well, 12 minutes.
How many minutes?
12.
15. Okay. I saw 10 minutes.
10 minutes for presentation and then 5 minutes for questions.
For questions. 15
No, but not 10. It's not 10. I said 12 past 3 years.
Negotiating the time.
Yes, we do now.
Okay.
Show me the
You
pointer.
can use this point. Okay.
The button, yes. And forward, this one is forward.
This one is forward. Okay. Or you can use these arrows.
Okay, and the microphone.
Okay.
Like that.
10 minutes when you start to speak, you start the time.
Okay. So good morning. First of all, I would like to thank the organizers for enabling to present my work. Let's start. Yeah. We are monitoring pomegranate fruit development during in a specific time point to study the molecular and the physiological factors that involved in fruit cracking and splitting, as you see. For simplicity, we divide the fruit development stages into three: early green fruit, fruit at mid-growth when the color changes like a breaker, and at the stage of later stage of pigment accumulation and ripening of the fruit. What we do, we usually during our analysis, we collect only the colored fruit, only the colored skin, without the spongy tissue. And we've been working about it for some years and published works about it. So I want here to present some highlights or something that may be, I think, of interesting to our audience. So first, we did a transcriptome to these three developmental stages of the fruit. And since transcription factors are those that control the activity of the tissue and its characterization, so we extracted from the transcriptome transcription factors and analyzed their activity. First of all, we were surprised that in the early young fruit, we have 50% of the transcription factors. It means that the young fruit is very, very active. And then at 25% at the mid-growth stage and 25% during ripening. The main activities that we can find in the skin of a green fruit is maybe as expected, cell division and differentiation, cell elongation and expansion, cutin biosynthesis, photomorphogenesis, secondary cell wall biogenesis, and signaling. In the ripening fruit, we will find transcription factors that direct senescence, stress, and of course, anthocyanin biosynthesis. So the next stage was to draw full biosynthetic pathways of pathways that we are interested in. Here, for example, is for anthocyanin because it's easy to follow. Let's see if we can show it. Okay, let's show it like that. Next to each gene, we have three boxes that describe the different developmental stages of the fruit. Okay, on the left is green fruit, in the middle would be mid-growth fruit, and on the right would be ripening fruit. Colors denote expression level. Red is high expression level, and blue is low expression level. As you can see during ripening, which is on the right, the side branches of flavonoids and proanthocyanidins are down-regulated during ripening and allow precursor movement into the main pathway of anthocyanin, which is upregulated all the way to provide us with all the right coloration of the fruit, of the skin. So now that we know that our system is working, we'll move on to ask the interesting questions. Okay, so pomegranate, we know it's a non-climacteric fruit, and the model for non-climacteric fruit is strawberry, where ripening is derived by abscisic acid. We were quite surprised to see that in the skin of pomegranate, abscisic acid pathway and signaling is not upregulated. You can see it here along the path. It's not really upregulated along the pathway. On the contrary, we saw a high expression of catalytic respiration, some expression of ethylene biosynthesis and signaling. We saw high expression of polyamine metabolism during this ripening, and also upregulation of jasmonic acid and its signaling. So we concluded that during the pomegranate skin ripening is driven by low ethylene levels combined with polyamines and jasmonic acid, and the involvement of abscisic acid, as in the model fruit, is not. It should be clarified. It's not clear yet. Next, we went to look at genes that synthesize the cutin, because it is said that cracking may initiate from microcracks at the cutin level. So data shows that most of the genes of the cutin are expressed again in early stage of the fruit, not in the ripening fruit. And this was further supported by GCMS analysis of Q10 metabolites. And this data is done with fruit from 3 different orchards that differ with location and microclimate. And you can see the total Q10 peaks at the green stage of the fruit, the very early stage of the fruit, and it declines during fruit development. It's similar in different levels, but similar in all 3 orchards. So reduction in Q10 could result from reduction from low activity of epidermal cells that are part of the skin, of course, and that are producing the Q10 at the cuticle. So we did some anatomical study, and we saw that the density of skin epidermal cells is reducing during fruit expansion. This is results from 2 different orchards. You can see here in the same images that in green young fruit, we have 2 layers of the epidermal cells. It's very crowded in 2 layers. As the fruit expands, this layer spread out into one layer, and when the fruit is ripens, the epidermal cells are even stretched even more. So we concluded, when we combine all the last data that I showed you, that epidermal cell density and Q10 biosynthesis in total amount are reduced during fruit expansion and ripening, and this may increase the susceptibility of the fruit for cracking. So to study further on climate and agroecological factors that may affect fruit cracking, we joined a European project, Horizon 2022. Our project is called CrackSense, and the project aims to address the problem of fruit cracking in citrus, pomegranate, table grapes, and sweet cherries. We are using real-time sensing technologies, and we work at the level of the. So come here. We collect data at the level of fruit level, tree level, orchard level, and regional level. And we do that by the regional level is collecting environmental data by satellites and by meteorological stations. At the level of the orchards, we use drones with different cameras. At the level of the tree and the fruit, we use, you can see sensors of fruit and tree sensors. We use thermal and RGB cameras to photograph the fruits and close. We measure stem water potential and stomatal conductance and leaf area index. So we try to make a comprehensive data collection about the growth. So now this, the work now is still ongoing. People are still working on modeling of the connection between all these factors that we measure with cracking indices. But I would like to tell you some experiments that we did and to show you something interesting about pomegranate. To challenge the situation of cracking, we applied whatever is written in the literature, that addition of extra irrigation will cause cracking of fruit, and reducing irrigation will reduce the risk of cracking. So that's what we did. We made some irrigation experiment. This is the control. In yellow would be high irrigation level with 20% extra more water. And the blue would be a low water amount by 30%. And we just use measured the volumetric soil moisture, and we saw that, you see here we have extra water. It's what I mean, the experimental setting worked. Okay, it worked. We could measure a high water amount and low water amount according to the way we designed it. And of course, in each graph that I will show, I also have the developmental stages of the fruit. Okay, so we measure the stem water potential and stomatal conductance. As you can see, I'm just going through it very fast, that the reduced water amount was really stressed compared to the extra water irrigation to the control. Also, stomatal conductance showed us similar data, that it's really stressed. And extra water was sometimes better or worse than the control, but not always statistically significant. We also can see a significant effect on trunk growth. Okay, we see that the reduced water amount is really, the trunk growth is very, very limited compared to the other two treatments. What I want to show you now, I mean, this is the setting of the experiment. The money time is here because what we saw, the yield of the stress plants was really lower compared to the others, but what we saw that cracking incidence didn't differ between the treatments. So we concluded that the irrigation treatments affected tree physiology and fruit yield, but not cracking incidence. It seems like the pomegranate can manage itself within the stresses and can regulate the cracking incidence or fruit quality. I don't know how to call it exactly. So at that point, I'm finishing. I want to thank my colleagues and thank you.
Can we ask questions? One or two questions. Question?
I just wanted to know, where were the experiments done? In which countries?
In Israel, yes. It's very different orchards with different climates. Okay, some dry, some semi-arid, so.
And all on wonderful.
Wonderful, yes.
Right, please.
If irrigation didn't contribute to any cracking, is there any genetic variable that you suspect that has contributed to cracking, besides only the irrigation and soil moisture?
For now, we don't really know because this was really a question that wasn't really tested, not in Israel, and I think not in other countries. I mean, the effect of irrigation. So we wanted to do that. We still don't know. This is the purpose of the big project, the European big project, because we are going to measure many aspects, including climatic. What we found is high temperature do affect fruit growth and skin quality, but I'm not sure yet if it is related to cracking or not. I don't have still clear
One
answer.
more question. Here.
I would like to go back to your toxicology analysis. Did you also look at genes rather than transcription factor, or you just focus on transcription factors?
No. At first, we looked at transcription factors to see what's going on there, because this shows you a very general picture, and then we went into series of pathways. I'm not showing it here; it was published, but. And I just want to mention the transcription factors. I did manual annotation. It's not enough to do the automatic annotation, but it should be manual and to look into papers to see what it exactly does. Otherwise, you won't get a precise answer.
Thank you. Great, thank you, Leticia. We really appreciate it. Thank you. Round of applause. May I call Simone out here to give us talk.
Okay. Good morning, everyone. Yesterday, you have seen a lot of things about Masseria Frutti Rossi and their products. In particular, they were also interested not only in the production of high-quality pomegranate juices, but also in knowing which could be the impact of some technological processes, such as sterilization, and in particular, the effect of pasteurization versus pasteurization regarding the chemical composition and the biological activities of these juices. So, in literature, there are some reviews related to the effect of high-pressure processing on the food beverages, in particular, only one related to pomegranate juices, which did not reveal any kind of pros and cons in terms of sensorial aspects. But indeed, there is also one study, a Chinese study published in Food Chemistry, which demonstrated that HPP treatment could be better in terms of anthocyanin and antioxidant content, and also on the preservation of the color. But this study has some limitations, and I was not so happy. So we decided to study two different pomegranate juices obtained by Masseria Fruttirossi after two kinds of sterilization processes in order to see if there are some differences which can be due to the change in the chemical composition. So we started analyzing the raw juice samples, two of them through a colorimetric analysis, the only one which could be not destructive and very, very not expensive. In particular, you can see the two different colors, which has been obtained not by direct vision through our eyes, but through a mathematical algorithm, which can also assess some chill-out parameters in order to establish which could be the influence of some specific components on the color. And indeed, as you can see, only the anthocyanins are present, and they can have an impact on the color itself. Indeed, these kind of juices are really turbid, so we have also to assess the importance of the sediment, not only in the appreciation of the taste by the consumers, but also in their impact for the chemical composition, the color, but also the biological activity. Then we moved beyond the classical level, and we studied through HPLC, DLT, the chemical composition in terms of vitamin C, some ellagic tannins, and also ellagic acid itself and derivatives, and also some anthocyanin. As you can see, the pasteurized juice, so PJ, is usually richer with respect to HPP juice, despite some slight differences, but this is also reported in the literature that the HPP process can have an impact on flavonoid and anthocyanin modification. Then we confirmed this analysis also through a qualitative and positive ion direct infusion electrospray ionization mass, and in order to establish a broader metabolomic approach, we also applied NMR, proton and carbon NMR, to these pomegranate juices through monodimensional, bidimensional analysis. And also in this case, we could confirm that regarding some amino acids, some sugars, some organic acid, especially gallic acid, we have a general trend in which the pasteurized juice was richer in terms of amounts of these compounds. And then we also try in chemical analysis in order to assess the total phenolic and total flavonoid contents, and also some in chemical analysis for the antioxidant and metal chelating activities. Also in this case, the pomegranate juice was more potent, slightly more potent with respect to the other one. Then, in order to move, I'm a medicinal chemist, so I'm more interested in also the nutritional properties and the health effects of a food, not only drugs. And in this case, we started also the analysis of some important enzymes important for the human health, and the impact of these juices against these enzymes. We obtained good results against tyrosinase. Then, in order to move on towards preclinical studies on mice, we have to assess some toxicity levels and concentrations. So we started with the in vivo toxicity test, in particular through the determination of the allopathic effect of these two juices against the germination and growth of free plant seeds. And indeed, we could observe only a slight toxic effect, but only at the highest doses. These are important also to assess the specific dose which can be used also in further experiments. We also assess this kind of LC50 also in the branch shift lethality assay. Indeed, pomegranate juice are not toxic, we know, but we have to assess the exact dose for the further experiments. And then we also moved to cell-based assays in which we tested these two pomegranate juices towards a normal intestinal cell line in order, because just the first compartment in which they can have an interaction. We tested these two kinds of juices, and we only observed a slight reduction of cell viability at the highest dose. Then, due to the fact that the pomegranate juice is not so easy to to be tested in a cell model system because it is so turbid, it is very acidic, it is rich of reducing sugars, so also that literature must be taken with particular attention. We also obtained some ethyl acetate extract in order to enrich this extract in terms of ellagic acid, in order to see if there is a correlation. And as you can see, there is a correlation because at the highest dose also of these extracts, you can see that there is a reduction of cell viability as well as also in the pomegranate juice. Then we also tested these juices and their extracts against macrophages without LPS, so as a sort of biocompatibility, and also with LPS. LPS is just an inflammatory stimulus in order to see if there is the possibility for these juices to counteract inflammation. As you can see, this is the control, this is the LPS stimulus, so you can see an increase of cell viability. It is normal, it is physiological. But then, after treatment with the juices, we can observe a reduction of this cell viability, so a counteraction of inflammation. And the same was also for the ethyl acetate enriched extract, in which we could have this kind of counteraction. So probably this kind of anti-inflammatory effect could be due to ellagic acid content. Then we moved also towards in vivo test using zebrafish. These tests are related to the possibility to modulate anxiety, but also to check some cognitive effects. Indeed, you can see this is the noxious stimulus, this is the mexazolam, so a positive control to treat anxiety. And as you can see, this kind of juices, despite the sterilization process, can counteract the noxious stimulus and can improve, for example, the time spent in the upper zone of the tank for the fish, just very close and very, very statistically significant with respect to the positive control. The same also for the locomotor activity. Then we move to the novel approach test. In this test, we put an object in the center of this tank. Usually the fish could be anxious in order to reach the object, but it could also be curious. It depends on the attitude and on the mood. Indeed, as you can see, if we have a lot of time spent in the inner zone, it means that the fish has no anxiety towards the unknown object. And whereas if we have a very high time spent in the outer zone, it means that the fish is very, very anxious about the object. So as you can see, also in this case, with respect to the noxious stimulus, we have that our pomegranate juices are tend to counteract this anxiety and try to reach also the levels of mexazolam as positive control. Then we have the YMS spatial memory test. Also in this case, the fish is able to study two different arms, and then we open a new arm for him or her. And then we can check if he or she wants to stand in the known arm or wants to explore a new one. So also in this case, we can observe the attitude to respond to a novel arm, for example, to recognize the known arm, so the spatial memory, but also the movement, because it's not easy for him or her to go over the angle and to see what there is beyond. Indeed, in this case, as you can see, the positive control is galantamine, so something which is important to inhibit acetylcholinesterase, so important for cognitive effect. And also in this case, we can see that these pomegranate juices can restore this kind of activities, it can improve the spatial memory, and can improve the response to novelty. Then we also applied this kind of novel object recognition test. This is the last one. And in this case, we put an object and we let the fish to recognize him. Then we put another object, a new one, and we measure the time that, or the preference that he or she has for the new one. Also in this case, galantamine is the positive control. As you can see, the pomegranate juices directly dissolved into the water tank can counteract the noxious stimulus and can reach the same level, also with the same level of statistical significance, the preference towards the novel object. So, final considerations. It is important the role of the consumer in terms of color, taste, and so on, but it's also important to know which parameters can impact on this kind of food beverages. And indeed, the HPP process is a good process, but must be carefully evaluated in terms of parameters. And also it's important also to study the amount and the composition of the sediment, because it is a characteristic trait in pomegranate juices. Then we are moving towards studies on marine models of health and disease, and also we are studying the impact and the metabolism given by our gut microbiota, and we will also study the impact of other treatments on the composition of this pomegranate juice. Let me only thank all the people involved in this project, so people from Roma La Sapienza, and in particular Professor Stefania Cesa, my research group, and also the university, some university in Romania for the experiments, in vivo experiments, and naturally Masseria Frutti Rossi for the economical support. Thank you so much for your attention.
Thank you very much, Simone. Do we have questions? Questions? I have one question. You have the PJ, which is the pasteurized, and the HPP. Do you think it's important to also have the one that you did not treat just pomegranate juice?
Yes, as I said, it is important also to see just the pomegranate juice without any kind of just before this kind of technological step. Yes, this is part of our next experiments. Yes.
Okay, great. Yes, go ahead.
This is just a curiosity, Simone. So, do you think in the near future we can use the pomegranate juice for treating anxiety for human application, or maybe as an additive in medicals?
This is a hard question. As a medicinal chemist, I am trying to move from food to drug. So the interval, the gap is just nutraceuticals or food supplements and so on. I think that at the moment we are moving towards in vivo studies, and we will see the impact on murine models also of disease, and we could see, assessing the correct dose, if there is an impact on this kind of disease. Maybe I can see something like coadjuvants and a correct diet in order to prevent this kind of, maybe the onset of this kind of disease, but it is a long way too. It's not easy. But in what I can say that in literature, I have heard a lot about the activities, healthy properties of pomegranate and also other fruits. Yes, they have this kind of activities, but you have to study very well the articles, because indeed there are no information about some tests. Indeed, pomegranate juice are very, very strange to be tested because also the protocols are very, very rigid. But indeed, there are some aspects which can be further explored. Yes.
Great. Thank you very much. Let's move to the next presenter. Thank you. Can you give me Brian will come through for the next presentation. He will be talking about post-harvest storage of pomegranate.
Hello, everyone. Dear Chair, dear commoners, thank you for this nice conference. And today I would like to present a speech about the effects of different treatments on the storability of pomegranate. It was a part of the master thesis of my student Osman Konut, and I'm doing this presentation together with my colleagues Erdus Anmaz. I can say that it's the first part of this studies. It's a preliminary study. The result of this study will be a way for the coming studies. As we all know, and we are mentioning since yesterday, that the pomegranate has a rich history, and it's among the first 5 fruits that is being cultivated. But for a long time, we know that it had little consumption, not little, but less consumption than the other crops. But after its health benefits are being confirmed, scientifically confirmed, the evidence made an increase in the both consumption and production of the pomegranate fruits. But at that time, of course, the hustle of extracting the areoles was another story that causing the less consumption of the fruits. Different studies are being carried, some to develop some varieties or cultivars that are being easily extracted, or some other different kinds of studies. All of them are aiming to improve the both production and consumption of the pomegranate fruits. But another important issue that we are facing, and my other colleagues, Prof. Ali and Prof. Favole, are also mentioning that because of the short periods in the harvest and differences in the southern and the northern hemisphere, the year-round existence of the fruit in the market can only be achieved by the storage activities, so prolonging the storability of the pomegranates. So some cultivars are adapted to different climatic conditions, as we all know, and we all know that it's a vulnerable crop because of its different characteristics. One of them is having lots of pores on it. The main problems that we are facing are weight loss, chilling injury, and microbial decay, as we can say. We have different options, some options to solve these issues. We are using the optimal storage conditions, relative humidities. Of course, we are using some other techniques like coatings, waxings, essential oils, chitosans, controlled atmospheres, or modified atmosphere packaging. We can say that since the beginning of the 21st century, the melatonin is being used for different purposes and for improving the storability of the crops. In my other studies also, I see a high impacts on the strawberry crops. It's reducing the decay, delaying the senescence, and mitigating the chilling injuries on different crops. And we all know that the modified atmosphere packaging is a good way for improving the storability. But at that time, we have been thinking that some essential oils, but with a different way of application, may cause the change in some enzymatic activities. But this is the limitations of our study, that we didn't make any tests on the enzymatic activities. Our aim was to see if we have some external or visual impacts, and then we're gonna test some enzymatic activities. We tried to test the carnation oil together with the modified atmosphere packaging as a combined strategy. And also the sulfur dioxide we have included in our studies, especially because of its well-known impacts on the Botrytis cinerea. So we have the modified atmosphere packaging, the melatonin, carnation oil, the combination, and SO2 generating parts. The fruits were all collected from same field, 14 years old, pomegranate orchard with wonderful varieties in northern Cyprus in 2024. The modified atmosphere, I'm gonna pass these details about the materials, just to have an idea here. The applications of the carnation oil was done as the 2 drops, approximately 0.5 milliliters of oil, by applying inside to the crown, and putting them into the modified atmosphere packaging, and the melatonin dose was selected as the 500 micromolar. So we have the control, we have the modified atmosphere packaging, we have its combination with carnation oil and melatonin alone, and SO2 generating pads. We use the randomly complete block design with the split plots for the each storage duration, 30 days, 60 days, 90 days, 120 and 150 days, 5 replicates and 3 fluids in each. So totally 450 fluids we have used. The quality assessment, the parameters that we have tested, the weight loss, soluble solids concentration, the table acidity, ascorbic acid, the decay incidence, we use the 0.0 to 3 scale, and for the chilling injury, we use again the 0 to 3 scales. The data obtained was first rolled in the Microsoft Excel, then in the SPSS to test the significant differences among the different treatments. We use the analysis of variance and the Duncan multiple range test, and the graphical explanations were done with the Microsoft Excel too. Here we see the change in the weight loss as expected. The treatment with the modified atmosphere packaging has higher impacts on reducing the weight loss during time. But at that time, we have seen that the application of carnation oil and melatonin, so combination with modified atmosphere packaging, gave some additional impacts on it, but it's not statistically significant. This is the thing that we were not expecting. We were expecting some statistical significance, but it was not existed. Here we see the impacts on the ascorbic acid content. It was also, as expected, highly reducing the control treatments, and it was stored a little bit more in the modified atmosphere packaging and together with the melatonin application. Also, we can see that the melatonin application alone is a way that can be used to prevent some quality indices on the fluids. A little, but we can say that no significant differences obtained on the impacts of these treatments on the soluble solids concentration, as it was little change in the time, but no significant differences in the end were noted. Here we see the results on the table acidity. Again, the highest reduction during the storage we obtained from the control treatments, and as with the other weight loss and the other other quality indices, the modified atmosphere packaging together with carnation oil and the melatonin had high impacts. The melatonin application alone also had little, but it was also less than our expectations that we had obtained in other fruits like strawberries or the others. Of course, on the decay incidents, we had some good results from both of the applications. One of the most promising things for me is that for us is that the melatonin alone also had more or less similar and statistically non-significant impact together with the modified atmosphere packaging. And for the chilling injury, again, both the modified atmosphere packaging, its combinations and melatonin was found to have the highest impact, as we were expecting. A few slides. Here are some images from the studies. These are from the 150 days, the insights. And as I said, we had some limitations, especially in terms of the quality parameters that we have tested. It was a master project of our student. We think that the combinations, especially the combination of modified atmosphere packaging with the melatonin, is more impactful and more we can say that it confirms the synergistic benefits of the combining modified atmosphere packaging with some other techniques. Thank you very much for listening and
thank you very much, Ibrahim. Do we have questions? One or two questions for Ibrahim. Question? Yes, go ahead.
If they were to use melatonin, is it expensive? Are there like non-pharmaceutical grade sources of melatonin that could be used in post-harvest?
There should be some studies to see the costs and the benefits, but for pomegranates and with these results, I can say that it's expensive. But for strawberries, the results that I have received before, I can say that because it's a less duration for the strawberries, we can say that it's economical. But for pomegranates, with these results, it's not.
Thank you for your presentation. I wanted to know the decay that you referred to. What was the decay? What were you getting in that story? Because I see your decay was at about 60% in your controls.
Yes, it was from the botrytis, and we didn't do any treatments for the fruits, for the control treatments. So no washing and no other things, no dippings, no cleaning, just introducing from the orchard to the cold storage.
And if I have another question?
Yes, go ahead, please.
I would be, when you used your SO2, the sheeting uses, did you see any signs of phytotoxicity?
We didn't observe, but some studies, I read some studies from Professor Mustafa Erkan from Akdeniz University. They mentioned that there are some phytotoxic impacts. More or less, we carried these studies similar times, and they observed.
Thank you.
Thank you very much. We will move to the. Can we keep the right round of applause, please? I will call on Mario. Is Mario here? Yes. Mario will give us a talk on analysis, comparative analysis of pomegranates.
Anche. Sì.
Good morning, everyone. My name is Mario Riguardo. I would like first to thank the company for giving me the possibility of presenting my work. Actually, this is our work, I would say. It's mainly the work of our former PhD student at the University of Catania, that is Paolo La Spada, and I'm today presenting his work. So just to show you a brief overview of our research group. So we are focusing on several aspects, focusing on Mediterranean tree crops, ranging from new breeding techniques, omics analysis, and I am a bioinformatician, so I am mainly involved in these tasks. Then agronomical trials and the characterization of plant biodiversity. So, pomegranate was characterized by an increase in production, both in terms of production and harvested area, and the main productive regions are Sicily and Apulia, but also the Veneto region. In this area, the cultivation took the place of more traditional cultivations, such as citrus, for example, or table grape. The new orchards relies on the availability of elite cultivars that were developed by specific breeding programs or by the selection of novel accession showing quality traits of interest. In particular, in the latter case, since pomegranate in the different cultivation areas is characterized by a wide variability, both in terms of genotype and phenotype, and this variability arose from the seed propagation that was used in history to propagate pomegranate, that was also coupled with easy vegetative propagation by easy vegetative propagation and also by the cross-pollination. So in this case, it is important to have ex situ germplasm collection to help this variability and to study this variability. In this example, for example, I'm showing you the germplasm collection that is held at the University of Catania, in which we are maintaining some local varieties coupled with some of the most widely cultivated international cultivars. And actually, this work is aimed at the analysis of two local varieties, both in terms of fruit quality and in particular morphological traits and texture of areoles and tegmen, but also we assess their susceptibility to cracking. And actually, these two local varieties were compared with a clone of Wonderful due to its wide cultivation worldwide. So, the two local varieties that we selected are Etna, that was developed by a local farmer and a local breeder, that is Gaetan Tirò, and the other one is Primo Sole, that is one of the local selection that is maintained at the University of Catania. In terms of fruit quality, we measure 10 different carpometric traits that are taken from the descriptor list for pomegranate. In particular, 6 of these traits are related to the fruit, moving from the fruit weight, the diameter, the height, and the others are related to the length and the thickness of both the areoles and the tegmen. These data were collected over 3 consecutive years in a commercial orchard owned by Gaetan Tirò again. So here in this PCA, we can see how these 3 cultivars behave. And in particular, the first principal component that shifts the sample from left to right is mainly influenced by these arrows over here, so that are related to the diameter, to the length, and to the weight of the fruit. In other terms, the fruit size move from Etna, there is the smallest one, to Primo Sole, and to Wonderful. We can also observe by looking at the projection of these arrows how these traits are correlated. On the other side, the second principal component that explains a lower percentage of the total variability, the 22% against the 77%, is interesting since it allowed to discriminate Primo Sole compared to the other two in terms of the size of the areoles, because the A and the L traits are related to the length and thickness of the areoles. So Primo Sole has bigger areoles compared to the other two cultivars. Then we moved on and we also looked at this cultivar in terms of texture. We used a texture analyzer, and also here these analyses were carried out for 3 consecutive years on 10 fruits per cultivar. We examined both the areole and the tegmen, and the first trait that we looked at was the hardness. And actually there is the force needed to a probe to penetrate inside the food matrix. And in this case, we can see that we didn't find any statistical difference between the two local varieties, Etna and Primo Sole, neither in the areole nor in the tegmen. While in both cases, Wonderful showed harder areole and harder tegmen. And actually, this result was also confirmed by the second trait we look at, that was the negative peak distance, or in other words, the negative force needed to a probe to go back to its initial position, so from the food matrix to the initial position. And actually, in this case, we didn't find any differences among the 3 cultivars for the areole, but again, we found a higher negative value, in this case, for Wonderful compared to Etna and Primo Sole. So this means that Wonderful in general has harder areoles, but also tegmen. And this could also hamper the use of Wonderful for fresh consumption. In addition, we also find that Wonderful has a larger size compared to the other 2 local varieties, but also the Primosole, even though it's characterized by the intermediate fruit size, showed larger areas. At this point, we moved on and we started to the consideration that there is an increasing wealth of data and empirical observation that Primosole has a higher susceptibility to cracking compared to most of the other varieties. Cracking often occurs during the last stages of fruit maturation, and it occurs more or less in all the different cultivars and seems not to be really linked to different areas of cultivation. It is very difficult to study fruit cracking since it is influenced by many factors, ranging from the weather condition, the cultivar, of course, so the genotype, the fruit developmental stage, and the agronomical practices. In practice, during a drought period, the peel loses its capacity to divide and expand. But then, when the plant is watered again, the meristematic tissues rapidly resume growth, while the same do not occur for the peel tissues, and actually this will result in cracking. Over the time, there are several studies trying to link several morphological aspects of the fruit to a higher or lower susceptibility to cracking, such as the fruit size, the fruit shape, but also the physical properties of the cuticle, for example. And in this context, we moved from this assumption and we start to look at the fruit cuticle on these three cultivars. So we selected, in this case only for 1 year, 3 fruits for each cultivar, and the cuticle was assessed by sampling the fruit in 3 positions, at the apical part, in the median, and in the basal part. So first of all, we were interested in looking at the differences within each cultivar. But actually, we didn't find almost any differences. So if we look at the data column by column, we didn't find any statistical differences between the apical, median, and basal part of the cuticle thickness. But for one case in Wonderful, where we did find the differences between the apical and the median part of the fruit, and this is probably due to the fact that the fruit is larger, but in any case, doesn't seem to be linked to a higher susceptibility to cracking. Then we moved on and we started looking at the cuticle thickness as a whole, so trying to look at the differences between cultivars. And in this case, we did find differences, with Etna having the higher cuticle thickness, followed by Primosole and then Wonderful. But also here in this case, since Primosole is the cultivar with the highest susceptibility to cracking, it has an intermediate cuticle thickness, so we also tend to discard the hypothesis that the cuticle thickness itself could be linked to a higher susceptibility to cracking. So then we moved and looking at the optical microscope images, and actually in red, in pink, we can see the cuticle, and actually what we did find different between Wonderful and Etna and Primosole is the extent of the cuticle invagination in the first layers of the cell. So we postulated that since Etna and Wonderful are showing larger invagination of the cuticle, this could increase the cuticle surface, making the fruit more resistant to cracking and to abiotic stress in general. Of course, this is a pilot study that is based only on 3 cultivars, so we are planning now to extend this work also on other cultivars in order to try to prove or disprove the hypothesis that the cuticle invagination could be related to a higher or lower susceptibility to cracking. In this slide, I finished my presentation. I would like to thank all the people that were involved in this project and the POVAX project that financed this work. Thank you.
Very interesting work. Do we have a question? Do you think in terms of liquid?
Maybe there are one?
I'll get to it. did you look into nutrition like element factor that might influence the integrity of the criticals, maybe calcium or something?
Not yet in this case, but we are planning to do this on a bigger study. Yeah. So we only focus on the optical images in these cases, but we would like also to extend also with some analysis like this one. Yeah.
Yes.
Okay.
Because we have the Pomsym infection, which one has. So that the impression of practical point of view is the productivity is very low, very low. But the stage of maturity is very low. Could you confirm or not these first impressions?
To be honest, I cannot confirm this because actually I was more related on the analysis of data rather than on this. Actually, these cultivars is still, in a productive orchard. So actually they are cultivating it and they are selling it. So actually I don't have now by heart, I don't know by heart this information, but I don't know, Stefan, if you want to add something, but.
I think you already saw also the plants of Pomsym, right? In our condition, we are not complaining about the productivity of these varieties. But I understand that in other fields can be a problem, and it depends on the local condition.
Any last question? I have one other question. Yeah. You've got these three cultivars, and are they all at the same harvest period? So the maturity stage, you were able to compare. Okay.
Yeah. They were all at commercial maturity. So actually.
And the critical characterization, was it done before the cracking incidents or after? Before, so you know.
Yeah.
Great. Thank you. Let's get that last question.
How's the internal color of this?
The internal color, you mean?
Iris.
Internal
color of the iris.
Okay, I get it. Actually, they all pigmented. So actually, we also have this data. I didn't show you this data today, but we assessed this using colorimeters. So actually, they were all pigmented. All the three varieties. Wonderful was more pigmented than Primosole, while Edna showed an intermediate pigmentation.
Great, thank you, man. Thank you very much. So we welcome Maria to the stage to speak about the effect of environmental condition on ripeness at harvest of pomegranates.
First of all, I want to thank the organizers for the invitation to present this work that was carried out at the Valencian Institute from Agricultural Research and the Agricultural Extension Center in Elche. Spain is the first producer of pomegranates in Europe, the producer and exporter with around 90% of the total production located in the southeast area, in the area of Elche and Alicante, that area. The main cultivar is Mollar de Elche. This cultivar is characterized by its outstanding quality that is given by low acidity, high sugar content, and soft seeds. Producers of Mollar de Elche are very interested in extending the storage period for fruits destined to export markets. However, weight loss, chilling injury, and post-harvest diseases are the main problems limiting the cold storage of this cultivar. For this cultivar, storage at 5 degrees Celsius and a relative humidity above 90% is recommended to reduce chilling injury although this chilling injury is not completely preserved, especially when you have long-term cold storage. As you all know, a chilling injury is manifested as with a external superficial pitting, and also internal browning of the membrane and the spongy tissue surrounding the areas. However, in recent seasons, increased internal browning of the sponge, internal browning of the membranes and the spongy tissue has been observed in the absence of external superficial pitting, which is also a characteristic of a chilling injury. Furthermore, this internal browning becomes apparent after a few weeks of cold storage under cold optimal storage condition, 5 degrees, without further storage at 20 degrees. This means that when the fruit is taken out from cold room, these symptoms is already manifested. Preliminary work done by the Valencian Institute for Agricultural Research and also the experimental extension in Elche, although we have seen that although this internal browning seems to be associated with agroclimatic factors, and here the orchard is a key factor, there are other factors such as harvest date and storage conditions that also have an effect on the incidence and severity of internal browning. Therefore, the objectives of this work were to study the effect of environmental conditions at harvest, and for this, harvest was done early in the morning with mild temperatures, and also late in the morning with maximum solar radiation. And also to study the effect of harvest date, either at the beginning and at the end of the season, on the physicochemical quality and internal tissue browning of Mollar de Elche pomegranates during long-term cold storage at 6 degrees, followed by a 7-day shell-like period at 20 degrees Celsius. Fruit were harvested from a commercial orchard in Elche at the beginning of the season and at the end of the season. And to evaluate the effect of ambient conditions, fruit were harvested early in the morning or late in the morning with maximum solar radiation. And at harvest, we evaluate the environmental conditions and also fruit temperature. The effect of ambient temperature differences between fruits harvest early in the season and late in the season were minimum, while relative humidity was slightly lower in fruits harvest at the end of the season. When comparing the effect of harvesting early in the morning or late in the morning, differences were significant, difference in temperature and relative humidity. This difference in the case of ambient temperature range between 14 and 15 degrees Celsius, and in the case of relative humidity, the difference were around 30%, 32%. This difference also translates in differences in fruit temperature that were significant and were around 14, 15 degrees Celsius between fruits harvest early in the morning and late in the morning. The fruit was transported to the IBIA facilities, distributed into the different treatments, and the fruit was stored at 6 degrees Celsius and for 8 and 12 weeks, and a relative humidity between 90 and 95%. And this cold storage conditions was followed by 7 days at 20 degrees Celsius. Evaluations included external disorders, pitting, and dehydration, internal browning, and for this we used a 5-point qualitative scale where where 1 means no symptoms and 5 represented more than 70% of the surface affected. We also evaluate electroleakage, juice quality, external ring and juice color, weight loss, firmness, and respiration. At the end of harvest, the incidence or severity of pitting due to chilling injury or dehydration was minimum, and we didn't see or observe any significant differences regarding or due to harvesting conditions. However, browning, internal browning was already observed after 8 weeks of cold storage. For both cold storage conditions, the severity index, the browning index, ranged between 3 and almost 5. This means that more than 50% of the surface was affected. We didn't observe, overall, we didn't observe any effect of whether the fruits were harvested early in the morning or late in the morning. However, fruits harvested at the end of this season show lower in browning index than those harvested at the beginning of the season. Furthermore, the number of fruits that were evaluated or ranked with the highest scores in the browning scale were lower in the case of the fruits that they were harvested at the end of the season. Electroleakage increased with the storage with the storage time, and basically what we observe overall, those fruits that were harvested at the beginning of the season show higher electroleakage, which correlates with the higher internal browning that we observe. And this implies also that there was a loss on the cell membrane integrity of these fruits. Juice quality was not significantly affected by harvesting conditions. These two graphs show the hue value of in the ground area and the red-bluish color area of the pomegranates. These two areas were differentiated because Mollar de Elche pomegranates don't have a uniform color. At harvest, the hue value, actually in the red-bluish color, those fruits harvested at the end of the season show lower hue values than those harvested at the beginning of the season. And there were no significant differences between whether the fruit was harvested early in the morning and late in the morning. This lower hue value indicates that they has had a redder color. In the case of the ground color, the ground area color, however, only we observe significant differences only between fruits harvest at the beginning or early in the morning and late in the morning. Okay, well, okay. With those harvests late in the morning having higher hue values, more towards the yellow color. Okay, this is a juice color, and in general, what we observe is that the hue value of fruits harvest at the end of the season have lower hue color. Weight loss increased with storage time. After 8 weeks of cold storage, we didn't, well, there was some significant difference, but we didn't find any conclusion. However, at the end of after 12 weeks of cold storage, with and without the shell-like period, those fruits that were harvested at the beginning of the season show higher weight loss, which also correlate with the highest internal browning that we observed. Fruit firmness was at harvest and after 8 weeks of cold storage was higher in those fruits that they were harvested at the beginning of the season. After 12 weeks, we didn't observe any significant differences between treatments. The fruit respiration was affected by harvesting conditions and also storage time. However, the behavior differ depending on the storage conditions. This makes difficult to draw any conclusion. Nevertheless, if we see the results after 12 weeks of cold storage plus the cell life, we see that we can see that the respiration was higher in those fruits harvested at the beginning of the season, and also this correlates also with the higher electroleakage and also a higher internal browning. But nevertheless, this needs to be corroborated in further studies. So in conclusion, we can say that although internal browning of Mollar de Elche pomegranates appear to be associated with pre-harvest factors, the results highlight the importance of harvest date on the incidence and severity of this physiological disorder. Overall, late harvest fruits were less susceptible to internal browning than early harvest fruits, and future work should explore the origin of this physiological disorder and feasible pre- and post-harvest management to minimize it. And then I want to thank all the group that work in this research from the IBIA and also from the experimental extension in Elche, and also, of course, the funding agencies or the funding projects, actually. Thank you.
Thank you very much, Maria. Do we have a question for Maria? One or two questions? Go ahead.
Thank you. If you have chilling injury at 6 degrees, so why don't you spot the fruit at, let's say, 8 degrees or 10 degrees? The recommendation of 6 degrees was for wonderful variety, but maybe this variety needs a bit higher.
No, let's see. Mollar de Elche, the optimum temperature to reduce or to reduce the incidence of chilling injury is 5 degrees Celsius. Okay, and is the one that is used at commercial scale in the packing houses. Now we have also done or studied the effect of storage at 7 at 10 degrees Celsius, and we have seen also that the incidence or severity of this disorder is decreased. The point here is that we have not observed in any of the cases pitting, external pitting due to the chilling injury. So we are still just trying to, and also one of the key points here is that we have seen an orchard factor. I mean, fruits from different orchards have different severity or different internal browning. There are orchards that is difficult. Well, now more or less is kind of generalize this problem, okay? But we have seen that there are other factors, pre-harvest factors. The point is that post-harvest factors that we can control, such as temperature and so on, also have an effect on this internal disorder. And we are still studying.
One comment that also in Israel this year with the early varieties, for some reason the fruit were more susceptible to chilling this year compared to previous years, and also we also see differences between plots.
Mm-hmm. Okay.
So very similar to what you also.
Yeah. Well, in Spain we are observing this not this year, but for the last 3-4 years, more or less. Okay. Mm-hmm.
Maybe next year you will observe it.
Okay. Let's see.
Any other question? Go ahead, please.
Good morning, but if you harvest at 9 to 11 in the morning, you can notice quality differences on the berries. So why didn't you notice any difference between early harvest and late harvest during the morning? Do you think because of the structure of the fruit, which pomegranate fruit is different from a berry?
Okay, I think it means I don't know if I understood correctly your question. You mean
the quality difference if you harvest early in the morning and late?
We didn't observe such a high difference.
Well, I'm asking why didn't you observe any difference?
Okay, I think this. I don't really know exactly right now, but I think those differences also will affect the time also to, I think internal quality is difficult to be affected by whether or not you harvest early in the morning and late in the morning. It might affect more whether or not you harvest at the beginning and at the end of the season. And in this case, we didn't even see that such effect, okay? Regarding color, external color, we observe those differences in the ground color, okay? But not in the internal color. Is more difficult sometimes to observe these differences.
Maybe because of the peel, probably.
Yeah,
is different.
exactly.
Yes.
And also,
one question I had, I work with Moladeshe.
the what?
Moladeshe.
Mola.
Mola. This one is.
Moyar.
Moyar, yeah.
Yeah.
This call is the same cultivar.
Yeah, this is Moyar de Elche.
Moyar de Elche. So the one I worked with when it was still being grown in South Africa was quite yellow.
Mm-hmm.
But yours is more red.
Well, it doesn't have a uniform color, okay? So you find, maybe you see, I don't know, you can go back to the first picture that I showed with the aspect of the Moyar de Elche after 12 weeks of cold storage plus the cell life. You can see a high variability. It's not well, it has a red area, but it's not uniform.
It's not uniform. No.
But it's not completely yellow either.
Okay. Good. Thank you. Thank you very much. Our last presenter before we break for coffee, Angela will present on pomegranate juice fermentation.
Good morning to everyone, and thank you, Professor, for introducing me. As we all know, thank you. As we all know, pomegranate is largely cultivated in the Mediterranean basin, but is consumed all over the world, not only as a fresh fruit, but also as a jelly juice or alcoholic beverage. The consumption of pomegranates as a fresh fruit or as a juice has had increased in recent years, also thanks to the higher consciousness of the consumer connected to the potential of a pomegranate, considering, for example, the nutritional values or also the presence of many bioactive compounds. So we can say that increasing demand of pomegranate has also generated a high byproducts, mainly seeds and peels. And today I just want to focus my attention on seeds. We know that pomegranate is rich in valuable compounds, but also seeds. They are rich in some carbohydrates and protein, lipids, fiber, but also in some minerals such as calcium, iron, potassium, and zinc. Fermentation process, we know that might provide new potential application for the valorization of byproducts, such as, for example, pomegranate seeds, but also with the aim to produce novel food beverage, for example, such as low alcoholic beverage, starting, for example, from pomegranate juice. Moreover, we know that pomegranate is a very challenging matrix because of the low pH, but also for the presence of many antibacterial compounds. For this reason, we hypothesize that maybe the fermentation to an autochthonous microorganism could be a very important alternative to consider, because for autochthonous microorganism, I mean microorganism isolated from the matrix. So the aim of the studies was the first, the microbiological characterization of pomegranate juice and seeds, that to the best of our knowledge, no one has already done. The second aspect regard the valorization of pomegranate byproducts, so pomegranate seeds, in order to obtain a nutritional ingredients that could be added to large-scale consumption food. And the third aim was connected to the production of a cider-like beverage, so a low alcoholic fermented beverage starting from pomegranate, thanks to the use of autochthonous microorganisms. As regards pomegranate microbiota, we can say that it's largely dominated by yeast that are probably environmentally born. And on understanding of the use of specific media for bacteria, we do not find bacteria at detectable cell density. For this reason, in order to isolate potential technological microorganisms that were able to ferment matrix, we try to isolate microorganisms starting from spontaneous fermented juice and spontaneous fermented seeds. After the spontaneous fermentation, only yeast were detected and were all the isolate biotype had identified. After the identification, all were identified as Ancinospora valdiensis species. We have 4 different representative strains, 3 isolated from pomegranate juice GL1, 5, and 6, and 1 isolated from seeds, so Ancinospora valdiensis SL1. And it is in this picture, I just want to show you the biotyping. As we can observe, they are very far from each other. This suggests that we have 4 different strains of Ancinospora valdiensis. We know that Ancinospora valdiensis is a non-conventional yeast that is often found during the first stage of fermentation, but is known also because it's a microbial driver for balsamic vinegar and cider fermentation. In some studies, some researchers try to use Ancinospora as the main starter for a driven fermentation, for example, for the production of low alcoholic beverage, but also for the production, for example, of cider. But it's important to underline that for the moment, Ancinospora is not recognized with QPS status, so we cannot currently use this yeast for industries fermentation for the production on a large scale. We try to ferment pomegranate seeds thanks to the use of Ancinospora valdiensis, and after the fermentation, the seeds were dried and grinded in order to obtain a flour that could be added as an ingredient in the production of granola snack. And as we can observe, we had a percentage of 22.2%. We obtained 3 different granola, 1 control without the addition of pomegranate seeds flour, 1 with the addition of pomegranate seeds flour unfermented, and the 3rd one with fermented pomegranate seeds flour. And then, of course, we characterized all the granola snack. As we can observe in this table, Ancinospora valdiensis is absolutely able to grow in the matrix, showing an increase on 1 logarithmic cycle after 24 hours. The most interesting results regard the mineral composition, because as we can observe in the thesis SL1, so fermented with Ancinospora valdiensis SL1, we have the highest increase in mineral, especially in calcium, potassium, iron, and zinc. That is maybe due to the higher acceptability and availability connected to the fermentation process. In parallel, we observe also a decrease of ash shift that maybe could be due to a decrease on a leasing of soluble salts. As regards the antioxidant activity, we can say that the antioxidant activity is estimated in terms of the ppH and FrAC decrease during the fermentation. And this could be due to the production of acetate from Ancinospora valdiensis during the fermentation. But remember this result because later I will talk again about the antioxidant activities. In this picture, with this picture, I just want to show you the 3 granola snack. As we can observe, it's very hard for us to understand which one is the control and which one is added with pomegranate seeds flour. Just a spoiler, the 3rd one is added with pomegranate seeds flour fermented. And as regards the panel test, we can say that panelists prefer the thesis added with fermented seeds flour compared to the unfermented one, because it was evaluated as sweeter and with a lower presence of particles difficult to swallow. As regards the antioxidant activity of the granola snack, the situation is a little bit different compared to the graph that I showed you before, because in the thesis added with pomegranate seeds flour fermented or not, the antioxidant activity is higher compared to granola control, because despite of the fact that antioxidant activity decreased during the fermentation, the antioxidant activity of seeds is always very, very high, and so have an impact on the final products. As regards the expected nutritional values, we can also say that we hypothesize that the granola added with pomegranate seeds flour will be richer in some mineral and fiber, and also will be characterized by a lower kilocalories. The last part of my presentation, I want to show you the part that regards the production of cider-like beverage. We know that the industry in general prefer the use of conventional starters such as Saccharomyces cerevisiae, because of course it's well studied for a high fermentation efficiency and also for its matrix adaptation. On the contrary, on the other hand, we can say that non-Saccharomyces yeasts are able to ferment challenging matrices and moreover can provide particular flavor characteristic to the final products. For this reason, we purchased from the retail 2 different pomegranate 100% juice that differed for the percentage of sugar, but as you can observe, the difference was very low. After, we tried to ferment pomegranate juice with the autochthonous starter Ancinospora valdiensis or in parallel with the commercial starter Saccharomyces cerevisiae in order to obtain 4 different cider. After the fermentation process, start the maturation process. or considered as is, so untreated, or after certification of filtration treatment. After the filtration and after the maturation, all the cider were characterized. In this graph, I want to show you this aspect is very important. We have a row of antinesporal is very important in the medium in the juice, more than 2 logarithmic cycle compared to the control. This suggests the higher matrix adaptation of antinesporal albiensis compared to the control. After 7 days of fermentation, this gap is very low. And considering treated cider, we can say that treatment, of course, have an impact on cell density, but it's unexpected results because one of the aim of the treatment was to reduce the cell density of microorganisms in order to reduce the risk of spoilage and unpredictable changes, of course. Because as we can observe, after 7 days of fermentation, glucose and fructose are not added, and this could be a problem. But we can observe that antinesporal albiensis is faster in the consumption of glucose and fructose. After the 7 days of maturation, all the sugar was completely ended. As regards the acetic acid and ethanol production, we can observe that antinesporal albiensis is better able to produce acetic acid, and in fact, is higher concentration in cider fermented with antinesporal albiensis, and the ethanol is higher in thesis fermented with the commercial starter. The maturation, of course, have an impact on acetic acid and ethanol that decrease, considering the most stressed treatments such as centrifugation and filtration. As regards volatile organic compounds, we can say that cider fermented with the non-conventional starter and antinesporal albiensis are richer in organic acid and the state of ester of organic acid that are responsible for fruity notes. In contrast, cider fermented with conventional starter are richer in hexanoic and ethanoic acid that sometimes are connected to the sweets or to the rancid flavor. Considering the impact of the treatment, we can observe that in general, cider after the treatment are poor in some volatile organic compounds, but overall, antinesporal albiensis are characterized by a more complex profile compared to the control. In this slide, I just report the 4 different theses. The second and the fourth are fermented with the non-conventional starter. In conclusion, I just want to say that pomegranate microbiota was largely dominated by yeast, all identified as antinesporal albiensis. And we know that the use of non-conventional yeast such as antinesporal albiensis is an important strategy also for the fermentation of challenging matrices such as pomegranate. The studies also highlight the potential of antinesporal to convert pomegranate seeds that are commonly considered as a byproduct or in some cases waste into nutritionally valuable ingredients that could be added in a large-scale consumption food. Moreover, antinesporal promotes the formation of volatile organic compounds that are very important for also for when we want to drink something. Our alcoholics are mainly the main characteristic that we want and we research. We also know that antinesporal actually could not correctly be considered as a starter for fermented food due to the lack of QPS and GRAS status. But these works contribute to a deeper understanding of non-conventional yeast in food fermentation and provide insight that may inspire also the development of innovative sustainable products. I just want to many thanks to my supervisor, to my group, and to all of you for your attention. Thank you.
Thank you very much. Such a very detailed and comprehensive study. Do we have a question for Santiago? Question? So this the yeast are native to the pomegranate here. So the one that you use in the fermentation is predominant. Is it the predominant or the fermenter, the most fermented?
It's the predominant, and it's the only that we try to isolate different technological microorganisms, not only yeast. We isolate, of course, different microorganisms, but all were identified as antinesporal albiensis, so it's the main.
Because there's one on one hand it could be the predominant, but it might not be the most fermented.
For sure.
So it is the predominant.
It's the predominant,
and also it's the most active.
Because it's the only one. It's also the most fermented.
Do we have any? I had a PhD student that did similar stuff. She graduated last year. So that's very interesting. How we all thinking alike, more like competing on doing. Yeah. Did you also try different location where the fruit is grown, because that can also influence?
Yeah, absolutely. No, we started only from wonderful pomegranate cultivated in Puglia, but just connected to a sustainability aspect, and so 0 kilometer product. But of course, maybe trying to characterize different varieties of wonderful pomegranate from different areas. Maybe the microbiota will be different, absolutely, because it's connected to the environment.
The environment,
Yeah.
yes. Any question before we break for tea or coffee, whichever one you like drinking? Which or juice. So on that note, I think we should give a round of applause. So I will be back for the next section at 12 o'clock of this. Thank you.
Vabbè. Ah, cazzo. Non so perché le persone sono cappite.
Beh, se no faccio da
Ciao Stefano, buongiorno. Come stai? Hai visto che ritorna la tematica delle harvest sul post harvest?
Ebbene, ok, after him. Allora, pomegranate presentation.
Esatto, perfetto.
Devo rinominarlo però. Lei a che ora?
12:30. Peccato che non abbiamo continuato a lavorare con il nuovo grande. Peccato che non avete fatto un gruppo con queste rapporti, perché gliel'avevo detto un sacco di volte, tribulato, con anima, ci ho fatto di tutto, soprattutto con Alberto, sono proprio un insensibile. Peccato perché oggi sarebbe stato importante.
Ok.
No, Amedeo.
Ok.
Io ieri ci siamo visti un attimo, però
Vuole controllare un attimo?
Sì, non vorrei che magari la compra. Perfetto. E ora Matteo è andato stamattina e se tutto non potrebbe venire. Che vuole vedersi. Può testimoniare anche lui che sono arrivato presto.
Confermo. Solo una nota sulla puntualità nel consegnare la presentazione. Sto scherzando.
I have two presentations for this afternoon.
For this afternoon.
By me, through the combination.
You are?
Zhang Ao Dance.
Ah, ok. This is the first one. Okay, if you want to check.
Okay, next correct. Okay,
Next one.
Okay.
Right?
Yeah. How to control this?
Okay, so now it's not connected because
Right.
This arrow will go to the next slide. Okay.
Okay.
This is the point. This button is the point.
Okay. Okay. All right. So.
Okay.
Okay. Great. Thank you very much.
You're welcome.
I have a presentation this afternoon.
Okay. You are?
I do faculty at 3:30.
3:30. Okay.
Please. My presentation.
No? Okay.
No, I'm just, it's very good. And now we're going to say, so that's what my big thing is now to grow pomegranate sustainable. So I can cover cultural practices. But obviously, yeah, you have studied, so you've got your knowledge there. But then, yeah, with other things, there's another thing. Then I started but obviously, I want to help the promoter.
Can I go out?
Yeah, because, but I am going to do horticulture now. They're encouraging me. They're not here. But they're going. I understand that it's two sort of variations.
As I said before, if you want, you can use physical
Okay.
Yeah, because I've got a young orchard, all wonderful. So I'm going to put the trees.
Okay. Thank you.
You're done.
But these are the main things.
Okay. Thank you.
See you later.
I'll pass it on to them. Yeah, it's important. Because the tracking thing is very. So I probably just 1.5 tons of fruit. But still, 1.5 tons of fruits, 1.5 tons of fruit. So it's, yeah. It is a waste. And I had enough sunburn waste. The other thing now, the question is, cracking sunburn, is there a link? And is it the same issue? Yeah. Well, this would be a future question. So it's very yeah, thank you. Yeah.
I'm sure that I won't be able to stay longer.
Yes, so he
So you are?
I'm the last one before lunch.
Before lunch, okay.
So it's in 12:45 to, that's it, yeah. Just making sure, you know, I have it on USB stick in case it's not there.
Okay.
It's should. That's it?
That's it.
Perfect, thank you so much.
Thank you.
I don't know, I'm
Always be careful.
I'm always double careful, maybe it's not necessary, but I rather be
No problem.
careful than be here and like, whoa.
It's right to do like this. Wait. So now we are. Let me check it.
Oh, tutti e quattro.
Altri 4 prima di pranzo. Quindi
ci sono tutti e quattro.
Adesso vedremo. Spanish, I don't really know. Hello. Luis
is here.
So we have Paolo, Enter, Aquino. Okay, ci sono tutti.
I think we can start. Polani, I think we can start.
Yeah, we can start. So,
Hello. Hello. Can we start here? Can we sit down, please? I hope you just can you test some more people inside? So we are going to start now. We've got 1, 2, 3, 4. 4 presenters to go before lunch, so they are in between lunch and us and lunch. So we will make sure we keep to time. So the first presenter is Luis. Can we welcome him? Can we give him
Thank you very much with Giuseppe and the organizing committee for giving us the opportunity to give this oral presentation, which is about antifungal radical coatings to reduce decay and preserve quality of boyardage pomegranates. This is a line of work that we started many years ago, more than 15, with my colleague here, Maria Bernardita Perez Gabo. She's a post-harvest physiologist and a food technologist, and I'm a post-harvest pathologist. So we try to join our topics of work to develop edible coatings, but with antifungal functions. Of course, being in Valencia, our main crop of work was citrus fruit, and we started developing edible coatings, composite edible coatings for citrus, but also in the last years we've done some work with pomegranates also. So for the introduction, the same slides that my colleague before, so about the importance of this autochthonous variety in Spain, so we can go fast on these ones, about the good sensory and organoleptic qualities of this variety, very sweet and very soft segments of the, very easy to eat. So it has a nice market in Europe, so it has been an important economical cultivar for Spain, which is, as Bernard mentioned before, Spain is still the first European Union producer and exporter of pomegranates. So for the storage, of course, we got the main problems are weight loss, chilling injury, and post-harvest diseases also. We got now still the fungicide fludeoxonil, which is still can be used, but it will not be, it will be removed very soon from the list of allowed fungicides in Europe. So we will face again a period without synthetic fungicide to control post-harvest diseases of pomegranate. So for that, our approach was to develop these antifungal edible coatings, which of course the main function is a physiological response to regulate gas and water exchange during storage. But by introducing an edible coating can be a good carrier of other type of ingredients, and among them antimicrobials and specifically antifungals can be incorporated into the formulation matrix in order to give this new functionality to the coatings. So we got a treatment, a post-harvest treatment with a double function, the physiological one and the pathological one. With the idea, in the case of citrus, for example, of replace the conventional waxes which applied formulated with imazalil or other post-harvest fungicides. So as Professor Fawali has explained very well, these edible coatings, they are composite. When we say composite, they use to have two main ingredients, an hydrocolloid to regulate gas exchange, and a lipid to regulate more the water exchange. And also we have to formulate them with minor ingredients, with emulsifiers, plasticizers, in order to get cost-effective and effective coatings. So as antifungal ingredients, the most common to add into being edible, they have to be natural compounds of food-grade condition. So most of them we have worked with food additives like grass is an American classification, generally recognized as safe. That would be kind of equivalent to the European legislation on food additives. Also, and as in this case, you can add also plant extracts, essential oils, and in some cases there are some groups even working with the addition of biocontrol agents as ingredients of these coatings. So this is another possibility. So the objective in this particular work was to assess the effectiveness of antifungal edible coatings. We did two different sequential experiments. In the first one, to select among a big range of coatings those with better weight loss control and also natural fungal decay. So for that, we only store the fruit at ambient temperature, 20 Celsius. And the selected ones, we did larger cold storage trial, storing the fruits at 5 Celsius for up to 12 weeks following 7 days of shelf life at 20 Celsius. So these are the list of the coatings that we evaluated. They were based on citrus pectin, that was with PEC, and carraginian, which is another hydrocolloid used for edible coatings. In all cases, we use beeswax as the lipidic ingredient, and then we developed several coatings combining the two hydrocolloids. Here are the characteristics of these coatings. So for the preparation of the emulsions, as we saw in the table before, were 9 different emulsions, and as antifungal ingredients, we used 2 different essential oils. Going back, which are essential oil from cinnamon and essential oil from lemongrass. Also, it has a glycerol as a plasticizer and oleic and palmitic acids as emulsifiers. So the homogenization first without essential oils, and once we got the emulsion, then we add the essential oils. Also homogenization for 2 minutes at 16,000 RPMs. So in the first experiment, as I said, it was just to select the best of the coatings of this list. So we dipped the Velarde pomegranates for 30 seconds. The control were just dipped in water for the same time. Air dry at room temperature, and then we just were looking for the control of weight loss with the storage for up to 8 weeks at this temperature. In the second experiment, we select some of the from before, and it was this formulated with pectin. We determined the initial quality at harvest, and then after we applied the coatings in the same way, by dipping for 30 seconds, and then the fruit was stored at 5 Celsius for up to 12 weeks. And in this fruit, as you can see in this scheme, after these 2 different storage periods, 6 and 12 weeks, we determined the most important parameters of physical-chemical quality, such as weight loss, juice quality, soluble fruit content, and titratable acidity. We also look for volatile ion content, ethanol and acetaldehyde. And also we look at some sensory attributes, which are very important any time that you work with essential oils, because, as you know, they have very strong odors and flavors, so you don't want this to be incorporated into your final product. And of course, we looked at the decay. In this case, we worked with natural decay. We didn't do any artificial inoculations. So we look at incidence of just how many of the fruits show decay and the severity. And in this case, we used a severity index based on from 0 to 4 scale based on the surface of the fruit affected by the decay. So the results from the first experiment, as you can see here, the pectin-based coatings were the most effective to reduce weight loss. So, well, it was a significant reduction with the pectin, but the carrageenan coatings were ineffective to reduce weight loss. And the addition of the essential oil as antifungal ingredient didn't affect the potential for weight loss reduction. So for decay, the natural decay, we differentiate between 2 different types of decay, those from latent infections and those from wound infections. And in general, we didn't get any effect of the coatings on decay, with the exception of the incidence of wound decay after 8 weeks of storage at 20 Celsius. It was also no influence of oil addition. And from ground decay, the most important was gray mold caused by Botrytis cinerea. And for wood decay, we found Aspergillus, mostly Aspergillus niger or Aspergillus in the section nigri, and also some species of Penicillium. So in the second experiment, after storage at cold temperature. So we got also a significant reduction of weight loss, especially after 12 weeks of storage. Regarding decay, we saw also in this case in wound decay, and wound decay was the one that we showed. We almost didn't found any ground decay in this experiment. So decay after cold storage was not reduced by the antifungal coatings. So also we found penicillium bronchitis as the most important causal species. Then we look also for the other quality attributes that they mentioned, and in any case, coating did not adversely affect these quality attributes. So just to conclude, the pectin-based coatings were superior to the carrageenan-based coatings for reduction of weight loss during storage at 20 Celsius and during storage at cold temperature. The selected pectin coatings were effective to reduce weight loss, didn't affect negatively physicochemical and sensory quality. So this is important, that the use of essential oils as ingredients of a coating can be an effective way to reduce the adverse effects of you apply the essential oil alone. So usually the fruit is coating like that, doesn't have a smell or doesn't have a taste related to the essential oil. So just to conclude, these pectin peel wax coatings containing these essential oils could be promising treatment, but we didn't get a good effect on decay reduction. So we should keep working to improve these formulations. Just to acknowledge, this work was done into an European project in the PRIMA program. And we got the collaboration from Victoria Alvarez. She was a postdoc with us in Valencia from Argentina. And these are the funding agencies. Also, some money came from the Valencia government. So thank you very much.
Thank you very much, Chris. Do we have one question? One question. Go ahead.
Louise, is there potential for using the coatings with sort of low-risk fungicides? So the carriers for the coatings as carriers for low-risk fungicides.
Well, it could be an idea or a possibility, but the idea of using edible coatings is mostly to not use fungicides at all, to use other type of antifungal ingredients. So you are able to go for maybe zero residue markets or even organic markets, depending on the ingredients. But the idea of using edible coatings would be more to replace completely the use of synthetic chemicals.
But if you don't get the decay control,
yeah, well, in this case, it was not. yeah, of course. So, but as I mentioned, the idea would be more to replace than to complement. But of course, as a matrix, they have, as Professor Favoli has explained very well, they have the potentiality to be carriers of many different additional function, giving additional functions to the coating.
I think we'll end it on that very interesting note. So thank you very much.
Thank you.
So our next presenter on South Africa, will be presenting on decay control for post-service programs.
Okay. Good afternoon, everybody. My presentation today is on Cytospora punicate, which is we found as a cause of post-harvest decay in long-term stored fruit, pomegranate fruit in South Africa. So just to introduce you to the work that we have done and a little bit more about South Africa, I think we all know enough about the superfruit characteristics of pomegranates by now. In South Africa, it's a fairly new crop. We started growing pomegranates commercially around about in the early 2000s. And that was more or less coming from a drive from industry to start to diversify. We are a big fruit producing country and mainly exporting most of our fruits, and citrus, grapes and deciduous fruit are the biggest fruits grown, produced and exported from South Africa. So more than 80% of the total hectarage of about 1,000 hectares currently planted in South Africa, that is pomegranates, are based in the Western Cape, which has a typical Mediterranean-like climate. Our flagship variety at this stage is Wonderful, and it makes up more than 90% of all plantings of pomegranates in South Africa. So South Africa, as I said, is a predominantly exported country, making the industry well aware that post-harvest fruit diseases are critical because they link agricultural production, the economy, food safety and sustainability for the industry. So that was why it was important to do a study to understand which diseases are associated with the South African fruit, to be able to decide on suitable practices and management options, which would be essential to reduce the losses and also ensuring high quality produce and sustainability, profitable trade, and also being compliant in terms of SPS agreements and which is reflected in our market access. So during this study that I've done at the Stellenbosch University, the post-harvest division, at the plant pathology department, and I'm also accompanied by the head of our department, Dr. Cheryl Lennox, and also Dr. Julia Mates-Hopkins, who's also attending the symposium. One of the interesting findings was the manifestation of a typical stem canker pathogen, Cytospora punicae, as a post-harvest disease. This was the first recording of Cytospora causing post-harvest fruit decay, and was also afterwards confirmed by researchers from Italy. All other reports, I must just mention it, of Cytospora punicae in pomegranate was related to stems and branch dieback, formation of cankers, and very limited, if any information really is available at the moment about Cytospora punicae infection in pomegranates. So obviously further studies would be necessary to confirm aspects like etiology and epidemiology, etc. So these diseases were recorded from Greece, California, from Iran and Cyprus, and as well as Tunisia. So those were the other recordings on Cytospora. Now just to introduce you to the symptoms. So there on the left you can see the external, it's not very clear here. External symptoms starting to develop. So you would first see it as an external yellow lesion on the epidermis of the fruit. Also with formation of pycnidia after long-term storage. And then internally you will see a dry cork-like yellowish appearance of the mesocarp. And then eventually also the necrotic seeds in the inside of the fruit. Okay, so the aim of this study, which is then, as I said, a part of the bigger study in terms of the total assessment of post-harvest diseases on pomegranates. And the main aim was to quantify and confirm the identification of Cytospora punicae cultures isolated from the deteriorating long-term stored pomegranate fruit. The second aim was to validate the potential mechanisms of fruit infection in pomegranate. So to confirm then the fruit infection potential through wounds, both on the fruit itself as well as through the stem end wounds. And then thirdly, it's to verify the endophytic or dormant nature of Cytospora punicae in the fruit peduncle and its ability to migrate into the fruit via the vascular tissue, which is the xylem, where it remains dormant until after harvest and expresses typical symptoms during storage. A preliminary assessment was also done to determine the incidence of latent Cytospora in 6 pomegranate orchards in the Western Cape through screening of harvested asymptomatic fruit after storage for the developing of the typical symptoms. Okay, don't stress, I'm not going to talk about everything. I just wanted to take you quickly through this. So about the methodology, first, the fungal isolations were done from 25 production sites, fruit collected from 25 production sites. And the procedure was by evaluating commercially collected and treated fruit, after which they were plated out on PDA and through the normal process of identification. We looked at morphological identification and also molecular identification. Then for the detached fruit inoculation method, healthy pomegranate fruits, sorry, excuse, I need my glasses, I'm sorry. Okay, for the healthy pomegranate fruits were collected and sterilized, and then we just created normal wounds and inoculated the fruit with plugs, with agar, with Cytospora punicae cultures. The fruit stem inoculations was done in vivo in the orchards. So we pretty much followed the same procedure where we inoculated the peduncles or the fruit stems with agar discs with cultures of Cytospora punicae, and obviously the control plants were inoculated with sterile agar plugs. The fruit stem end scar inoculations were done on detached fruit, where we also collected presumably clean fruits. We harvested them with the peduncle, and in the laboratory we just snapped it off, and then also inoculated that stem end scar with agar discs with the culture, and the controls also sterile agar discs. And for the incidence of naturally infected fruit in the orchards, the mature pomegranate fruits of, as I said, 6 locations within the Western Cape were selected. They were just sanitized, harvested and sanitized, whereafter they were incubated and stored for 14 weeks at 7 degrees Celsius, and then we screened them for the developing of Cytospora symptoms. And then the percentage of the fruit with the decay symptoms was confirmed. Okay, this is just a snapshot of the work being done in terms of the complete assessment of all the different post-harvest diseases that we collected from our study. What is interesting is to see basically, and I think it's the story of most pomegranate growers' lives, is that the penicillium group made up 77% of all isolates. So this is a big culprit. Fortunately, I think we can manage it to a certain extent by means of production practices and harvest practices and controlling of wounding. And then just to show you the incidence of Cytospora punicae that we have found is there indicated in red. So it was more or less a 5% incidence of Cytospora punicae. In terms of the results, the identification, all isolates exhibited morphological characteristics of Cytospora punicae, as described by Lawrence. So there in the pictures, you can see the beginning of the growth of the cultures. This is what they look like. Oh, sorry. This here on the left is just starting to grow, and then later, after about 28 days, you will start seeing the browning of the mycelia. And later on, you will also then recognize the globose media forming on the media. Eventually also the identification, to conclude then on that, of C. punica isolates from South Africa aligns with the known morphological characteristics and molecular identification of trunk isolates that we find in the other countries that I've mentioned. For the mechanism of infection, those are just pictures to show you what we did in terms of surface inoculation. So what we found is that all fruit surface inoculations were done, and we had 100% isolation afterwards of C. punicae. So you can see it visible again, as I've mentioned, the yellow discoloration of the skin and the internal symptoms, dry cork-like symptoms. In terms of the detached fruit, we had the same results. The inoculation on the stem end scar of the mature fruit led to the development of stem end rot symptoms with distinctive discoloration of the vascular bundle in the mesocarp. So I'm a little bit pressed for time. I'm just going to move forward to explain a little bit more why we came to this conclusion, and that was also a result from our endophytic theory that the Cytospora punicae is an endophyte which can move through the vascular tissue of the fruit. So where we inoculate the fruit stems after harvesting and incubation, we found that there was in 95% of the cases developing of symptoms of the incubated fruit. To further illustrate the theory is that I found a very old publication from Sinha and Joshi that explained how the vascular system of the pomegranate work and within the fruit, showing the anatomy of the stellar primary vascular bundles on the outer part of the mesocarp and the vascular bundles that run uninterrupted through the interface between the stem and the fruit. So in a longitudinal section through the pomegranate fruit, in the figure B that you can see there, you can see it illustrating the primary vascular strands that that show a typical discoloration where you get the infection from Cytospora. And then at the bottom, figure C, you can see a cross-section through the infected fruit with the discolored primary vascular bundles reaching inwards towards the calyx end of the fruit, and with tissue necrosis stretching then eventually into the mesocarp of the infected, from the infected bundles. Okay, and then just shortly the incidents from our preliminary trials. So we found that decay symptoms developed in stored fruit from orchards that we harvested. Quite surprisingly high percentage of infection. We got between 5 and 27%. So the significant differences that we see here, we still not know. So we have to do further trials to see. It could be prescribed some things like different origins of the plant material, microclimate differences, or even different orchard practices that was followed. To conclude, the South African fruit isolates of C. punicae align with the known morphological characteristics and molecular identification of trunk isolates from other countries. So the potential infection pathways could be through biological mechanically induced wound and also transferred to fruit through the stem end wounds. So Cytospora can migrate from the fruit stem or the peduncle into the fruit via the vascular tissue, obviously then from the plant. So the endophytic latent presence of the fungus in the plant can cause the fruit decay during prolonged storage. So this was during the preliminary orchard assessment. We have to follow up on that and follow up studies necessary to determine the actual importance and potential management options for this fungus in the future, especially where prolonged storage become more and more relevant in the industry all over the world. Thank you.
Thank you. We are out of time. Is there any very brief question? Very important one. Nice. Thank you very much.
Thank you.
Thank you. We have two more presentations to go, and our next presenter is already on the stage. Salvatore.
Salvatore, yes.
Salvatore, yes. We're looking forward to your presentation.
Thank you. Okay, so thank you very much to conveners to let me be here for to present these results related to combined treatment with modified atmosphere packaging and water deep set treatments and fungicides to reduce overall spoilage and disorders of pomegranate. And com'è che si... Ah, ecco, perfetto. So pomegranate is an ancient crops in the Mediterranean area. It has always been grown in the Mediterranean basin and also in Asia for its important cultural and nutritional and medicinal value. In Italy, it is always been a very popular crop, a very popular fruit. Basically, it was cultivated in the southern region of Italy until the end of the last century, but it was only in the last 2 decades that the pomegranate industry has grown very, very speedily, a lot. And actually, right now, the production is increased a lot, especially in Sicily and here in Apulia, but even in other regions of the north of Italy, the cultivation of pomegranate is increasing too, like in Veneto. And the global production is increased in Italy too, and obviously grapefruit, pomegranate are appreciated for the health promoting compounds, especially polyphenols and anthocyanins, flavonoids, and other vitamins. But despite all these important health benefits, grapefruit have a lot of issues, of post-harvest issues. And because grapefruits are not climacteric fruits, so when fruit are harvested, they not continue to ripening. So the harvest timing is a critical step, a very important time, as shown in previous work by Dottoressa Gaza and from Spain. The ripening stage is very important, not only for the internal quality of the fruit, but also for the expression, the development of post-harvest disorders. And post-harvest disorders that basically are 3. We have 3 kinds of post-harvest disorders. We have Ask Browning, we have chilling injury, and we have weight loss that represents one of the major causes of loss of post-harvest quality of grapefruit. And pre-harvest cultural practices and pre-harvest management of the field is very important, because if not properly treatments to control decay or pests are not conducted, a lot of problem can arise during the post-harvest handling of the fruit. As we can see, we removed the tissue of the crown in that fruit, and I don't know if you can see very well, but there are different conidia of penicillium inside the crown, but there are infestation also from many bugs, and in the field we have also seen infestation by Ceratizis capitata. So pest control and decay control in the field is very important to preserve the fruits post-harvest. As you can see here, these fruits are not stored. This fruit here in the bottom. Okay, why? See. So this fruit here are fruit affected, are decayed fruit, but these are not stored fruit. These fruit were just harvested 1 week, and we left them in the lab, because when we selected the fruit for storage, we saw that there was something not good in this fruit, and after a couple of days from harvest, this fruit developed this decay. So it's very important. It's very important the integrity of the peel, because if there are big break, it's not a problem, because one can select the fruit at the beginning of storage. But if we have micro cracks on the peel, that can represent an avenue, an entrance of pathogens, and that will develop during storage. So the objective of this experiment was to evaluate combine the treatment with the fungicides, and we used in particular imazalil, which is a worldwide used fungicide to control several diseases in citrus fruit, and it's an old fungicide. But we also used fludeoxonil, which is a fungicide right now approved for the control of several post-harvest diseases of citrus, but also pomegranate. And the imazalil is fludeoxonil is very effective, effective against a large range of fungi, but is very active against gray mold, which is a key pathogen, a key post-harvest pathogen of pomegranate. So we use the fludeoxonil, imazalil, and a combination of the two fungicides, and we applied these fungicides at 500 milligrams per liter. And when the two fungicides were combined, we used half doses, so 250 grams of each one. And we gave these fungicides by dips, and we dipped the fruit in emulsion or in water at 2 or at 48 to 50 degrees centigrade. 48 to 50 degrees centigrade, because the tank we used, I'll show you the tanks, how we did the heat temperature. But anyway, when we immersed the fruit, the temperature was 50 degrees centigrade, and after a minute, the temperature decreased to 48 degrees Celsius. So we say 50, but actually the temperature was between 48 and 50 degrees centigrade. And then half of the fruits, film wrapped half of the fruits with a neat Jenga film. So we did a unipack, and we eat the fruit individually, and we stored the fruit at 2 or 8 degrees centigrade. Why 2 and 8? 2 because 2 is a typical temperature that causes chilling injury, but is a typical temperature that is worldwide adopted to prevent quarantine pest. So for quarantine treatments, generally 3 weeks at 2 degrees centigrade is the standard treatment for quarantine treatment, and 8 degrees centigrade, because 8 degrees Celsius is the temperature or at which scald develop. Okay, and we stored the fruit for 4 or 8 weeks plus 7 days at 20 degrees centigrade and 50% relative humidity, to simulate marketing conditions. And here you can see how we did the treatment. These were the fruit at the beginning. We selected the fruits. We used this tank with a thermostat, and the water was continually mixed to let the temperature be homogeneous in the tank. And you see here is a 49, something like that, but the temperature, as I told you before, ranged between 48 and 50 degrees centigrade. These are the treatments, and these are the fruit after the treatments. This is the unwrapped fruits, how they appeared, and these are the fruits which were wrapped. So I go very quickly. We evaluated these disorders: ask Browning, chilling injury, ask Browning on a scale of 0-4, with 0 no symptoms, 4 with browning of the peel of the rind greater than 50%, chilling injury as a pitting of the skin, and as browning of the white tissue of the mesocarp and of the septa surrounding the areas, then decay incidence, weight loss, and the Jewish parameters. I go quickly because I see that the time is going to finish. So what we saw was that first of all, there was a treatment that increased, that was phytotoxic, and increased as browning, and this treatment was all combination treatments with mazarin. As you can see here, all the fruit treated with the mazarin get very rapidly get brown color of the skin, and this refers to fruit after these are fruit treated with fludeoxamil and dipped in water at 48 degrees centigrade after 8 weeks of storage. And this is the fruit of rapid fruit. These are fruit with fludeoxamil, and this is the control fruit. This is the control fruit, and this is the fludeoxamil treated fruit after 4 weeks of storage. So as you can see, in rapid fluids, there is no symptoms of a scald, and it was slight in fludeoxamil treated fluid. There was some browning in control fluid. As browning obviously was higher at 8 degrees centigrade than at 4 degrees centigrade, at 2 degrees centigrade. After 4 weeks, basically all fluid at 2 degrees centigrade did not develop as scald, especially those which were dipped in water, or heat emulsions, and especially those which combined the fiber wrapping with the treatment. Chilling injury developed more in fluid stored at 2 degrees, obviously, but as you can see, in fluid which were wrapped with the plastic film and treated with fungicide, there was no symptoms of chilling injury, neither after 4 nor after 5 or 8 weeks of storage, and the same occurred obviously at 8 degrees centigrade, while fruit which were not treated with hot water or with film wrapping developed signs of chilling injury, and the chilling injury developed both as sunken areas, pitty area of the peel, getting this brown color, but also as browning of the mesocarp, the white tissues of the mesocarp and the membrane, and as browning and whitening of the areas. And regarding weight loss, it was impressive the effect of the film wrapping, because even after 8 weeks, weight loss never exceeded 2.5% of the initial weight, while fruit which were stored for 8 weeks at 20 degrees centigrade lost almost 25 and even more than 25%. But here you can see the fruit, wrapped fruit and unwrapped fruit after 8 weeks storage at 8 degrees centigrade and 20 degrees plus 1 week at 20 degrees centigrade. And you can see that the peel of the fruit turned to a yellow, dark yellow color, but this fruit was red. It was liquor. And but there was what the important feature was that when we cut the fruit, the area was still good, despite all this fruit lost in water. Because in particular, this fruit lost about 30% of the initial weight. So you see that there is no thickness of the rind. We cannot see the peel, while these were the fruit which were rapid and still has an appreciable thickness of the peel. And regarding decay, well, overall feelings obviously was a combination of the results from chilling injury, as browning, as scald, and weight loss. And as we can see, all rapid fruit were still very good even after 8 weeks, while fruit treated with imazarin or fluid, which were not rapid after 8 weeks, were not anymore marketable, and we have as index number as limit of marketability 5 as index number as limit of marketability. But what is important is the decay incidence. We had a lot of decay, probably because the fruit were not properly in the field, were not treated properly in the field, and gray mold was the main pathogen. But there were other diseases, as you can see, and even at the end of the storage, we still saw the presence of mealybug in these breaks here. So decay was very important. Film wrapping was very important to maintain and to almost completely suppress any kind of disorders, physiological disorders. But when the fruit were not treated with the fungicide, okay, I'm finishing. So when the fruit were not treated with the fungicide, we had more than 50, you can see 60% of decay. So it is not possible to use a film wrapping without combining with a proper fungicide treatment. So in conclusion, what we can see? We can see that the film packaging is a good option. It's a good marketing treatment, but it absolutely needs to be combined with a fungicide or even with the treatment that by itself reduces but not very effectively decay incidents. And we can store food at 2 degrees centigrade because with film wrapping, chilling injury does not appear until 4 weeks of storage. And if the objective of 2 degrees centigrade is too Celsius, is too for the control of the insect, of the pest, for quarantine treatments, 3 weeks are more than sufficient. So it's a good picture. But if it is not required, a quarantine treatment, 80 degrees centigrade is the best choice. Thank you very much.
Thank you very much. We'll move on to the next presenter. I'm sure if you have any questions, we can add to have one that I will engage with you during lunchtime.
Okay, thank you.
Thank you. Ursula? Yes. So you are between us and lunch. So hopefully we'll finish on time. So we're looking forward to your presentation. Yes, not the time will be set.
The time is set.
No pressure.
That's for me.
All right.
right. Good morning. It's this presentation and then lunch, so no pressure, right? Okay, so I'm very happy to report two different trials on the performance of pomegranate cultivars at different elevations in the southwestern United States. I'm at the University of Arizona, and so this has been done with my colleague Glenn Wright. So in the United States, there are about 14,000 hectares cultivated in 2022, according to statistical data. 99% of those are in California, and Wonderful dominates the production, almost like it does worldwide. So we were interested in looking at how can we expand production into Arizona, which is immediately to the east of California and also has a semi-arid climate. And so we were looking at to determine vegetative growth and reproductive development of 10 pomegranate cultivars, including the industry standard Wonderful. And some growers also use the cultivar Angel Red when grown at different elevations in Southern California. So here are the different elevations, and you can see the first trial was done between 2013 and 2015, and we had 3 locations, and they differed quite a bit in elevation, between 50 to 1100 meters. And here, these are the USDA climate zones. These are what you could expect in terms of minimum temperature in these climate zones. Most of the sand was sandy loam or sand, and then we used flood irrigation or subsurface irrigation in that first experiment. And you can see the pH is alkaline, and we have no problems with salinity at any of these sites. And then in 2022, we planted another trial, and that one was done at 700 to 1300 feet because we found, meters, because we found out that the site at the lowest elevation is not ideal for various reasons. So you can see the sites over here. They're all located in southern Arizona in the Sonoran Desert. And Tucson is part of both of the trials. The red ones are the first trial from 2013 to 15. The blue stars and Tucson are for the second trial. And here you can just see what our subsurface drip irrigation looked like. As you know, we're in a very, in a bind for water, and so trying to look for ways to minimize irrigation and maximize the use of water is very important. So these are the 15 cultivars, Angel Red and Wonderful being the standards that we were using. We're using 2 cultivars from Southern California. They are heirloom cultivars with low seed hardness and low color, Josefina and Sosa Carrillo. And all the other cultivars were obtained from the USDA germplasm repository in California near UC Davis. So these are the results on the phenology for the first trial, and you can see the high, medium, and low elevation over here for the 3 years. For the last year, we didn't have the high elevation because we had also in 2013 some significant freezing damage. What you can see in the right column is the temperatures that we had between January 1st and March 31st, so prior to bud break and anthesis. It got really cold, minus 12 degrees, and the year prior to that, when we were planting the trees, the temperature was like minus 18 degrees, and a lot of the plants froze down to the ground and had to regrow again. So freezing is definitely a problem, especially for the younger trees. Usually once they get older, they obtain a higher freezing tolerance. The general finding is that, of course, in the lower elevation location, bud break and anthesis are first, and at the higher elevation, bud break and anthesis are last. And in general, we had between 31 and 49 days between the onset of bud break and anthesis. And then this is the most recent result, and it was very similar. In this case here, we had a late freeze, and that pretty much killed all the flowers. So there were flowers then later, but we did not record them anymore because it was so far out of the regular growing season. So freezing can be a problem both for the canopy, for the branches and everything, and for the freezing of the flowers then. This is the growth index. That's just an average of the two canopy width and the canopy height. Not surprising enough, we're having the smallest canopies each year at the high location, and larger ones than at the lower location, and the same pretty much over here. In terms of the cultivars, the ones that are labeled in yellow, those were the ones that grew larger, and the ones that are labeled in gray, those are the ones that grew smaller. So we had interaction effects on the growth of the canopy based on the year, based on the location, and based on the cultivar. For harvest, we looked at the fruit weight per plant and at the average fruit weight, and we found a lot of interactions by location and year, by location and cultivar, and essentially we found that the fruit weight per plant was smallest at the highest elevation location, because also we had the smallest canopies there. And in general, from the first year to the next year, plants started to increase in the fruit we could harvest, except in Yuma, it decreased. And we think that low location was just too hot for the cultivars that we were using in this study. Maybe some cultivars from India that are better suited for the hot temperatures would be better. But the ones that we used did not perform very well. And we always found the greatest harvest in Tucson for the 3 years and in 2024. And overall, when we looked at the cultivars, we found quite a big difference. On the right side, the cultivars are listed that were doing very well. And you can see that in terms of the site, the Tucson site produced the largest fruit for each of the trees. And then when we looked at the different years and at the different cultivars, again, you can see on the right sides the ones that performed the best. And not surprising, from 2013, then for the following years, the fruit increased in terms of harvest. Then when we were looking at the total soluble solids or bricks and arrow color, we found that the bricks, they fluctuated between 13.5 to 17 in the first experiment in the 3 locations, and then last year from 14 to 17.4 between the 3 locations. And the high elevation Bowie and then the medium one, Tucson, they had higher bricks than Yuma. So in that warmer climate, the plants did not produce the sweetness that we would expect from a pomegranate. And then we also had differences in bricks between different cultivars, as you can see. And in the most recent year, the highest bricks were again at the highest elevation in Wilcox, and then lower than at the decreasing elevation. And we had the lowest bricks and arrow color development in Yuma, but delaying the harvest may have increased the total soluble solids. And the aryl anthocyanin color was darker with the cooler temperatures, so we did not get the darker coloring. And here are just a few examples. On the left side, those 3 cultivars have lighter colored aryls, but we can see at the higher elevation in Bowie, they do develop greater anthocyanin content, whereas in Yuma, then the majority of the juice that we expressed was like light yellow, barely ever even turning into a pink. On the right side, you see the darker colored cultivars, where the arils usually turn dark red. And you can also see that we have in Ui, the high elevation, we have much darker aril color development than in Yuma at the lower elevation. So overall, the quality is quite different between the three elevations. So in summary, the greatest canopy volume was in Tucson at the 700-meter elevation site in both trials, but break and anthesis was started first at the lower or the warmer elevation and then was later at the higher, cooler elevations. And it differed between the cultivars and the sites. The fruit weight per tree was affected by location and year and by cultivars. Average fruit weight was affected by cultivars and year. And there are we found 6 cultivars that had similar fruit weight as the industry standard. So we see a potential of some of these to definitely be grown for commercial purposes. And then bricks and aril color were affected by cultivar location and year. Higher elevation locations produced fruit with the higher bricks. And we did have cultivars that produced fruit with acceptable degree bricks at harvest. Darker aril color at higher elevation, lighter at lower elevation. Yuma site is definitely not ideal for the cultivars that we chose. Maybe there's even a lack of chilling. We don't know if that is necessary. But overall, the trials demonstrate that cultivar selection and climate at the different sites where pomegranates will grown affect several of the characteristics like yield, fruit size, degrees bricks, and aril color, and all of those are important for economics and consumer preferences. Thank you so much for hanging in there.
Thank you very much. So I suppose we can go to lunch, right? Yes, so a round of applause to all the presenters in this session. So that concludes this section, and I look forward to welcoming you back for the next session. Thank you very much.
Yeah. Yeah, exactly. I think the next year is the next year, so we need to
Yes,
it took like
wow, 25 minutes, so it's too much.
We can leave our bag.
Yeah. Sì, mi occupo un po' di tutto, quindi va bene anche questo.
Se tu che ti occupi,
Sì, no, ma tu che fai? La risposta sì. Quindi sì. Un attimo che sto caricando un altro.
Tanto la proposta è la stessa.
Salve. Could you give you the presentation? Yes. Flash? Huh? Okay, we have a problem with this one. Next one. Pomeriggio? Allora, adesso riprende dopo pranzo. Nel pomeriggio.
No, non so come ce li avete organizzati.
Sì, lei che è?
Sono bravo, sono contento.
Ma avete parlato della situazione? Cioè, quindi alle detto 17 16:45. Ok. Vuole controllare? Sì, sì. Così poi va in mirroring, quindi può anche vedere le note poi. Perfetto. Un attimo. Ok. Sì, sì.
Sono il speaker della sessione.
Allora, no, se vuoi posso lasciare.
No, preferisco fuori.
Perché mi hanno anche detto che devo indicare, diciamo, che quelli che sono sempre e poi ci sono insomma le affiliazioni. Sì, 15, ok.
So you can check because
I have already one of your presentation.
You have one? By
Yes.
now?
Maybe we can replace it. No problem.
It is the same. Maybe the email images are compressed. Yeah, 15.
Okay, we replace it.
Can you check if it is working?
Yes. If you want, you can.
Yeah.
If you don't want to spoil nothing, we can avoid to put in presentation mode, okay?
That's fine.
Okay. Okay, thank you.
So can I
Yeah.
Thank you so much.
Thank you.
Questo è per domani?
Per domani, ok.
Domani pomeriggio.
Prof, può chiamare Donato Gerini perché non legge la sua... Può anche portare... se non è già andato a pranzo.
Sì.
Quindi...
Dovrebbe essere le 15, non me lo ricordavo.
Allora.
3 un quarto?
No, non legge la chiavetta. Scusate.
Vai, tranquillo.
Se no riscrivo.
Se non fa cosa.
Sì, se vuoi. Io c'ho il numero pure.
Siamo tutti...
Sì, a posto.
Siamo tutti quanti. Noi ci siamo incontrati a svolta.
Sì, è vero. Sì, anche con lui. Ciao.
Qual è il problema con la presentazione?
Non legge la sua chiavetta, semplicemente il computer dice che ci sono problemi a leggere la chiavetta. Venerito, giusto?
Sì.
Ok. T3 15 e 15.
Come pomeriggio stanno portando l'ultima uva.
Eh sì, ci ha avvisato Claudio che ha detto domani, giusto?
Domani, sì.
Ok.
Sì, la data forse l'ho messa giusta.
No, è un giorno prima.
No.
Niente.
Mi siamo ritorni solo.
15 e 15. Questo lo eliminiamo. Ok? Già.
Sì. Grazie.
Niente. Devo controllare.
Ok.
Devo controllare sta.
Io la posso fare oggi, domani.
Quando vuoi tu, Andrea, tanto.
Sì.
Domani
Domani
fino alle 12 non ho un cazzo da fare. Nel senso che potete anche venire prima, possiamo venire prima se volete provare proprio qui.
No, io sinceramente riesco a venire qua. Tanto c'è quell'aula nel corridoio che è disponibile per la così.
Se hanno prenotato fino a tutti i 3 giorni questa stanza, anche metterti qua. Cioè,
No, vabbè, ma c'è pure quella, io mo la stavo, anche se proprio c'è quest'aula dove c'è così, infatti c'è
il posto.
sì, c'è un cartello dove sta scritto un foglio.
No.
È da ricaricare.
No.
Ma magari solo carica batteria.
No, quello su quello non so.
E come si fa?
Giovanni e
Dove sta il
Gigi.
microfono? Cioè, qual è?
No. Allora, questo è... Quindi sono 1, 2, 3, 4. E questo 5. Ah, c'è Gerin. Gerin, giustamente. Gerin.
Quando dobbiamo fare sta pila? No? Quando dobbiamo fare sta pila?
Ma è spento, spegnilo prima.
Vabbè, si spende automaticamente. Vabbè, secondo
Ma
me
è ricaricabile? O bisogna cambiare?
Serve il caricatore della pila.
No, ma quello non penso sia manco ricaricabile.
Sì, vabbè.
No?
Al di là di ciò, non serve una pila come questa.
Non lo so, perché lì la scritto sempre con
No, andiamo a un negozio di cinesi,
Ma non è che c'è un modo per ricaricarlo?
No, la pila è...
Non ho caricato tante volte. Comunque abbiamo funzionato.
Insomma.
Vabbè, io ci ho provato.
Sì, sì, è quando la luce è arancione che non... Io lo capisco qual è il problema.
Quindi eventualmente lì possiamo mettere la cosa, no?
Funziona, no? Però mentre lo utilizzi, spesso della luce diventa arancione.
Diventa arancione perché va in stand-by.
Sì, mentre tu parli, vai in
Mentre
arancione, perché
parli.
secondo me ha un problema di contatto.
Io non ho notato sta cosa.
Perché se no non lo mettiamo uno scotch-cola.
Sono buoni. Questi sono i servizi che ci dà l'Unib.
L'Unib.
Il microfono l'ho preso dalla Cina.
Perché questo è quello che mi piaceva. Scusami, guarda che schifo.
Ci hanno fatto le guerre puniche.
Vieni a vedere, Andrea.
No,
Funziona.
Io chiamo proprio Gerino.
Io ho scritto, aspetta, non ho una risposta.
Io mi sto ancora mo dice che l'ho rotto la penna.
Vabbè.
Senti, ma qui le note si vedono, si riescono a vedere?
Sì.
La linea guida non la sto facendo,
nemmeno la legge proprio. Cioè. Non ci credo.
C'è un problema.
E guarda qui, è tutto rovinato. Tutto...
Ma voi non vedete mangiare?
Sì, sono a contatto con il professor Gerini.
Che è successo?
Vabbè, andiamo a trovare la messa, tanto...
Senti, Simo, il microfono funziona comunque.
Gliel'ho fatto caricare sempre questo.
Da dove l'avete fatto caricare?
E gliel'ho detto loro, l'organizzazione.
No, perché non so caricare.
Avranno capito una pila, non so.
Ma comunque non funziona. Cioè, direi che non funziona.
Penso... Sì. Sì, sì, sì.
Sì, c'è un ragazzo.
Sì, ho visto che sì, vabbè, no. Mi aveva detto che avevano difficoltà con... Vabbè, aspettiamolo.
That's okay, I'm gonna go back into my box. Oh dear. Yes, no, yes.
I'll leave it. Thank you. You want the a feet?
Yeah.
Oh, yes.
So you know what to do. I mean, depending on how many people you can take over. It's not going to take over. I mean, when it comes with when you go to 10% say, and then, but the room is because the room because the speakers are also not here. So
Yeah, the speaker can also use it.
the let's see the
Salvatore da Cruz, the chairperson. This is the first one. But together we are almost at 3.
Yeah. So we are 30 minutes late.
Okay, not too bad. Then you
Maybe
have this.
I don't know about.
Is it this what I was thinking? Oh, this one.
But it will be just.
Oh, it's 15 minutes. It's not.
We will give a speech, but just.
A video. Okay. Yeah, it's also not.
So we have to take time very fast, please.
Yeah, but I also there's also a work. No.
No, but the people that
people that is at the end, so people might not hear it. Yeah, we have to take. Yeah, maybe we don't go for coffee.
No,
Yeah, we don't go for coffee.
we are waiting for the.
Yeah.
Shareholder session. Salvatore Latuna.
Okay, we can ask him to call me.
Okay, you.
Okay.
Because he's an
Okay.
Okay. So do you know how many speakers are around now? So we have to drive. Do we know because then we can decide whether to start?
At 7, they will set up there.
Okay, so we have one.
Yeah.
Yes.
Maybe we are.
Yeah, what I can know is the election.
Yeah. We try to finish the decision before the. I asked them to start what we do at 7. Well, it's supposed to, we are supposed to finish.
Ascolta, ma il timer lo devo attivare io?
No, faccio io.
Fai tu.
Sì.
Io devo salvare.
No, it says at 7.
Un attimo, che faccio una cosa e adesso le dico tutto. No. Mancava una presentazione.
Stiamo in ritardo. So che ci può
Ma siamo praticamente guarda già
Siamo a mezz'ora di
Sì. Adesso lo stavo salvando.
mi sembra una Stefano.
Ok? 11 Sì.
Onestamente non mi sembra giusto perché delle relazioni che si vede, cioè relazione da solo, cioè non mi sembra carino, non credo.
Lo so, non vorrei accumulare troppo.
Comunque, dico. Allora,
qui aspetta tu.
No, allora, diciamo che qui io ho segnato chi effettivamente parlerà.
Aspetta, c'è un cambio, perché il primo non è più questo, ma non può fare.
No, allora qui. Quindi calma. Allora, questo si può buttare in mano, questo. Ok, quindi prima
Non questo, non è questo.
È calma. Non ho una penna che sono. Oddio, forse devo
Sottolinea al secondo.
Io consiglio di fare su questo, perché questo si può vedere anche da tutti gli altri. Vabbè. Ecco qua.
Ho già impostato il programma per il
Grazie. Mi passo il primo. Cristina Pensata. Quello sottolineato, quello del qui se vuole, l'ha messo anche da c'è Sabani, non ho trovato Pomsym, però la presentazione ok? Ma io parlavo e poi e poi l'ultimo Però forse non mi interessa.
Sì, questo tu me l'avevo detto stesso.
Lei, della mano.
Ok. Su field management? Maybe session field management? Okay. Which one? This one.
No, parto dalla di Browning.
Field management, yes. Okay, so because we have this one, maybe this was
No.
not version. Okay. Okay, so I can delete this one. Okay, this is the new one. This is the old one.
Okay.
Okay, so I can delete this one.
Okay, delete.
Okay.
You can check if
Yes. So it's
yes, okay.
I will put in. Okay.
Okay.
Vabbè. Yes. Sorry. Okay. Thank you.
Tra 3 minuti iniziamo con mezz'ora di tempo, quindi dovremmo cominciare mezz'ora.
Tanto c'è il time, insomma, quello che le persone lo rispettano.
Niente domani.
No, dipende. No, in realtà se finiscono ad orario Pomsym, assolutamente sì. Se chiaramente si prendono 10 minuti in più rispetto ai 10 minuti.
Perché già abbiamo iniziato a aiutare.
Vabbè. Diciamo che, come ha detto anche lei prima, sarebbe un po' irrispettoso non concedere domande.
Cioè, ma infatti allora avevo già in 4 minuti.
Noi dobbiamo iniziare.
Vedrei a questo cavolo.
Però qui dicevano, visto che siamo mezz'ora in ritardo.
Quello che stiamo dicendo però. Ok.
No, quello funziona.
So we're going to start with session and Dr. Karamanoglu can start his presentation and speak about the field methods to predict the number of alleles in the weight. Please.
Hello everyone, good afternoon. I think we all here, and it's one of the worst time to make the speech after the very good lunch. I would like to try to keep it short. My presentation today is about the non-destructive methods for the prediction of number and weight of the pomegranate alleles. We have prepared this study with my colleague, Dr. Ersin Çağlar. As we all know, and mentioning since the beginning of the symposium, pomegranate is one of the oldest domesticated fruit species and historically cultivated for its nutritional and medical purposes. And since the 21st century, the scientific interest in the fruit has surged primarily because of the studies which are confirming the health benefits of this super fruit. Oops, sorry. And we all know that there is a growing concern over the global food security, which is impacted by the or driving the force on the precision agriculture, which integrates the sensors, advanced machinery, and the information systems. These are also all impacted by the increasing global population, the climatic uncertainties, and of course the sustainable use of natural resources. So at that time, the precision agriculture, which the efficiency, quality, and the sustainability are becoming an important issues for us. So at that time, the new approach focused on the efficiency, quality, and sustainability in the production. And the accurate, fast, and the most important point is the non-destructive quality assessment is essential for the process optimization for the quality control during the marketing or the supply chain. We know that the non-destructive methods can estimate the number of areas and the weights without cutting the fruit, supporting the post-harvest handling and the marketing classification. There are several studies on different kinds of fruits, and several technologies have been developed which scanning the fruits and making some predictions about the inner quality of the fruits, which enables better sorting, reducing the waste, and improving the site-specific decision-making, enhancing the food availability. In this case, our objective was to test some interactions between several characteristics. I will explain in the methodology part. So the main aim, we can say, to non-destructively model the number of and the weight of pomegranate areas. The study that been carried out in northern part of Cyprus, similar with my previous presentation, the fruits were all collected from the same field, same orchard, 14 years old pomegranates with the wonderful cultivar, harvested last year in October with around 18.2% of the soluble solids concentration. What we have did, 30 fruits of different sizes without any external mechanical or pathogenic damage were selected. The weights of each fruit were measured with precise balance, and then the fruit width, fruit height, the crown diameter, and the stem end diameters were also measured with calipers. Then the main job started. We carefully extracted all the areas by hand from the fruits and then counted them one by one. Then the total weight of the areas were also measured. What we have used as the models for predicting the numbers and the weight of the areas, we use the multiple linear regression models. We use the principal component analysis. We use the artificial neural networks and support vector machines to understand if these five parameters, the inputs as the fruit weight, fruit height, crown diameter, and the stem end diameter are related with the number of areas and the areas weights. Of course, there is and there must be a correlation or relation, but how was this relation and which parameters are impacting most, we try to understand. For performing the evaluation of the models, we use the root mean square deviation, mean absolute error, and mostly the R square to verify the models. When the R square is close to 1, we know that it provides a great accuracy. We use the R software to carry out the statistical analysis. Here we see the first results, the correlation and the multiple linear regression analysis. In these results, simply I can say that when estimating the number of areas and the areas weight, the models, of course, the impacts of different parameters are different. Some of them providing negative impacts on the correlation, some positive. And when we look to the R square values, we see that predicting the areas weight with these 5 parameters is a better way. But on the number of areas also, it provides about 70% of the accuracy in the model. Then we look to the correlations among these input parameters. We see that the highest correlations was between the fruit height and the fruit weight as expected, and the lowest was about the fruit height and the areas numbers. This is an also expectation that the number of areas is difficult to estimate with these parameters, but of course there is some ways to predict if we use more than one input parameters. And this was confirmed with the, sorry, my slides a little bit comes down. When we look to the artificial neural networks for the number of alleles, we see that the performance of the models increased when we increased the parameters. So with 3 parameters, fruit weight, and the fruit height, the R-square values was 0.7. With 4 parameters is a little bit higher, but with all parameters, it's becoming, it's providing better accuracy in the models. So we can say that the highest accuracy for the number prediction, which is not very high, but we can say moderate, can be obtained by using all these input parameters. When we look to the graphical explanation also, we see that here the actual numbers are the blue and the red ones are the predicted. So more or less close interactions, close estimation can be done with the artificial intelligence model for the number of areas. But when it comes to the area weight, we see that it's better and it's more than 95% accuracy can be obtained for the areal weight. And the other parameter that we have, other model that we have tested is the support vector machines. Again, here we see that for the number of areas, the R-square is 0.75, but for the areal weight, again, it's a little bit better, 0.88 or something like that. Again, in the graphical explanations for the number of areals, the predictions, especially for the numbers between 400 and 600 is little bit, let's say, with highest error when it is compared with the actual values and the predicted values. There is differentiation. So for the small fruits and less numbers of the areas, the prediction is becoming a little bit difficult. And finally, for the areal weight, again, we see some fluctuations in the predicted values. As a conclusion, we can say that, yes, I come to the conclusion, the best performance for the areal weight prediction can be the artificial neural network model. But of course, the support vector machine model is effective, especially for the nonlinear weight prediction. Fruit weight and the stem parameters, stem and diameter are the key parameters among these 5 parameters that we have tested. And of course, refinement of these results and large-scale industrial applications could improve sorting, grading, and handling. But of course, the studies should be expanded and with more number of replications, more number of fruits, and maybe including some other parameters, we can obtain better results and may sign a way for further studies. Thank you for listening.
We are late, so if there is a quick ask, but please very quickly. There is someone who has to ask, want to ask something, so we pass to the next presentation. Thank you.
Maybe continue doing.
Okay, thank you. So now we have Dr. Cristina Pesaro from the Valencian Institute of Agriculture in Spain. She's going to speak about the baby kiwis, the small kiwi, which generally is eaten as a fluid, and which is gaining a lot of popularity. Please.
Thank you very much. Good afternoon, everyone. Thanks to the organizer for the opportunity. Okay. Okay. As introduction, just say that baby kiwi or kiwiño, as we call it in Spain, belongs to the same family that kiwi fruit. The scientific name is Actinidia arbuta. It comes from East Asia. And in the recent years, it has begun to be grown in USA, Canada, Chile, New Zealand, and some countries of Europe. Not yet in Spain, but there is a great interest in it. Compared to kiwi fruit, the size is smaller. It's like a kumquat, more or less. The skin is edible and is rich in antioxidants, the skin. And due to the increasing interest in this crop in our area, there is a experimental orchard in the station of Villarreal, from where we harvest fruit in order to evaluate the post-harvest behavior of this crop, of this fruit, when it's grown under our agronomic conditions, agroclimatic conditions. So we carried out a cell life and the evaluation of a cell life evolution up to 14 days. And in parallel, we store fruit for up to 30 days at 1 degree. And after 15 and 50 days, we simulate a cell life period. Sorry. At all these points, we evaluate the most basic physicochemical parameters: firmness, acidity, total soluble salts, color, maturity index, and weight loss. And at these points, we perform a sensory study with consumers in order to know the first impressions of consumers when testing for the first time this fruit. The test that we perform is called word association. It involves 100 fruit consumers, but actually any of them knew or have ever tested kiwiño. It was divided in 3 steps. The first step, we show them a whole fruit, and we ask them to write down the first 2 words, image, sensation, feelings, association that came to their minds. In the second step, we gave them a fruit cut in half, and again, we asked to write down the 4 words that came to their minds. And the same after asking them to taste the fruit. In addition, after tasting, we evaluate the acceptance, the liking, and purchase intention. To evaluate acceptance, we use this scale from 1, I extremely dislike it, to 9, I extremely like it. It's a standard scale used in this kind of studies with consumers. And I would like to explain that we know that consumers don't like to use the strings of the scale, so actually we can think in a scale from 2 to 8 with these levels of liking and the same for disliking. For purchase intention, we use this other scale from I will definitely not buy to I will definitely buy. Let's see the results. I will start for the cell life period. Here we can see the evolution of the firmness. At harvest, the values were around 55 newtons. Firmness decreased gradually until day 8, and then fruit softening more markedly. And I would like to remark that the error bars represent the standard deviation. We can see that they are quite high from day 5 to day 12. That means that not all fruits soften at the same time. In fact, it was during the period between day 8 and then day 12 that most of the fruits reached the ready-to-eat stage. Regarding soluble solutes, at harvest time, the value was slightly higher than 80 bricks. And this value, 8 bricks degrees, is considered the minimum value for harvesting fruit. So the soluble solutes increased gradually, and again, we see that after day 8, there was a most pronounced increase, which was related to the softening of the fruit at this point. Here we have the acidity. It decreased gradually, and also after 8 days, we see that the decrease was more pronounced. This led to a market or a drastic increase in maturity index after this point. And here we have the weight of loss, which reached values of 6 after 12 days and 8 after 14 days. Previous studies have established 6 as the limit to preserve quality, because higher values implies a too dehydrated skin. And we can see here in the pictures, as after 14 days, the fruit is too soft, the skin is too dehydrated, and the fruit is not commercial actually. And it was between the 8 days and 12 days period where the fruit reached the ready-to-eat state. We will see now what happens during cold storage. Here we have the graphs for firmness, total soluble solus, acidity, and weight of loss. The blue lines represent the evolution during cold storage, and the orange lines the evolution after the cell life periods. If we look at the firmness, there was an importantly decrease of firmness during the first 15 days, and then fruit continued softened. And also during the cell life period, there was also a decrease of firmness. Okay. So we observe that it was during the first cell life period where most of the fruit reached the optimal stage to be eaten, and after the second cell life, fruit was too soft. And I will move now to weight of loss, because these two are the limiting factors. So I will pay attention to these two aspects. Regarding the weight of loss, the limit of 6% of weight loss was achieved during the second cell life, what means that at the end of this period, fruit had not enough quality. And again, we corroborate this with the pictures. We can see that after the second cell life, fruit was too soft, even the skin was translucent, and fruit had not enough quality. And we will see now what's happened with the word association task. When consumers, when we gave consumers the fruit as a whole, the most cited word was small, soft, olive, grapefruit, green color, or unripe. It was only after they saw the cut in half fruit that they related baby kiwi to kiwi fruit. They also use other words like small, juicy, interesting, appetizing, appealing, or juicy. So the first conclusion that we can draw is that it's necessary to include an image of the cut fruit on the label. Otherwise, people cannot relate kiwi nut to kiwi fruit. This is a package that we found in internet, and they are not showing the fruit inside, and we think that it's really important to do it. We designed this label that we use in other studies, and it works quite well. After tasting, the most cited word was sweet, and they use also other words like acid, soft, kiwi fruit, pleasant, juicy, or ready to eat and tasty. So consumers describe this fruit as soft in the mouth with a pleasant flavor reminiscent of kiwi fruit, juicy and sweeter than acidic. When we asked them how much they like it, the rates were quite high, 7.5, which is a very good result, especially we think that it's the first time that they have tasted this fruit. And the purchase intention was as high as 75% of people willing to buy a kiwi. So the conclusion, softening and dehydration were the main factors limiting post-harvest life of kiwi fruit, of baby kiwi. The cell life was about 8-12 days, and the cold storage must be no longer than 15 days. Baby kiwis were very well accepted by consumers with high liking and purchase intention rates. They were described as soft in the mouth with a pleasant flavor, reminiscent of kiwi fruit, juicy and sweeter than aphid. And an image of the cut fruit must be included on the commercial packaging so that new consumers can better understand the nature of this fruit. And that's all. Thank you for the attention.
There is any questions? Okay.
I changed the personality now. I'm not a complete expert, but I'm still very leader in this comment. What has two questions? The name of the variety, just one variety.
Yeah, Ananasaya.
The mix at the harvest time.
Okay, the variety is Ananasnaya. Okay, and the blizzard harvest were slightly higher than 8 because 8 is considered the minimum for harvesting. So we, based in the literature, we decide to harvest at this point because actually we didn't know this variety before. So we base in the information, and the minimum established is 8 bliss.
Okay, this is a difficult part, to my opinion. This at the harvest is very difficult part of the varieties. As far as I know, in Italy, some farm, also in Poland, in other countries, in other countries, they start at 6. 6 bricks to 7. But this is really a questionable point, okay? But it changes completely the shelf life and the storage capacity. And also, of course, growth quality at the end.
Yeah, we are still knowing and studying this fruit. Thank you.
Okay, and I have a question.
Okay.
Sorry, I don't know about kiwi, baby kiwi. So is there a climateric fruit or no?
Yes, it's a climateric fruit.
So if you optimize the harvesting time, do you think that in the future, with properly post-harvest technologies, the post-harvest life can be extended as the normal kiwi?
Yes, sure. We also, YNCP also works with baby kiwi. We tested, and it's not so effective like in kiwi fruit, but it has an effect. And yes, it's a possibility that combining the optimization of the harvest time and post-harvest technology, we can prolong the cell life.
Thank you. Thank you very much for your presentation. So we are going to now. Okay. Okay, sorry. Now Dr. Renato Giannini is going to discuss a presentation about Coniella granati, a real trade with plants and fruit storage. Please.
Thank you. Good morning to everybody. Thank you for the introduction. And today I will talk about the work that we done with Coniella granati regarding the susceptibility of different pomegranate cultivars. So just a few slides to introduce the topic, because the Italian pomegranate cultivation is increasing during these last years. From 2013, there was less than 100 hectares, and they increased in the last year, according to the Istat data, up to about 2,000 hectares. And this is probably due to, of course, to the introduction of new cultivars with new functional properties, and also the production techniques are improved also to get more product to collect at harvest time. There are different diseases that can affect pomegranate, fungal and bacterial disease, and according to integrated pest management, so starting from the biological means to the chemical means, the registered means are, for example, different Bacillus species, amyloliquefacens, subtilis, and again another strain of Bacillus amyloliquefacens against Alternaria potatiscinerea. There are natural substances, a mixture of eugeniogen and olentimol against Botrytis cinerea. The different copper formulates against bacterial disease like Xanthomonas and Pseudomonas caused the disease, and Cytospora punica, that is a fungal disease. And about the chemical ones, we have boscalid, ethyl phosphate aluminium, and potassium phosphonate, in this case also against Phytophthora species. It is very useful also to consider, this came from a literature research that I performed, and to consider that it is very useful to know the biological cycle of the pathogen, because they can use this information to develop forecasting models and then schedule it better the timing of application, reducing the application same, so manage the disease in a better sustainable way. And the last thing is due to always regarding the literature research, considering that later in this year it is the research it is involved to found resistant or tolerant cultivars, as this Tunisian paper that compare the susceptibility of different cultivars to Cognera granati. And again, so from the side of Cognera granati, just to introduce the pathogen, it is known as Fuma granati. It is a Colomycetes fungus, and it is important because for different reasons, two of them are because it is very spread in different pomegranate producing countries, and it can cause different disease. For example, crown rot disease. In this picture, at the end, the fungus lead the plant to the deed, and so different deed plant in the orchard are present. And it can also cause fruit rot, as in these pictures. On fruit, the fungus produce the pycnidia, that are the asexual shape of the fungus, and so as we can see in this picture in the bottom, 7 days old PDA, different black pycnidia, the detail of the pycnidia under optical microscope, and so the conidia, that are many conidia inside the pycnidia, and so they spread the disease very easy in the field. Here just few papers that after our first report in Italy and the first report of the colleagues, we have contacted of many other colleagues inside the other region of Italy, and so with this collaboration we deepened the aspect regarding Cognera granati and pomegranate. With this slide, we can go inside the work that we performed. It is carried out in two parts. One part is to assess the in vitro susceptibility of pomegranate cultivars against Cognera granati. We focused on two kinds of cultivars, the autochthonous Apulian cultivars coming from the Basile Caramia Research Center, located in Locorotondo, very good place, and 7 commercial cultivars provided by a nursery of San Michele di Bari, Viva Fortunato, and just we obtained self-rooted plant just to obtain leaves continuously. Leaves were, as you can see in the picture, placed on soft agar medium to allow that they are maintained alive for about 1 week, and then we inoculate the leaves with Cognera granati, as you can see in this picture, and symptoms that are essentially the lesion diameters were assessed for different days, and data were collected at 3 and 5 days after the inoculation. Here we can see the summary of the results from this part. In the upper part of the graph, you can see the response of the autochthonous cultivars. No statistical difference were observed among them, but we can see that Gigliola and Cardetta, for example, showed a lesion diameter more smaller with respect to the other, like, for example, Carbonelli. And regarding the commercial cultivar, Wonderful and Valenciano showed variable lesion diameter, so 2 centimeters at 3 days, and diameter, of course, fill the entire leaf at 5 days. And interestingly, Parfianga and G2 show the small lesion diameter that was statistically different to Wonderful and Valenciano, for example. And here in the middle, we can see an example of a susceptible and tolerant cultivar compared to the control inoculated with Cognella granati. The last picture is just related to the work that is in progress. We want to confirm this data inoculating other pomegranate organs like cane in this case of the cultivar Wonderful, and just assessing the lesion diameter and the severity and compare them among the different cultivars. Another part of the work was done in collaboration with the University of Padua, the colleagues Silvio Tundo and Rita Musetti in particular, because the collaboration is started because they also found Cognella granati in Veneto orchard, is a region of northern West Italy, and so we started this collaboration. The procedure is the same, and so inoculation of pomegranate plants, 2-year-old plants, isolation of the fungus, and in this case we have observed in more detail by using microscopy analysis what happened. So you can see that this study allowed us to understand that Cognella granati starts to colonize parenchyma, and then here you can see inside the vessel the IFA of Cognella granati, this one. And in this picture, the observation on the control compared to Cognella pomegranate, this is Wonderful inoculated with Cognella granati, you can see that the color is different, probably because the substances produced by the plant, again Wonderful in this case, is different in presence of the pathogen respect to the absence of the pathogen. And we want to define also, in this case, the reason, and eventually take two messages useful for the different susceptibility of pomegranate cultivars. So, as outcomes and work in progress, with this study we can observe, of course, that there is a different response to pomegranate cultivar, at least in vitro, on the tesseract leaf assay against Cognella granati. And we want to continue inoculating different tissues, different organs of the different pomegranate cultivars. There is a Tunisian study that reported that the response of a cultivar on fluid and canes is different, also with the same cultivar. And in addition, the last is that it is also important to assess how the pomegranate plants response to Cognella granati, and eventually take two messages using for the susceptibility of the different cultivars in order to manage this disease, the Cognella granati caused disease, in a sustainable way. Just another thing, because we know that Cognella granati cause also fruit rot, as I said before, but there are also different pathogens like Botrytis cinerea, Alternaria alternata, and Aspergillus species. These last two are important because they can produce mycotoxin, ochratoxin A. First of all, that there is an important aspect for the accumulation of the toxin in the juice, for example, and there is a poster presented here at the conference, and we can talk in front of it if you need some explanation. It is just regarding the use of trichoderma isolates in vitro, and we are we are moving on in vivo to manage this pathogen in the field and also in post-harvest. Before to go to the conclusion, just another slide, because Cognella granati, I said before that I'm arriving. I said before that it is involved on crown rot disease, and so wood disease, but recently we reported also other pathogens, the aborted species, Neopestaloxopsis, Opioscioma, and Xenotremonium, that we presented like a poster. You can scan the QR code if you are interested in it. That can affect, of course, the pomegranate, and this was presented at the NPU conference that was in Bari in the first days of July. So I have concluded, and just a last slide to thank the funding that allowed us to produce this research, the people involved in the work, the whole research group, and thank you all of you for the attention. Thank you.
Thank you for your presentation. Is there a question? Okay.
Yeah, thank you for the presentation. It was particularly interesting to me. Did you find any significant differences in incidence of Coronella in orchards and as post-harvest experience?
Okay. Thank you for your question, first of all. Yes, we found difference in the field, of course. Just a number to give an idea, 20% in average, but it's dependent from the material coming from the nursery, how two plants were managed. So if we remove fastly the plant, the disease spread very slow in the field. Okay? In post-harvest, regarding Coronella granati, really it can be very a problem, but in post-harvest mainly botrytis is a problem for Coronella granati. But there are other correlates like me, for example, on my section that can perform a research within it. There are also literature is very rich in this regard.
Two things. One, Coronella granati is Coronella granati Saccardo. Who's Saccardo? Because my uncle is Saccardo. So I went to study it. He was the director of the botanical garden of Padua, one of the oldest botanical gardens in Italy. He found there was another name at that time, the last century, in Treviso, which is nearby my town, Verona, is in Berkovich. So that means something. Anyway, the question is, your results are orientative or substantial? I mean, Parfianca, which is the less affected, is really more tolerant or not?
Good point. On leaf, yes. On leaf, yes, of course, because we do experiment, we have replicates, so on leaf, yes. But there is a very interesting study regarding the inoculation of other cultivar, not Parfianca, and there is a difference regarding in the response to inoculation of Coronella granati. We are talking about tolerance, not resistance. And there is a difference on the same cultivar regarding the organ. And so, I don't know. I have results about leaves. I started the experiment regarding sticks, so just canes. I don't know. I have to try, and when I have results, I will share. And so that's it. I hope.
Okay. So grey mold is a great problem for post-harvest management of grape fluids. Probably in the next future, synthetic fungicides will be completely banned. And now the Professor Baghi is going to discuss eco-friendly treatments done with the essential oil extracted from Baisapuleia montana. Okay, so please. Thank you.
Good afternoon, ladies and gentlemen. I start my presentation regarding the eco-friendly control of post-harvest gray mold by using of the savory oil. This research has been done by the private research institute in Iran, by the Fruit Science Company, and also the partnership of the French company Zeta International, that I'm the international technical manager of the company. Okay. In regard with the pomegranates, yeah, it's about 7 to 8% of the global pomegranates will decay every year. And also all the post-harvest challenges are chilling, injury, peel, scald, weight loss, and decay. And also the now post-harvest technology till now, which has been commercialized, it is controlled atmosphere and modified atmosphere packaging. In regard with the Iran, we produce 900,000 tons to 1.3 million tons per year. The most important part of the pomegranate production is Fars province, which the city Neiris is very famous. Though there is as well the Isfahan, also we have production in the Khorasan and the other countries, and more or less we have pomegranate everywhere in the country. So we export about the 30% of the production, and we have a big decay as well, about 30%. In regard with the post-harvest technology and the history of the post-harvest technologies in Iran, it's a very ancient model of the controlled atmosphere or modified atmosphere. They put the bins, the wooden bins inside the clay mix, and then they store from the October to late March. And they use these pomegranates as a very respective fruit for celebration, for Nowruz celebration at end of the March, and also for gifting, for the weddings and these things. So it is a very, very ancient, maybe the most ancient documented technology of pomegranate storage. The other, the point of the decay in my country, the most important pathogen is Botrytis cinerea. So we lose about 30 to 50%. And it is only as the primary pathogen. And after infection by the botrytis, we have the infection of the other pathogen like penicillium as well. So what about the importance of the treatment, post-harvest treatments of the pomegranates? Pomegranates in my country, it is more or less 80% organic production. They don't use any chemical or pesticides for pomegranates production. So keeping the safety of the fruit and chemical and residue free is very important. And one of the approaches that we are following is application of the essential oils, and the other point is the application. So today we had presented a lot of coatings and a lot of treatments, but it is important that the industry could be used these technologies. So coating or waxing or polishing of the pomegranates is quite not accepted too much by the industry because it damages the crown of the pomegranates. But fogging it could be a very good technology. Yeah, the process that we did is to have a sustainable supply of the disease control for the pomegranates. We collected out of the scientific literature 17 essential oils, and then out of these essential oils, we ordered from the sustainable and accessible suppliers to suppliers in the world. We ordered 17 essential oils. We screened 4 essential oils, and then after we did the other part of the formulation and the pathogen control assessment and the final formulation basis on the project done in the 2020 to 2022. Out of this screening of different things, you see different suppliers sometimes have different quality because the essential comes from the nature, and depending on the climate, altitude, and other parameters, the components and ingredients are different. But for 4 essential oils, we had the more or less the highest control and also the same quality of the 2 suppliers. So out of the thyme oil, clove oil, savory oil, and cinnamon oil, we choose the savory oil, and I will explain you in the discussion why we choose this essential oil. So the procedure that we did after the screening of the savory oil, we did the first the plant material. We used the Rabab and Neiris, which is the most important export variety, which has a red peel and also very dark red ions also. and also about the fungal inoculation and the treatment. We treated in different way. We did the hot water brushing and we did also the fogging. And about the pathogenicity tests and essential oil, MIC and MI assessments. About the trials, as you see here, we did a prototype hot water brushing system to treat, and also a spray brushing system in different concentrations of the soury oil by the hot water brushing, and also as a market control, we use fluodeoxonil and primethanil at 2000 ppm, and also we use the hot fogging of the soury and as well the control. You see here the trial, the fogging trial that was done, and it is the industrial that we use in every year. We treat around 100,000 tons to 85,000 tons of pomegranates in Iran by hot fogging of the soury oil. In regard to the disease control, you see that in 500 ppm, the control starts, but we have the full control 1,000, and our recommendation is 5,000 for the field trials. In regard to the assessment of the important point is that the golden time, the time between the harvest and treatment, that we can reach to the curative effect. So as you see here, all of the treatments has had a good efficiency, and here is the hot water brushing, and here is hot fogging. But when it extends the delay between the harvest and the treatment, you see also the control. It is working fine in both. It has a good control. But when we delay to 48 hours, the control it has been changed, and we see some scalds on the surface of the fruits. In regard to the controls that we reached, we had a very good control by the hot fogging in the 10 milliliter per cubic meter. We had a good control in the in after 12 hours and after 24 hours and 48 hours. Also, we had good control by the chemicals, but the best came from the hot fogging. And we see when we use the water has a good control by the mixing with the fungicides, but also the water by penetration inside, it makes also some decay. So it's the reason that the hot fogging has a better effect than the water treatments. The other point is about the severity. It's very important that because when we have a good control but high severity, it has no market value. So as you see here also in the hot fogging, we have less severity. So it could be also another point. So as a conclusion, in regard to the severity and the disease incidence, it's important to know that we find out the Saver do Montagna or soury oil. It shows a very good antifungal activity against the botrytis and even the secondary disease that comes after the botrytis. The other point is that the golden time and the time, the best time recommended, we are facing with the natural product to have a better persistency of the activation. We recommend 12 hours after harvest, but it works still less than 24 hours. About the effective treatments, we recommend 5,000 ppm or 0.5% for hot water brushing, and also 10 milliliter per cubic meter. It means that around 50 per ton. The other part about it is a sustainable solution and chemical-free solution. About the mechanism of activity, I told you why we choose the soury, because soury, the ingredient of the soury, thymol, carvacrol, it works in different mechanism control the disease. It disrupts the membrane wall, it disrupts the energy system of the disease, the mitochondria, and also the pumps for the transportation of the product. So these have weapons in three ways that affect. And also the other point is that to control the severity, the balance between the oxidants and the antioxidants inside the soury oil is very nice balance, because the ROS comes from the fruit, also comes from the disease. It's very important that not only antioxidant, you should have a balance between the oxidant and antioxidant. And the last point is that the idea came from here. The best solution is non-contact treatments for pomegranates. That we designed a new system that vaporized superhumidity. What is the idea? As we know, based on our understanding, the most important to control the weight loss of the fruit and avoid the host scald or chilling injury is humidity level. So by the superhumidity level, by vaporizing the water molecule and attaching the essential oil, by the percentage of 99.9%, we can have a very good weight loss control and very good disease control. And what is the idea? So in control atmosphere, we reduce the oxygen or we increase a bit the CO2, so we reduce the respiration and we control the weight loss. What happens if from this side, we increase the humidity, and we control also the respiration. So we got very good results out of the apples in France. So it is the future program for the technology for superhumidity vaporization of the essential oils for future. I appreciate my colleagues in Iran, the scientists, and also my colleagues in the France. Thank you very much.
Thank you very much, Professor Badi. Is there a question, please? Okay, I have a question.
Thank you very much.
I wonder if this essential oil can be phytotoxic for the peel of the fruits, and if they leave some off flavor or at least the odor of the essential oil, because very often a lot of these oil compounds alter the flavor of the fruits.
Yeah, very good, very nice question. First, about the peel change and about the phytotoxicity, you're totally right. The combination, and it is each essential oils has something like a fingerprint. The component, for example, the beta-caryophyllene or alpha-pinene, they are less than 1%, but this percentage are very, very important. So the composition is very important, and the point that we concluded in this research is, for example, if the thymol goes up more than the 35%, you will have the phytotoxicity. If you usually increase more than 60%, you will see the phytotoxicity. So there are many learning points. The other point about the smell.
Yes.
How we can cover the smell. The first is that the application is encapsulation in the products, that we can encapsulate the product. By encapsulation, by nano encapsulation, we can delay and we can cover the smell of the fruit and the flavor change. The second approach is packing of the nano particles in the nano corridors, nano crystals like hydroxyapatite. So these two approaches are for covering the smell.
So this microencapsulation maybe can in the future use this product in the field to control gray mold and prevent latent infection, which probably we are not able to control with this treatment, because you can control only recent wound pathogens, but not all the established pathogens with your treatment. Is it right or not?
Can you repeat again?
I mean that you treated the fruit within 24 hours before artificial inoculation, and the treatment worked well. But what about latent infection? Infection that you have in the field, so you are not able to control in this way. You don't control that kind of infections.
Yeah, the infection, the botrytis, the most important, it is a wounded disease. So normally, if even attack to the fruit when the wounds happen during the handling, harvesting, or something like this, it is important that this time it's attached to the wound and start the proliferation. So it's important for this part. And the other point is that about the controlling the disease, the curative effect. The curative effect, it is essential oil. It is not chemical. So it's important that what we know with which approach, it is chemical free, it is healthy, but the point is that we should have a procedure in the pre-harvest to reduce the mechanical damages and everything to have the maximum efficiency of this product.
Okay.
Thank you.
Thank you. Thank you very much. Now there is the last presentation of the section. To present this work is Dr. Naya Fabadi. Is going to speak about margarine, very nice superfood with a very short shelf life. So you tell us how to manage this food. Thank you.
Hello everyone. I am Ayub Fati, and today I'm going to talk about an overview about the research work about the characterization of mulberry and how the fruits ripen and behave after the harvest. And this context is very important to understand the ripening of the fruit in order to improve the storage and utilization of the fruit. So the title of our work is Characterization and Post-Harvest Behavior of Mulberry During Ripening. Mulberry is cultivated widely, especially in Asia, and China has around 626,000 hectares. However, many countries in Asia, Near East, and part of Latin America also grow mulberry for fruit consumption. And at the present, mulberry extract market is growing, driven by demand in functional foods, dietary supplements, cosmetic, and so on. And why is global mulberry consumption rising? It's because of health benefit that they are rich in antioxidant, phenols, vitamins, and research attributes these anti-inflammatory, anti-cancer, and anti-diabetic effects to this compound found in the mulberry fruit. And it is because of plant-based demand that it fits the global trend for natural plant-based superfoods. And mulberries are very perishable, and for that reason they are widely processed, which helps extend their availability beyond the short fresh season. And they are also used in traditional medicine and has long history of use in Asian and Mediterranean diets for health and wellness. And as it was mentioned, mulberries are widely processed into products such as jams, jellies, stews, dried fruit, powder, frozen mulberry, or fermented products from mulberry like mulberry wine or vinegar. However, fresh mulberry fruit is rarely commercialized due to its limited harvesting season and storage sensitivity, during which a considerable portion of the fruit is ruined, resulting in a significant economic loss. So the challenge we have is to improve approach to sustain and extend the shelf life fresh mulberry fruits. But currently there is a lack of information on harvest and post-harvest characteristics. So the first step to resolve this problem is to understand the harvest and post-harvest properties of this fruit. So having outlined this background and challenges, this study was driven in two main objectives that were understanding the metabolic behavior, metabolic indicators, and physicochemical parameters of mulberry at harvest, and also determining the effect of storage on the changes of physical chemical parameters. So in this study, the mulberries were harvested from an orchard in Foggia, Italy at the end of the June, and the fruits were classified in 9 classes of ripeness. And later, the metabolic behavior and physicochemical parameters were assessed, and we also measured these parameters after the 3 days of storage at room temperature. Now I'm going to show the results. That this graph shows the variation across maturity stage. That at beginning, for example, mulberries in stage 1 had about 0.5 gram. It was a greenish color and with the hue around 137. And this, during the maturity stage, these parameters increased, and at fully maturity, berries weighted about 5 to 6 grams and showed deep red-purple color. And the color change reflects chlorophyll loss and anthocyanin build up during ripening. And also mulberries typically progress from green to red, then dark purple-black during ripening. And this graph showed the firmness decline, that in the stage 1, the firmness was about 23 newtons and increased to almost 2 newtons, and it's reflecting the cell wall pectin breakdown. And we also measured the soluble solids accumulation in the fruit, that in the it rose from almost 8% to almost 20% at stage 9, showing steady sugar accumulation. And regarding acidity and pH change, ripening reduced acidity and raised pH from almost 4.6 in ripe to 7.1 in ripe berries. And this shift in sugar and acid raised the sugar to acid ratio, giving ripe mulberries their sweeter taste. And one of the central questions of this study was whether the mulberries were climatic or no climatic. So after measuring the ethylene production and respiration rate, we found that in the intermediate stage of maturity stage, meaning that in stage 4, 5, 6, we had a peak in ethylene production and CO2 production. But by the contrast, in the early stage, these production are very low or steady, and in the late stage, they decrease. And regarding the total phenol compounds and antioxidant capacity, in the early mid stage, the total phenol is relatively low and stable, but in the later stage, there is a strong increase in these compounds. And relating to antioxidant capacity, it is very high at the beginning, stage 1, likely due to elevated level of primary antioxidant. And it decreased a little bit, but after that, from stage 7 to stage 9, antioxidant rise again, peaking in stage 8 and 9. And regarding to vitamin C and anthocyanin, in early to mid stage, vitamin C levels remain relatively stable, and a sharp rise occurs at stage 5, and where ascorbic acid vitamin C content reaches its maximum. And after this peak, the levels gradually decline. And regarding the anthocyanin, levels are nearly absent or very low at the early stage, but they increase, they start increase moderately, and a steep increase occurs, peaking at stage 8 and 9. And regarding to sugars and organic acid, in the earlier stage, sugars are almost absent. And in stage 3 to 5, sugars begin to accumulate with total sugar reaching to 30-40 grams per kilogram fresh weight, and it decreased during the rest of the maturity stage. And regarding the acids, they are abundant, dominated by malic acid and citric acid, but in the almost mid stage 4 to 6, both malic and citric acid contents drop considerably. And here I am going to show the result of physicochemical parameters after the storage at 20 degrees centigrade after the 3 days. That in the intermediate stage, in the stage 5 and stage 6, we had a significant difference between the color in day 0 and day 3, and in other parameters, especially the important parameters that are soluble solid contents and pH, we had a significant difference between the values between day 0 and day 3. However, in early stage, the fruit failed to ripen, and in the late stage, the fruit exhibited and manifested senescence. So from this study, we conclude that mulberries were characterized in 9 ripeness stages based on metabolic behavior and compositional change of the fruit. Mulberries exhibited climatic-like behavior, particularly pronounced in intermediate maturity stage, which displayed distinct ethylene peaks. Earlier stage failed to ripen properly, and later stage exhibited senescence. Fruit should be harvested at stage 5 or 6 to match their climatic peak and ensure optimal sugar development. However, to confirm mulberry's climatic nature, further studies are needed, including ripening response to exogenous ethylene and its inhibition by 1-MCP. In parallel, examining the expression of key ethylene biosynthesis and signaling genes would provide molecular evidence to validate the climatic-like behavior observed in this study. And in the end, I wanted to acknowledge the persons that they made this study possible, and a special thanks to University of Foggia and Post-Harvest Group. And I wanted to thank Professor Colelli and Amudio to supervise this work. Thank you for your attention.
Thank you very much for this presentation from Mr. Badi. And is there any question? Okay, thank you very much.
You're welcome.
So, with this presentation, we conclude this section, this afternoon section. And now, Professor Carrara is okay.
Just want to notice your communication for the tomorrow morning visit to the Masseria Frutti Rossi farm. The meeting point will be at 8 o'clock close to the railway station. So it's not outside this building, but it's maybe 200-300 meters far away from me, close to the, you can see here on the map, and you can also scan the QR code. You all received an email with this information, and also on the entrance there is a paper with all this information. So tomorrow morning, meeting point close to railway station, Piazza Sorrentino is the name, and we'll be leaving at 8:30. So we will be back for lunchtime at the same restaurant. It's everything clear. If you have doubts, you can write, but you will be received. You already received the email, okay? And now there is a video presentation of the next sponsor, Unitec. And then after this presentation, there will be the genetic session with the chairperson of Ilaria Marcotu.
Sì, finiamo questa cosa. No. Dimmi di carico Marco.
So now we will be showing this video.
Hi everybody, my name is Laura Vignoli. I'm from company Unitech, and today we have been given the chance to participate in a conference. First thing, I really would like to thank you for the invitation and for giving us the possibility to give this short introduction on our company and on our post-harvest technology. So the first big thank you is to all the organization behind the 6th International Symposium. What I would like to do today with you is giving a brief introduction on Unitech, our company, and then to explore together all the food service technology and sorting possibility that we have for the main product of this symposium, the company. So starting from the company, Unitech is an Italian company. We are based in Lugo in Emilia-Romagna. And for over 100 years, we have been dedicating ourselves to the manufacturing and designing of the sorting technologies. Currently, we can work with more than 50 different types of fruit and vegetables, and we are active in more than 65 countries in the world. We basically operate directly, or we operate thanks to the help of branches and of dealers and agents. Whatever we manufacture and we design is fully, basically fully done in-house, meaning that we take care of the 100% of the process. In Unitech, we start from the designing of the technologies and we take care of all the aspects from mechanics, electric and electronics development. That follows up then with, of course, the commissioning and installation of our machinery, so that can be integrated also with automation and addressability systems. Among all the technologies that we care of, probably the core business of the company is the vision system for external quality selection. What you see here represented is basically the list of products we work with and type of technology we can work. We have mainly two different types of technology. What we call vision, that represents the work of the external quality for products, and what we call unique, so quantitative internal quality, that covers the range of technology to inspect the internal characteristics of each food. So, thinking specifically in pomegranate, what we can offer today is a pomegranate vision to see the good surface of the fruit and to separate the good from the bad. And the unique pomegranate that represents instead the tools that we can offer to establish when a fruit is good inside and when one is bad. Talking specifically about pomegranates, what we can offer today is a complete series of technologies that covers from the product intake till its packaging, meaning that we care of the fruit arriving to the packers, and we are able to offer some solutions for manual or automatic depalletizing and tipping. Of course, when it comes to beans, since in some markets this product is also harvested in beans, we have the possibility to offer automatic tip with both dry or wet infeed. Then normally, let's say the process continues with a manual pre-sorting of this fruit and its cleaning. In terms of cleaning technologies, depending on the market, we can offer different types of solutions from a single brusher, or we can also include the boxing technologies. And then, of course, we go more into our sorting aspects, meaning that of course we take care of the alignment, quality selection, and the packaging of each fruit. If needed, of course, the solution can integrate also the site technology for palletization, strapping, or traceability. As I was mentioning before, our technologies for pomegranate are basically two: the pomegranate vision for the external quality and unique pomegranate. We talk about pomegranate vision, the innovation that we can offer is the possibility to inspect the totality of the fruit, making each fruit rotating 360 degrees and being able to take photos of each single area of the fruit, and based on that, being able to assign a class to each specific fruit. When we go inside on internal quality, what we can do is basically being able to see when a fruit starts to develop internal blackening. I will now go a bit more in details on the two technology. When it comes to pomegranate vision, what we can do today is basically being able to separate the fruit according to each single weight of the fruit, the size of the fruit, and also being able to, of course, use the two parameters, weight and size in combination, to being able also to identify the density of the fruit. Of course, we can integrate the color classification, so being able to separate the fruit that are not 100% red and that present, for example, discoloration or yellow spots. And of course, we go also on external quality code, whatever implies basically seeing the effects on the fruit surface. What you see here in the photo is basically an example of the application of this technology. So we have a protein synthesizer with the pomegranate vision tunnel on top. The vision tunnel, of course, includes inside a set of cameras that are able to take the photos to each single fruit. And these are a bit the impact that we're able to see on each fruit. So when it comes to pomegranate, I believe that probably the most complicated and the most important defect to see are, in terms of external vision, the crackings. That can be, of course, both, let's say, fresh cracks, so when the fruit is open, but also closed or healed cracks. Together with that, of course, during harvesting, it's possible to create some mechanical damage of the fruit that can be both from puncture, branch punctures, or phenomena of peeling, for example. All these type of defects can be checked and seen by the vision systems. And of course, together with the mechanical damage, we also have some, let's say, natural type of defect that can arise in the presence of russet, or in the worst cases, the presence of decay on the surface of the fruit. What comes next is, of course, the possibility to see some burns, as we were saying, also the possibility to see discoloration of the fruit, together with the frost damages. The presence of leaf that you see in the bottom is, of course, something that the vision system can see. It's more, let's say, a warning to see that maybe it's not possible to classify properly this type of product. So, let's say, what you see here represented there is more or less the ranges of defects that it's possible to see without the need of a human operator. When it comes instead on to internal quality, what we can offer with our unique pomegranate is the possibility to identify the development of internal browning, normally depending from the presence of alternaria or botrytis, to a very precise and reliable degree. Meaning that it's possible to see these defects also when it's just, at the very beginning of development. Of course, the advantage that this kind of system can offer is that, again, it's a system that does not require the human being to be involved in the process. So it's a completely automatic system, and you now see represented in the photo, again, the tunnel with the technology for internal quality inspection. The interesting thing probably again about this type of technology is the possibility to use it at a different level. So you see that it's possible to see the blackening when it's just affecting a few areas. So when the blackening is very beginning and a very, say, initial stage of the opening. At the same time, you see that we are also able to see when it's affecting just one section of the fluid. So the particular thing about the polymers is that they do not have, if we compare that to other fluids, a uniform flash inside. So we can decide, of course, how deep we're going to have this classification. So from pure layers to sector blackening, till the end when we have complete blackening inside. Unique and vision systems are definitely, I think, extremely helpful technology to be used when it comes to component classification. One of the advantages of using this technology is that it can be, I would call them flexible, meaning that this technology can be longer. It can be used on a simple sampling line, so a line dedicated to the sampling, to the quality control of batches of fruit, and at the same time, they can be installed on sorting machines. Also on the sorting line, we have the possibility to install this system, not only complete lines, so not on all the lengths of the machine, but just also on a few lengths, maybe on one length or two lengths, again, with the possibility to combine the sorting with some sampling activities. If I have to make a sum up, which are the advantages of using this type of technology when it comes to an automatic system for sorting and packing fruit, I would say that of course some advantages are shared between sampling and sorting activities, and we can see that together, of course. Definitely, unique and vision system in both cases allows to have an undestructive analysis of each single fruit, meaning that there is no risk to ruin the fruit. There is no need of preparation, so when the fruit arrives to the pack house, they can be simply put into the line, and then it's going to be just the line that will take care of the sorting process. Of course, this reduces the human labor and the workforce needed for the sorting and packing operation. It's a very fast type of classification because it requires a few seconds to take the photo and analyze and giving the proper classification to each fruit. Of course, once we combine vision and unique, we start getting more and more information on the fruit, the weight, the size, the color, information on the external quality and on the internal. And of course, this allows to get not only the data from the batch, but also to start building the information on what is coming inside our packers. All these data, all the information we got from the machine can be exported, so meaning that you can start building your reporting on the quality of the fruit that then goes into the pack house. And of course, they allow, especially for the sampling operation, the possibility to repeat, to do more, say, the same type of task, but more and more time. This means, of course, creating also a standard. Then, of course, if we apply this technology to the sorting, we have also possibility to, of course, eliminate whatever is not good from our pack up, and of course, we have the possibility to divide each batch into different classes. So I believe that the application of these technologies to the sorting process is definitely one of the most important things that we can do to speed up the sorting process, but most of all to supply our final customers with consistent quality. And we are able with this technology to guarantee the quality of our food. My presentation is finished. Again, I would like to thank you for your time. What you see here is a QR code also with my details. So should you need any information or would you like to get information about Unitech and the technology that we can offer, just feel free to contact us. Thank you again.
Okay, thanks for listening. Now it's the genetic session. And Ilara Marcotuli will manage this session.
Good afternoon to everyone. So we start with the genetic section, and we start with the first speaker, Zanao Deng, from University of California, USA. And his presentation is on improving pomegranate resistance to Coleophilum goriosporides, identification of sources of resistance, dissection of mode of inheritance, and development of genomic resources.
Okay, thank you. Thank you very much for your introduction, and also I'd like to thank the organizing committee and scientific committee for this opportunity. Thank you so much for organizing this symposium. It's a huge effort. As I mentioned, my talk this afternoon will focus on improving pomegranate resistance to tetratrichine. So as several speakers mentioned, in the United States, the pomegranate production is mainly for concentrated in California on the West Coast. And in recent years, there are some effort in Arizona, Utah. They're trying to grow a pomegranate there, and the climate there is the same area. But also in the southeast United States and the south United States, Texas, Florida, Georgia, Alabama. So we are trying to grow pomegranate there. But in our area in the southeast United States, so our climate is characterized with a high temperature. So high temperature in the summer, and also lots of rain, which is very typical in southeast Florida. So like in several months, we almost have daily rains every day. So this creates a big problem. So in the springtime, you can see our water looks pretty nice. But once the rainy season started in May or June, then you can see the leaves became with many leaf spots. And then the fruit, you can see they have all kinds of fruit rot on the plants. So with this diseases on the leaf and on the fruit, we think disease-resistant varieties are absolutely essential for us to grow pomegranate in southeast Florida. Okay, so first I will focus on finding resistance to fruit rot. So in our field trial, we can say in March, April, we don't see fruit rot, but we starting in April, we started say fruit rot, and by probably I guess here change the I guess it's so by June, you can say with some varieties, almost all the fruit became rotted. So that's a big problem in southeast United States. So to find the disease-resistant varieties, so we screened the 35 varieties in the field in 2021 and 2022. And in this case, we grow the opacity in the lab, and they produce suspension of the spore. Then we inoculate that into the young open flowers or young fruit, and then we check the fruit almost every week, and they see how big are they leaching on the fruit. Then based on that, and also we inoculate several fruit with just water without the opacity, and also we bag some fruit. In this case, just natural infection. So then based on the size of the lesion, then we convert into a scale of 0 to 6 based on the severity of the fruit rot. And you can see we have very good correlation between natural infection and also artificial infection in 2021.
Also very good coordination in 2022, and also good in both years. And also coordination between the 2022 and 2021. So I think our data is pretty reliable, and based on that, here I'm showing just the data from 30 varieties. And you can see, so for each variety, we have 4 bars reflecting the artificial inoculation in 2021, natural infection in 2021, the artificial inoculation in 2022, and then natural infection in 2022. So for most of the varieties, you see pretty consistent. But in some cases, like in this one, you say quite I guess variation from year to year. But overall, we find out 6 varieties: Arecta, Azadi, Eversweet, and the Fleshma, and the JB Rogue. They have very good resistance to foot rot with much less severity, especially Azadi with needle foot rot after inoculation and also natural infection. And then also we want to see whether this resistance is expressed on mature fruit. So we shipped fruit from California, from the USDA germplasm to Florida. Then we make a wound on the fruit and inoculate the fruit with spore suspension, and then you can see big differences among the different varieties. Like this one, you rarely see any rot, but this one is a big area, and in some cases you can see the rot getting inside, and in this case just limited to the skin. So anyway, we think anthracnose fruit rot resistance is expressed on the mature fruit. So this will help us to simplify, future screening process. So that means we can use a mature fruit for screening for disease resistance. And then here we list these 6 varieties with good resistance. And you can see, 2 of them are from California, including Eversweet, which Dr. Chater mentioned that was selected by his grandfather. And also, we found that 2 are from Florida. They have showing resistance. But also interestingly, we one from India and one from Turkmenistan, Azadi, also showed a very good resistance in Florida, which is quite far from India or Turkmenistan. So this indicates that the resistant varieties have diverse origin. And then, so Azadi has this light color, Eversweet and Christine, they all have light skin color, and also very light color inside, every color. So at this time, we don't know if there's any association between foot rot resistance and the fruit and area color. It appears so, but we don't know, because Arecta has a more red skin, more red area. I think that's a brief presentation of our searching for foot rot resistance. Now I'm going to sketch switch gear and talk about the leaf spot resistance. So in our field in 2021-22, we noticed there's a big difference among the 31 varieties. Like you see, several varieties, they have very little, very light leaf spot disease, but there are some other varieties very severe. So this is on a scale of 0 to 4. So overall, based on this is in the field. From the field, we can see that Boris number 2, Bana, Miro, JBRO, Narkin, and they have a pretty good resistance. And on the other hand, the Purple Heart, Bagua, Wonderful, Gherkinuts, they are pretty susceptible to leaf spot diseases in the field without inoculation. So to confirm resistance, we collected the cuttings from 10 varieties and then root the cuttings in the greenhouse. Then we spread the cuttings or young plants with a suspension of this bacteria, fungal pathogen. And then after that, we counted how many leaf spots on the top 5 leaves. So this are the number of spots on each leaf. And again, we can say resistance in Azadi, Boris number 2, and the puffy anchor. And then also we see some variation from different experiments. In this case, we knocked the cuttings in 3 experiments. So we repeated 3 times. Okay. And then again, this 3 varieties has good resistance. Subsequently, we also inoculated the seedlings because in the previous trial, we only could work with 10 varieties. Now we would like to work with 34 varieties. So we have the seedlings, inoculated seedlings with the pathogen, like showing here. And then we see big differences among the 34 varieties. We can say Desertini, Boya, Napis, Eversweet, and Azadi, and the Down Summer South. So they have a pretty good resistance. Okay. So subsequently, we selected 9 varieties, and the 9 varieties we crossed and made 26 populations. And with some of the, with several hundred, 5 to nearly 300 seedlings for each population. So based on this data, we find out, so we have some very good data. So in most cases, we see segregation like this, like a normal distribution. You get some resistant, some highly susceptible, but most of them are in the middle, moderate resistant or moderate susceptible. But in some cases, we say, most of the seedlings are very susceptible, and in some cases, most of the seedlings are very resistant. And interestingly, we found out that the broad sense heritability was really low, about 2.1. And in some cases, we found out when we cross 2 susceptible varieties, we have very good resistance, very highly resistant seedlings. So this means there's a good potential to develop new varieties with leaf spot resistance. So anyway, in summary, we find 12 varieties, 7 with resistance to food rot and several with leaf spot rot. And most importantly, Azadi is highly resistant to anthracite fruit rot and also moderate resistant to leaf spot. And then we sequence the genome of Azadi on the PacBio, sequence to 31X coverage, and also we use the high-C sequencing to face the genome. And then we use RNA data from our experiment and also 19 RNA sequence data from published studies. So we were able to generate a high-quality genome of Azadi with 2 haplotypes. So the first haplotype is about 305 megabase, the second about 380 megabase. And the largest chromosome is 55 to 62 megabase. And then we identified 28 to nearly 30,000 genes. And also we identified several structural changes in both haplotypes. Yeah. And then also we characterize resistant genes in this genome. As you know, there is the NBS containing genes is the largest disease resistant class, and there are several subclasses. We found out that in the other genome, we have other genome has about 383 NBS containing R genes, and then they have about 410 transcripts, and then there are some other genes, R genes on every chromosome, and the chromosome 8 has many R genes. And most of them are clustered, and then we found out other D has 11 unique R genes. Okay. And then we also, in collaborating with Dr. Chater, that we sequenced the 58 varieties and genotypes, and then we identified R genes in these varieties. And we found that 46 R genes has a high impact SNPs in theirs, and then two genes that have contaminated high impact SNPs. And we found that we look at the 19 database, and then we found about 88 T and L subclass R genes expressed, and about 50 to 64 C and L subclass R genes expressed, and then 32 to 37 NL genes, then 5 R and L subclass genes expressed in pomegranate. So overall, the root and the leaf have the aerial have the highest number of R genes expressed, and then in leaf and flower fruit we have less R genes expressed. And okay, so overall we found sources of resistance to anthrax, nose fruit rot, and sources of resistance to leaf spot disease. And then also we create the breeding and the secreting populations. We have data on inheritance of leaf spot resistance. We create the genome assembly and they resequence the 58 genomes and identify R genes. Okay, so this picture I took in Georgia. We are still struggling in Florida. Hopefully, with our research, we can in the future we can produce this beautiful pomegranate in Florida. Okay, thank you very much.
We don't have time for questions, so we have to pass to the next speaker. Sorry, we are running out of time.
All right, okay, thank you.
So the next speaker is Cristina Besada from the Valencian Institute for Agricultural Research in Spain. And her presentation is a new horizon for pomegranates, red coloration and in-mount properties of molar vert in a new hybrid.
Okay, hello again. As introduction, I would like to comment you the results from one of our previous study. In we asked 320 people if they used to eat pomegranate during the season. 55% of them say yes, I am an habitual consumer. And we asked them the reasons to consume. And as you can see here, the main reason by far was I really like them. That means that for consumers, quality is related to organoleptic quality rather than nutritional quality or other properties of the fruit. And when we talk about consumers and organoleptic quality, we can differentiate 3 stages of quality evaluation. The first one is when people are at the supermarket and they will evaluate the appearance of the fruit. At this point, they will also receive information through the labels and the price, and based on all these aspects, they will decide if buy it or not. The second stage is the peeling and preparation. At this point, they will perceive if the fruit is easy or difficult to peel. They will also evaluate for the first time the appearance of the arils, and the first aroma will be released. And the second stage is the consumption itself, where we will perceive textural properties in the mouth, taste properties, and also the flavor. Say that, I will move to the study I want to show you today. In Spain, we have a breeding program, which is a collaboration between the experimental station of Elche and the institution where I do my work. And the objective of my team is to include sensory studies and consumer preferences as a key step in the selection process of new varieties. And there is a premise that is quite obvious, but we need to keep in mind that for a new variety to succeed, it must have a quality that consumers perceive as equal to or higher than that of the varieties already available. So the objective of this study was to evaluate two new preselected varieties compared to their parentals that are commercial varieties. And they have the same period of harvesting, which is important to know. The two parentals were Moyar and Wonderful, that all of you know quite well, and the new varieties were H327 and D2712. At harvest, we evaluate the main physicochemical parameters, including the parameters related to the seeds, and we perform a sensory study with 94 consumers, and I will focus on the sensory study. Based on these three stages that I just commented, we asked them how much do you like the appearance of the fruit, how much do you like the appearance of the arils, and then they tasted the sample and we asked how much do you like the sample you just tested. And they asked them to taste the sample again and check from this list of attributes, of sensory attributes, all those attributes that they were perceiving in the sample that they have just tasted. And as you can see, the attributes, the list of attributes include attributes related to taste, like the intensity of sweetness, the intensity of acidity, the bitterness. Attributes related to texture, like the astringency, juiceless, juicy, also related to how hard or soft was the seeds and the arils. And attributes related to the flavor, to the intensity of pomegranate flavor or intensity of woody flavor, which is related to the seeds. So let's see the results. I will start with the internal quality after testing, what they say after testing. As we have many consumers in the study, we were able to perform cluster analysis, which allow us to differentiate different preference patterns among consumers. So we found three groups of consumers. The first group, with 20% of participants, clearly show preferences by Wonderful. The second group show exactly the opposite pattern. They dislike Wonderful and like a lot the other three varieties. And the last group only liked Moyar and D2712, with 34% of participants. So if we look at the whole data, Moyar and D2712 were the two varieties that liked it to most of the people. Okay, we will see how they describe each variety. Here we have the corresponding analysis between the samples and the sensory attributes. In this graph, if the samples are close to each other, that means that the varieties were very similar, were described similar as with similar attributes. And if they are opposite, far away in the graph, that means that they were very different. So, Moyar and D2712 were very similar and were described with attributes like sweet or very sweet, medium or intense pomegranate flavor, not very acid, medium aril fir, no woody flavor, juicy, lotus flesh, and soft aril and soft seeds. In the opposite side, we can see Wonderful. It was described as acid, but mainly very acid. Not much pomegranate flavor, not very sweet, slightly astringent. Seed occupies all, and seeds are hard, and firariel. And A327 was the variety with the most intense woody flavor. It shared with Wonderful that it has seeds and hard seeds that occupy all and are hard. It is acid, but not so acid as Wonderful. And on the other hand, it shared with Moyar that is sweet and has an intense, medium intense pomegranate flavor. So now we can understand the preference patterns. The first group liked very acid and not very sweet varieties, and for them the presence of seeds was okay, was not a problem. The second group liked sweet varieties with low or medium acidity. They accept level of acidity higher, and they have not problems with seeds neither. And finally, the third cluster, they like only very sweet and not very acid varieties with soft seeds. So I remember you that Moyar and D2712 were the varieties that liked the most of the people. So what's happened with the appearance, with the other phases of evaluation? We will see. Here we have the liking of the appearance of the alleles, and it's clear that consumers show preferences for red intense alleles. So this was an advantage of D2712 compared to Moyar. Exactly the same happened at the external level. Consumers show preferences by the red skin. So D2712 was much more appealing for consumers than Moyar. If we look at all the aspects of organoleptic quality, organoleptic properties, we can see that D2712 is a very promising variety. It liked to 80% of people. It shares internal properties with Moyar, but is much more appealing for consumers because of the red allele, intense red alleles, and red skin. Once we have identified this very promising variety, the next season we wanted to know how it works during cold storage. So we store the fruit for up to 3 months at 6 degrees, plus a sale-life period of 7 days at 20 degrees each month. We evaluate the main physicochemical parameters, but I will focus on the consumer studies that we perform after each sell-life period. In this case, we asked them about the external appearance and the internal appearance, but we showed the fruit cut in half and open because we wanted to allow consumers to see the inner skin of the fruit, not only the alleles. And they also tested the samples and evaluate the liking after testing. So in this graph, we have in black the liking for the external appearance, in red the liking for the internal appearance, and in green the liking after testing. All of them were very good rates, and the most important is that were kept constant during the storage. That means that there was not a loss of quality. But I would like to comment that we see internal browning that affected the inner side of the skin, not the alleles. Actually, we don't know if the alleles are affected or not, but it's not visible because it's so red that we can see brown in that. So we think that probably the attention of the consumer goes to the intense red alleles, and they don't pay too much attention to the browning. So they don't penalize the presence of this disorder, which is also an advantage compared to Moyar. So as conclusions, there are different preference patterns among pomegranate consumer. Affinity, sweetness, and seed perception are the main drivers of liking or disliking. D2712 is a very promising variety. It shares internal properties with Moyar, and it's much more appealing for consumers due to the red coloration of alleles and skin. And it can be cold stored for 3 months without losing its high quality properties. And that's all, and thanks for the attention.
Thank you, Lucia. Any questions? No? Okay. Thank you so much. We can move again with Zenodel from University of California, USA, and Florida, sorry. And you will present developing copy Le Pomme as a model for pomegranate genetic and genomic research, characterization of its short humanity, genome, and flowering kinetics.
So you can see.
Okay, all right.
Be on time.
All right, sure, thanks. So Pepe Le Pomme is a new variety in the United States, mainly sold as an ornamental plant. So as an ornamental plant, you can say this plant is a pretty small dwarf and with lots of flowers on the plant. And also they can say the fruit, many fruit actually in the greenhouse. So, and another characteristic that they can flower and the fruit year round. So this is, I guess we think maybe have some potential for genetic and research, because for seedlings from standard varieties, normally you need 3 to 4 years for the seedlings to flower and the fruit. And this, of course, to many people doesn't matter. But let's say if you are a student and you have 4 years to complete your study, that means in the 4 years you can only maybe have 1 generation or maximum 2, and you can only generate F1 or F2 population, and you may be able to handle tens or even hundreds of standard varieties. But if it's more than that, it will be really hard. So that's why we think with the Pépinier palm, maybe we have good potential, especially for genetic research, for breeding research, because generally this plant only need half a year from sowing the seeds to produce, to open flowers, half a year. And also, on average, the plant probably is 0.3 meters compared to 1 or 2 meters tall. And in 4 years, you can have 3 or 4 generations. That means you can move from F1 to F2, F3, F4 generations. And because the plant is so small, you can handle hundreds or even thousands of plants from this variety. So we think there's a very good benefits for research and breeding. Okay. So first we would like to find out, what if we sow the seeds from this variety? Do they flower, in several months? So we did this in 2 studies. First, we collect the seeds in July, in the summertime. They sow the seeds in the summertime, 2022. And in this case, we grow the seedlings for several months. So in this case, about 70% of the progeny, they open the flowers in 7 to 8 months. About 30% didn't flower, within 1 year. Again, we collect seeds in January 2023 in the winter time. So in this case, about 70 or more than 70% of the progeny flowered in about 5 months, and about 16% didn't flower in 1 year. And then because this case, we see differences. In the first batch, the plants will grow in long days, here in short days. And they seem the plants even flower quicker. So now we would like to, to find out whether this trait will be expressed in F1 or F2. So in this case, we crossed, Pépinier palm with another variety, and then I guess here is another variety. We sow the seeds in 2 batches. In this case, the blue bar, the seeds were sowed in July, the red bar in February. And you can see when the seeds were sown in the summertime, some of the plants flowered, in F1 when you cross with a standard variety. And then when you sow them in February, the plants didn't flower. And with this variety, you see, all the plants flowered when you sow the seeds in summer, but some plants, flowered when you sow the seeds in the winter time. So based on this study, we think the juvenility, the short juvenility is affected by parent and also by growing season. And when we self this seeds, we self this plant, then they start to flower again in F2. So F1, F2. So based on this, we think this gene, we think this gene is a recessive trait. It's short juvenated. Juvenated is recessive, and it may be controlled by 2 or more genes. That's based on our preliminary data. And then we want to use this, characteristic to look at the inheritance for some traits expressing flowers. So in this case, we focus on double flowers. Double flowers are observed in quite a number of varieties. In this case, we use this variety, Habuku botan, because this flower has a different color. It's a creamy color, and also they have functional male and female. So we cross this with pépinier palm, and then we say some of the plants are flowered. We can say double flowers and single flowers. In this case, we have about 20 double flower progeny, 16 single flower progeny, and all the flowers are red. So we think the double flower is controlled by a single lotus. And then we cross this with another variety with the creamy flower, but the single flowers. So in this case, so all the flowers are red, all the flowers are single, and also we say color segregation in F2 generation. So that means the pépinier palm has some utility for inheritance study, genetic study. Okay. So then we use this segregant plants in F2. We did resequencing 64 genotypes, including some from USDA, from California, and then based on this, we found out there's one gene, I guess AP2 like a gene, and chromosome 3. They have SNPs, and based on this, we developed a marker for double flower trees. And in this case, you can say the single flowers have one peak, and then double flower have 2 peaks when we analyze with this molecular marker. And so then, so I guess now we did the sequencing, pack biosequencing of pépinier palm 229X coverage. We use the high-say rate to face the genome, and then we were able to develop a high-quality chromosome scale haplotype resolved genome assembly for pépinier palm. And this the genome size for each haplotype is about 290 and 327 megabase. Overall, pépinier palm genome is smaller, about 12 to 65 megabase smaller than the other published pomegranate genomes. And then, so this is a genome assembly of pépinier palm. And then we look at the flowering time genes. So flowering time genes are well studied in Arabidopsis. In this plant, there are 4 main genetic pathways controlling flowering time. So including the age or developmental pathway. So they have the genes to control flowering in the meristem. And then the second pathway is called the night. In this case, the photo period and the quantity, red or far red. So they control the flowering through these genes. And then also there's a temperature pathway. So these genes are involved in the temperature pathway. And then the fourth is called the autonomous pathway. So there are genes, in these genes that control the flowering. So anyway, in total, there's 174 flowering time genes in Arabidopsis. You probably know some of the genes such as FT, SLC1, TFL, etc. So we use this information to find the flowering time genes in pomegranate, in pépinier palm. So in this case, we download the protein sequence of this 174 Arabidopsis genes, and they search this in pépinier palm genome, and then we found out 1,166 genes, highly homologous to Arabidopsis flowering time genes. And then we compared these genes to the Tunisia genome, to the RLD genome, because they are standard varieties. Okay, then we find out 119 genes from pépinier palm. They are different from Tunisia genes, from other D genes. And we found some of the genes, they can cause the plants to flower early and also cause the plant to dwarf. I think yesterday Dr. Sakashi mentioned that dwarfism can be very useful for pomegranate breeding. I think Pépinier palm or Nana, they have this trait, may be useful. So in summary, we found out the Pépinier palm offers some good potential for genetic, genomic, and biotechnology research, such as maybe CRISPR gene editing, and also maybe potential for rapid breeding. And this short juvenility likely to be recessive, controlled by 2 or more genes. And we have genome assembly for Pépinier palm. We identified the candidate genes for early flowerings. So we developed one marker for double flowers. I think that's all I have. Okay, thank you very much.
Thank you so much. Any questions? Okay, I have a question.
Thank you.
I have a question.
Oh, sure. Yeah. Sorry.
Yeah. Did you check if the genes that you find were differentially expressed in Pépinier palm?
Which gene?
Differentially expressed.
Okay, so we identify some. We focus on R genes, so they are expressed. But these flowering time genes, we don't have the RNA-seq data, so we have not checked the expression of these genes.
Okay.
All right, thank you.
So we can. Any other question? No.
Okay,
we
all right.
can move to the next speaker.
Thank you
so much again. So next speaker is Domenico Rau from University of Sassari, Italy, and he will present his work on genetic structure and genes in an international programming collection, including Sardinia and germplasm.
Okay. Good evening. First of all, let me thank the organizer for the opportunity to speak about our work and our results. That is the product of several people from different institutions. So our work is on the study of genetic structure and the phenotypic and genetic relationship in an international pomegranate collection that also includes Sardinian germplasm. At a first glance, on the rear left, you can have an idea of the Sardinian collection. It is of 54 accessions collected by different institutions present on the island. And while the international collection was of 109 accessions collected in the year by Dr. Ferdinando Custio, accessions that were from different countries all around the world. Our aim was, we had 3 main aims. The first was to resolve the population structure of this collection, then to quantify the relationship between genetic cluster and fruit quality phenotypes, and demonstrated the utility of this pattern for signature selection mapping, which was essential for gene tagging. And overall, we want to provide a benchmark for future and expanded analysis. We started with sequencing all the 163 accessions on Tunisian reference. We applied standard filtering procedure, masking repetitive sequence, and to perform calling. Downstream, we perform a batteries of multivariate and population genomics method and statistical analysis to identify genetic groups, study clonality, the covariance between genetic and phenotypic variation, as also to look for signature of selection mapping and signature of selection and marker trait associations. Our resequencing was able to build a little bit less than 22,000 high-quality SNP that are well distributed across the assembly. Okay, with this marker, hierarchical clustering identify 2-3 main groups, but the silhouette analysis indicates the 10 groups as most likely. We refine our analysis with the descriptive analysis of principal component that was able to identify the event cluster. The APC was more precise in describing the population structure of this panel, reallocated several accessions, and also resolved several substructures that the work method was not able to do. So we continued our work considering 11 clusters. So the question now is which accession are in each cluster and how do the Sardinian and international collection combine? So we have combined very different collection, and this was the first question. So as you can see, we have 5 clusters that contain only international accession. We have 1 cluster, that the one that is in blue, that contain only Sardinian accessions, and we have 5 other clusters that contain both Sardinian and international clusters. So in general, in terms of cluster richness, the Sardinian collection is not so bad. I mean, it contains a little bit of some diversity, but it is clear that the island was not reached by all the genetic background that are present in the international collection. We show also the most important, or at least some of the most important international accessions that fit within each cluster. So how divergent are these clusters? We have used an index that is the FST, and in global AMOVA give an overall average FST of the psi of 0.156. That means moderate divergence. However, when we compare pairs of different clusters, we can go from small but not trivial divergence to moderate high divergence. So there is a quite complicated scenario. Excluding the Sardinian cluster does not change too much this picture. So we moved toward the determination of pairwise SNP differences between accessions. At panel-wide level, you can observe several distinct genotypes on your right, but on your left, you can observe a small lower tail of strictly related genotypes and likely putative clone duplicate pairs with less than 1% of SNP differences. What happened within clusters? So what are the differences within clusters? Here we observe that there are clusters that retain high diversity, but we have also clusters that have a depleted diversity. And the distribution of putative clones follow the same trend. We have one cluster that essentially comprises most all cases putative clone pair. So the question is, which cluster is this? This is the Sardinian cluster. So the Sardinian cluster, the Sardinian cluster has 34 accessions and a high level of clonality. So we look inside this cluster more in detail by network analysis, and we were able to observe a cluster of a network, let's say, a line of 9 very closely related accessions. 2 of them in red stand out as hub in this network. And overall, this picture is consistent with the notion that with clonal, with the propagation of a few successful genotypes by clonal propagation. So we expect the phenotypic variation is very low within this cluster, and However, it is not completely true, because if we look at this strictly related individual, we still observe different phenotypes, at least regarding, for example, fruit characteristics. And this is true also if we consider other varieties that have connection with both other. So this warns against considering very closely related genotypes as a mere trivial replicate, and also suggests a possible role for somatic variance in the evolution of this crop that is perennial with easy and widespread vegetative propagation. So looking at the variability of phenotype across clusters, we observed high variability, as you can see from the different color. For example, we observed that the C1 cluster it comprised several cases of fruit that are dark colored, while the C2 is characterized by fruit that are pale. Similarly, we have very hard seed in C4, and we have soft seed, or at least less hard seed, in C9. We formalized this observation by ANOVA that showed that all genetic clusters are significantly associated with the four morphological traits, and seed darkness are the strongest association, taste the weakest. This is true also if the Sardinian cluster was removed from the analysis. We removed the Sardinian cluster from the analysis because it's a little bit aberrant compared to the others, so it's a sort of sensitivity analysis. So now it should be noted that by themselves, ANOVA cannot establish whether the geometry of the phenotypic variation mirrors that of the genetic variation. So to achieve this aim, we perform Procrustes analysis that show in general a strong congruence between the two structure. The structure, in general, we can say that the conjoint variation of morphological traits explain 60% of the molecular variance. I will drop this part. Okay. So we proceed with the FST genome scan, and it was interesting for us to note that there are several parts of the genome where divergence is very high. In particular, we were able to observe a quite long block on chromosome 1 that is consistent with the chromosome 1 selective sweep described by Lu et al. In 2000. So this was confirmative, but for us also was encouraging because we can move with more confidence in the analysis of the other peak in our case. I'm finishing, sorry. The last thing was to perform a genome-wide association study. We performed this exercise considering all the traits. Here I show only the case of seed hardness and fruit taste with E-Link and FARM-CPU that are two multi-locus mapping methods. As you can see, we were able to observe several associations that pass the Bonferroni threshold, so quite stringent threshold, and the strongest association was on chromosome 1, where there was the haplotype that was also associated by Luo and coworker, and more recently by Chen and coworker with seed hardness. So the system is working properly. For this, guided by this result, we decided to established a new common garden in Sardinia with a representative subset of about 100 accessions. This will provide a uniform condition for long-term validation and standardized phenotyping. So in sum, this panel is now well characterized, and we show that it can effectively support informative studies in pomegranate. And in the next step, we will refine following several ways the observation that I presented here. Thank you.
Any question? I have one question. Maybe I didn't follow, but the international collection, in the international collection, you have genotypes from all over the world, and you know the origin of each genotypes or not?
Yeah, we know we have covered several geographical origin, not all. Okay. So, of course, there are some missing countries, but each accession has its origin. So, California, France, Portugal, Spain,
so the main
Tunisia, the main region, California. So, these are all represented. Yeah.
Okay. Thank you so much.
Thank you.
So, if there are no questions, we can move on to the next speaker. So the last speaker of the session is Tommaso Giordani from University of Pisa, Italy, and he will present his work on exploiting genetic variation in FIG3 to identify molecular markers linked to abiotic stress responses. So, Tommaso, the stage is yours.
Thank you. Okay, thanks to the organizer for giving me the opportunity to present our work that is part of a project PRIMA named FIG. Why a project on the FIG3? Well, this species is one of the oldest known domesticated species whose cultivation has been carried out for 12,000 years. Currently, the fig tree is an important crop with a very old tradition in all the Mediterranean basin. And we believe that it's a great potential for expansion due to valuable nutritional, nutraceutical, and energetic characteristics of its fruit, and also due to valuable secondary metabolites produced in fruit, as well as in leaves in the latex. Thanks to this trait, in combination with its great ability to adapt to adverse climatic conditions such as drought and soil salinity, and also its very low environmental impact of its cultivation, this made this species extremely promising as a sustainable crop in a context of a climate change. Despite these characteristics and despite the century of cultivation and the existence of numerous cultivars, this species has undergone a low level of genetic improvement. Population retain high genetic diversity that is still an underutilized resource. So research is awaiting for the characterization of existing cultivar that are stored in germplasm collection. Traditionally, germplasm characterization has been based on morphological and agronomic traits that are strongly influenced by environmental conditions, complicating cultivar identification. And more recently, ASA's marker has been used. However, these markers based on DNA characteristics are less informative than markers obtained through next generation sequencing technologies. These new tools allow the detection of million genetic variants along the genome, supporting population genetics, evolutionary analysis, as well as trait mapping through genome-wide association studies. These tools can address many issues of these species, such as the short shelf life of fruit, its susceptibility to the attack of insects like Acles taiwanensis, that is dramatically impacting the fig production in some Italian regions, such as Tuscany. In addition, it can improve food quality and production, the development of disease-resistant cultivar, and enhance tolerance to abiotic stress coming from climate change. So the objective of our study are analyze the genetic variability of fig tree cultivar in 3 germplasm collections of Mediterranean basin, and exploit this genetic variability to identify candidate genes linked to abiotic stress tolerance through genome-wide association studies. So we analyzed a total of 286 fig genotypes conserved in 3 germplasm collections in Spain, in Tunisia, and in Turkey. In this map, you can appreciate the different geographical origin of our genotypes. So for genotyping, we followed a whole genome sequencing approach. Briefly, genomic DNA has been isolated from each cultivar. It was fragmented, and each fragment was sequenced through an Illumina platform. Then, through a bioinformatic pipeline, each sequence has been aligned to the reference genome of cultivar dotato that we recently assembled in our department. After the alignment, it was possible to discover variants, and after variance filtering through statistical analysis, all the genotypes has been compared. With the aim to identify genetic or molecular markers associated to abiotic stress response, we performed 2 different experiments, one in 2021, a drought experiment, and the second in 2022, a salinity. It doesn't work, I don't know. A salinity experiment. In the first one, stressed plants were irrigated with 70% of water consumption, and in the second one were irrigated with 100 millimolars, with a solution 100 millimolar sodium chloride. In this experiment, in stress and control plants, we measured 6 parameters linked to water status and to plant growth, like leaf temperature, the relative water content, the chlorophyll content through SPAD measurement, the number of leaves, and plant height, stem diameter. All these parameters were analyzed through a ratio between stress and control plants to obtain stress tolerant coefficients. Finally, through genome-wide association studies, genotyping data and phenotyping data were combined using a linear mixed model and the GEMMA software. So, moving to the results, from all genotypes, we obtained a total of 6 billion pair ends, resulting in an average coverage per genotype of about 18 pairs. After mapping and filtering and variant discovery, we were able to obtain more than 1 million high-quality SNPs and more than 2 million indels. But what was the distribution of such mutations along the genome? As expected, the majority were located in the intergenic region. But interestingly, over 700 SNPs were found in genic region, not only in intron, but also in upstream and downstream regulatory region, and also in coding region. Moving to the coding region, what can be the effect of such a mutation? Interestingly, most of mutations were non-synonymous, so with a moderate effect on gene function. But interestingly, over 40,000 mutations, in particular indels, determine a frame shift or change in frame shift or in stop codon. So with an high effect on gene function. Moving to population structure, principal competent analysis showed a clear separation of Tunisian, Spanish, and Turkish genotypes, as we can see. The Tunisian genotypes in red, the Spanish in green, the Spanish in red, and the Tunisia in purple. Structure analysis show that here on the right show that our genotypes have the three ancestors and confirm the analysis of principal component analysis, showing that here you can see the ancestor in blue, in purple, and in orange, with a different predominant. So through this different predominant, we can separate Turkish, Spanish, and Tunisian genotypes. Also, phylogenetic analysis confirmed the separation, the clear separation among Spanish, Turkish, and Tunisian genotypes. But this analysis with in combination with kinship analysis allow also the identification of closely related genotypes, I mean possible synonymous from the same country, for instance, from Spain, the genotypes clone 300 and Granito, or from different countries. Indicating a possible historical exchange of germplasm among different collections. On the right, I report other genetic variability parameters. All these parameters indicate a moderate nucleotide diversity in each collection. And observed heterozygosity was slightly higher than expected heterozygosity, suggesting that possible recent event of cross hybridization among initially isolated population has been occurred. Moving to the last part of our research, genome-wide association studies allow the identification of more than 200 associated traits linked to drought or salinity response, and the identification of candidate genes involved in abiotic stress. Here, just I report just an example. A Manhattan plot where a peak on chromosome 3 include numerous SNPs associated to relative water content in the salinity experiment. I conclude with a list of candidate genes we identified through genome-wide association studies. There were these genes were associated to traits such as leaf temperature, relative water content, number of leaves, and plant age. And literature analysis confirmed their involvement in pathway related to stress tolerance, like stress signaling, cell wall remodeling, and redox homeostasis. So, moving to the conclusion, this study reports one of the most extensive genome investigation of figure carica based on 296 genotypes from Spain, Turkey, and Tunisia. The genetic variability analysis revealed more than 1 million SNPs and more than 2 million indels, with about 45% located in genic region. Most mutations were non-synonymous, and over 40,000 caused frame shift or stop codon change, having a high impact on gene function. Population structure analysis identified 3 main clusters corresponding to the countries of origin, but also highlighted possible intermixing among genotypes and possible synonymies. These results provide robust estimates of genetic diversity in the Mediterranean, supporting effective management and conservation of germplasm. Finally, genome-wide successful studies identify 200 SNPs as well as several candidate genes potentially linked to abiotic stress tolerance, offering valuable targets for future functional validation and the development of more resilient cultivar through breeding programs. I conclude with the acknowledgement of my collaborator from my department of the University of Pisa and my colleagues from Spain, from CCTech and CSIC, from Cukurova University from Turkey, and from the University of Tunis from Tunisia. Thank you for your attention.
Thank you, Tommaso, for the next presentation. Any question? Just one question.
Yes.
Did you validate some of the genes that you find associated?
Thank you for this question. No, not yet. We plan to analyze gene expression, and why not? Recently, there is a protocol for genome editing in FIG, so the construction of mutants can help the validation of these candidate genes.
Thank you so much. So the session is finished, and now we can rest with the coffee break.
Only few communication. I kindly ask you for a fast coffee break, so to be back in 15 minutes, because we are really late and we need to run the last session and then the ISHS business meeting. Another one, I remind you about the meeting point tomorrow. Take notes of the location, and then since I received a lot of requests, all the oral presenters can mail their presentation to the organization, so we can share to all the registered participants the presentation. Okay? See you in lounge for the coffee break.
We assign to one person and yes. I think we're having coffee now. If they have problems, we will show you some. We'll make one tomorrow. Okay. Strategy for meeting protecting that's why it's 2 months. 2 weeks. Oh yeah, if you get in it. So well, you're forgetting it. Yeah, so so what I yeah, so
South Sea, close or next to the sea. Yes, of course, all programs also going on now. We have new animations and we checked it for 5 years. The second one,
Zwiwa zwi henefha vha khou zwi vhona musi a huna ndi yo sedza ngei ndi sokou But ladies and gentlemen, it's time for us to
also now some new companies in Turkey interested with also that is also in some companies here. But in some companies there is the people understanding from Hungary is different from all over the world. Different from China, different from India. Americans bring some samples from Dubai. In Dubai market, in Arabic markets, products by India. From Russia, if you take to India, and the close their eyes, and the difference is nice and sweet. Yeah, there is no aroma up to that.
So obviously anything that we do, we need to make sure that it's something that
I'm also normally in this situation, and we produce. And we produce, of course, all these people making everything. They say, yeah, that's not good. The other is not good. The other language is not enough. And you put you in producer, then things okay.
What's your right is like a Sunday, that's a big problem. It's a lot more union in Southern industry and more.
The right is not features and molecules. This in every stage they are so susceptible to some doing chromatography. Yeah, it's the same as I come to the lab. That's why it is so important issue here. If you organize, if you talk to that, in every stage, there is so high resistance level to the soil. And also, someone is directly affected from your irrigation system. We are really shaping or something like this, or using external vaccine.
Some products that we added on the solution.
But I mentioned that's the management and of course, and if necessary, sometimes we use extra containing materials, some which can be found organic materials.
saying, do you manage it successfully? But not giving away your secrets, unless the regulation and it's done. Sorry, unless the regulation and it's done. Yeah. It's funny, we ask everybody at with this, how much water per hectare is how much do you? What are your policies? Either I need my selections to be within for I gave maximum 600. That's what they got. Yeah, 600 cubic meter per hectare. Per hectare? Per hectare, so that's 6,000. That's it, 600 million. Yeah, 600. And yeah, maximum. That's why I try to choose resistant one for also I can say efficient irrigation, resistant to sunburn, because we choose that among towns. And I think maximum 600 millimeters of water per season. And the other thing, the grounds we have different maps. We have open one and also closed one. Even we have some hybrids completely closed and also not sold. Why? There is some machine. That's why we also thought we developed and they are really what we don't use. In the last report with the ground one, I don't know, even whole ground and they put the simple stocking machine and they check it. 50 years and then what happened? The result was but you can't cut it. Yeah, even those ground like that's interesting. We should have it because we're busy looking now at expanding our selection of what's your irrigation level during fasting?
From January, August to August, we irrigate 7 days a week, 2 times a day.
2 times a day?
Predominantly, we have very simple. So
less than half a week is
yes, half a week so we can't keep a lot of water here.
Of course, you are also doing, I'm sure you are doing your best for Panama competitions. But I may say this, by managing a good irrigation, of course, you will see someone, if your is so open to directly someone, and also your I prefer to make plantation 1 to 2 for example, we show to explain you 5 meters between the rows, 2.5 between the trees, and like this. We like to close the line and open the main branches in the rows. And why are we are trying to make a shading of the touching? No, exactly no. And of course we have to put a space for letting light. And by this shading, I cannot say we protect the sun, but the shading in management of this one, can you find labor? Usually, yeah, that's
On average,
So it's 150 per hour. But the productivity isn't that high. Yeah. For regular time, explain some small activities around and cleaning
I'm trying to see the sense for
I'm checking the laptop once
Okay.
So it's time to start the last session of this afternoon. It's a session dealing with field management, and two contributions are expected. The first is given by Julian Bartual, and concerns canopy position and fruit quality attributes affect arid growing disorder of the maize. I recommend all the speakers to stay in the assigned time.
I don't know where my presentation.
It's a PDF, right?
Is this Good afternoon. Well, sorry. But it's PDF, no es PDF.
Okay, it should work.
Yeah.
Okay.
Did you get it? Thank you. Good afternoon. Congratulations for all members of the committee, but it doesn't work. So,
Ok, dobbiamo chiudere queste. Chi è? Mandare queste in... This one?
In order to...
Maybe I don't know if it works with the PDF. Okay, this one, next slide. Okay, this one. This is the part.
Only
Okay.
Well, I'm going to start again. This is our work we made in the experimental station of Felce in collaboration with the IEA in Valencia. This is the history of an issue. We have a problem. As you already saw this morning, when Bernard showed the presentation, we have a problem with the husk. We have a browning husk
and internal browning that appeared maybe in 2020. It was started or come here, or appear in the asking for what was the problem. Have to maybe Bernard saw in the presentation, so that is maybe another is one way to solve, but not the cause. But in 2022, appeared another problem. Maybe the question is the same. Why? Maybe genetic, maybe environmental, maybe management, I don't know. But it was the problem. In order to clarify the difference between this type of browning, on the left we have the husk browning. We have it. I'm going to see in the inside of the husk, but we are going to go. I'm going to see in the post-harvest, in the storage, maybe after 4 or 8 weeks, after with 5 degrees Celsius. But the average we're going to see in the pre-harvest, maybe in July, in the north hemisphere. Maybe 2 months after the flower. The description is, but maybe there's some description about this problem was having farmers, but I don't clear it's the same. Because it's not so unfortunate in normal varieties. But I don't think it's not the same because we have a black dot in the problem. A black dot in the segment. And what is more important, we have impression taste and visual appearance. So it's normal in the quantity. Some reports show the physiochemical differences in areas in the browning and no browning are fruits. In higher levels of total sugars, reducing sugars, total soluble solids, starch, a polyphenol oxidase enzyme is in browning areas. But on the contrary, we have less level, less content of the moisture, proteins, anthocyanins, phenolic compounds, yaminas, and fine. In 2022, take advantage we have studied the Hart's browning index. When appear the area browning index, we studied some orchard. And we can see in this slide, the same orchard has a little score in half browning, but a high score in early browning. So it maybe one thing we have to see. Where do our current lines of work in the experimental station and EVIA? We have to main line is plant material selection, ensure the identification of traits associated with tolerance, the study of physiological aspects as stomatal density, metabolic changes, etc. After that, we went to select and multiplicate of tolerant genotypes, incorporating browning resistance data into breeding programs. As this session is management crop testing cultivation practices we are going to do in this essays is pre-harvest foliar spraying with elicitor nutrients, optimizing fertigation strategies, for instance, partial root zone grading, using mulches and saddling nets, or new tree training systems. The products we are using for pre-harvest foliar spraying with elicitor nutrients is natural biostimulant elicitors or salicylic acid, jasmonate, acetylsalicylic, aminobutyric, mineral and organomedical products as calcium and copper,
osmoregulators, or glycine beta or uricalcium enhancers, because it's fungi or it's bacteria. We think these compounds, also these compounds, can trigger that defense mechanism because the temperature during the last 150 years is rising. I think it is the cause of this issue. Maybe not radiation, not directly the radiation, but maybe the human and night temperature is rising. The first trial is study the fruit position within the tree canopy. So I'm going to see the lower and upper part of the canopy, the orientation, north, south, east, west, for the exposure, inner or outer the canopy. We evaluated in a scale of no symptoms, 1, slight, moderate, severe, and very severe. Important data is in the traditional training workshop in Spain. Sorry, yield is in the lower part of the tree. 64% of the production is in the lower and the middle lower part of the tree. Another interesting data, maybe result is very severe. When we account all the fruits, the 60% were very severe, and 17 was severe. 33% was severe for scale. But if we divide into the study, for instance, the lower part we have to see the red bar. In the upper part is the higher level, the highest score from all the results. Okay. When we talk about orientation, you can see the red bar and also some bar, and in this case, west and south has the west was the higher level of score in the severe injury. After that, the south. After that, the east. Even with less, the lesser was the north. Okay. When we see the inner and outer part of the canopy, the inner part was lower than the outer part of the canopy. Summary: when we count the number of fruits with low level of score or less injury, it was north orientation and low orientation. The second study was physicochemical traits and other characteristics associated with Browning disorder. We used 80 pomegranate extensions, adult trees belonging to agricultural experimentation of Elche and harvest. But has Browning was studied after 4 weeks on storage at 5 degrees Celsius. The trace study was fruit and early weight, peel and early color, sheer hardness, table acidity, pH, and total solid solids. But at the yield, if we have suckers or no suckers, the harvest date, very early, medium, late, or very late, and also other physiological disorders as cracking, sunburn, has browning, and alternaria, fungus, or blackhead. This was the scale. The colors, but we have to unify cream, pink, dark pink, red, and dark red. The trace study, physiological chemical tension disorder. The more of the areas was between 30 and 50 grams, 100 areas, and it was really more than 60 grams of the areas. About 29 of the study genotypes were tolerant to early browning, while the rest showed moderate or severe susceptibility, maybe 30%. This is the histogram, but we have to highlight the very difference among this genotype of cracking from 0 to 64% of percentage of fruits in cracking, and some are from 1 to 50%, and alternaria from no symptoms to 14. And taking a look to the correlation table, we can see we have positive in blue. No, in red, sorry. Positive correlation with fruit areas, with area weight and pH. pH is positive, area weight is fruit weight, and negative correlations was found between area browning and intensity of the red color and acidity, highlighting the importance of genotype in physiological disorder. The analysis of principal components, we saw that the more of the genotypes that with severe injury from early browning is this group, is for near mollar de Elche, gorda sátiva, mollar de Albatera, and something like that, they are pink areas, green pink, sweet, and very soft seed. We have an important association with aerial browning and display component. More affected as Peronski, Dulce Málaga, Gorda Sátiva, all of them lower of 0.4% of acidity. Y less affected, Candahara, de Caneroso, Ariana, Mollar de Valencia. There are one Mollar with less affected group, Rugalate, PTO weight, and Born. Maybe hopeless is a difference between Mollar clones in the same orchard. We have scored some of them with 1.5, maybe. Another one, a very severe damage. As a conclusion, canopy position significantly influences the severity of early browning. Physiochemical traits such as organic acid are associated with browning susceptibility. Genotypic variability suggests the possibility of selecting and propagating mortal and accessions. Further studies are underway to evaluate use traits, including antioxidant capacity, phenolic contents, anthocyanins, and organic acid. This threshold is not worth that we have it. This is the imagine as a picture of a mollar de Elche. It's not the same that we saw in South Africa. You remember it's very green because the conditions is very different. It's not the same, but it's mollar de Elche. Thank you for the attention.
It's important to cover with quality worms in the field. Unfortunately, we do not have time for questions, so I have to call the next speaker. We can have a discussion later, hopefully. Thank you very much. Next speaker from Stellenbosch University, South Africa, Mutianzene Rabiumaso. Sorry for the bad pronunciation. The title of the presentation is Emergency and Canopy Factors Influencing the Propagation of the Pomsym orchard under semi-arid conditions. Please, the floor is yours.
Okay. Good day, everyone. I'm going to be the last speaker today. Basically, I'm from the ARC, the Agricultural Research Council, not Stellenbosch University. But anyway, this study is part of a bigger project that was ran by Dr. Teresa Volsking, who's my supervisor. So As we all know, there are two major processes that are responsible for water uptake in the ecosystem. It's normally transpiration and evaporation. So for this study today, we're going to be dialing into the transpiration to check what are the major drivers of this transpiration. So normally, as we know, the canopy plays a role and the weather variables as well. So as we all know, we are battling with climate change, and the main thing that we know is that the temperatures are rising up, and South Africa is not immune to that. So the other thing that we know about South Africa is that it has a mixture of arid, semi-arid, and Mediterranean climatic conditions. And these climatic conditions, or rather in South Africa, the country is prone to drought, and other major challenges that exacerbate that is the climate change and seasonal variability. So when we talk about seasonal variability, we're talking about the El Nino Southern Oscillation. So sometimes you can get very dry years, and sometimes you can get very wet years. So during very dry years, that is when the issues come about. So in the Western Cape, which is the study site, it's a Mediterranean climate, receiving rainfall during the winter time, but the production is in the summertime. But it's important again to mention that this Mediterranean climate has semi-arid characteristics. So the major challenges, or rather maybe the main challenge that is need to be noted, or rather what the audience need to note down as the high temperatures, the high evaporative demand, the extreme drought conditions, and the erratic rainfalls. So with all of those challenges that I've mentioned, there are two simple strategies that South Africans can actually use by picking out drought-resistant crops and understanding the limitations of those drought-resistant crops. And lastly, is to schedule their irrigation accurately. So the main objective of the study, we are looking at transpiration, or rather the water loss from the plant from a daily perspective, and comparing it with the canopy dimensions, and then comparing it as well with the weather parameters. So we wanted to dial in as well into the hourly data to see what are the main drivers on those two scales. So the study site is in the Western Cape. The young pomegranate orchard was a volunteer, which was between Gouda and Porterville. And the mature one is just 15 kilometers outside of Wellington, which was Welgemoot. So to get our data, we use two weather stations. And with the weather station data, we use it to correlate it with the transpiration. And then we calculated the VPD and the evaporative demand as well. So to quantify the transpiration, we use heat ratio method. We picked out 4 trees on each orchard, the mature and the young orchard. And then from that, we correlated to the weather parameters. So to quantify the canopy characteristics, we use an Alcupa LP80 septometer, as you can see there. And then we chose 8 days without any cloud. And then this happened during the 2023-2024 growing season. So for the results, so we did linear regressions, and then we compared weather variables as well as the fractional interception and the leaf area index. And what we realized was that the fractional interception and the leaf area index are not good indicators for transpiration throughout the season. You can actually see their contribution during the beginning and the end of the season, but in the middle, that is when the weather parameters play a huge role. And then we realized as well that the net radiation, as we know, had the highest R-square values when we just checked the young orchard, the mature one, and both of them combined. However, the other thing that we saw was that the minimum relative humidity was the second best indicator of water loss, and the T mean for the young orchard. And what we speculated was that it might be due to the row direction, so the orchard orientation against the prevailing winds. So out of the 8 days that we did the fractional interception measurements, we chose at least each day during the November time, whereby we looked at weather parameters with the transpiration. And then what we observed was that the transpiration, or rather the net radiation, is the initiator of transpiration. And then when we looked at the temperature, we realized that it is kind of a regulator. At about 25 degrees Celsius, you see stomatal control. And then this is for the young orchard, and then for the mature orchard, similar results were observed. So again, as we spoke, the wind direction, or rather the wind speed, as well as the row orientation played a role here. And the wind speed was observed to be an instigator. So it hypes up the transpiration if there is any increase in wind speed. So we then took the transpiration data on an hourly scale, and then we compare it with weather variables on an hourly scale as well. And then what we observed was that there is differences between the young orchard and the mature orchard. And this difference is due to the microclimate within the orchard, because the prevailing winds are southwesterly, and then the road direction, it's east-west for the mature orchard, and then it's north-south for the young orchard. So there is more air movement here as compared to there. So that you will see as well having an effect on the temperature as well. So what we observed again on the temperature, if you look at right about 25 degrees Celsius, that's when you see a change in direction of the transpiration. However, when you look at the mature orchard, the transpiration starts out very low, and then at 25 degrees Celsius skyrocket, which can be explained by the effect of the road direction, as I've explained. So the other thing that we observed as well was that relative humidity, the transpiration decreased with the increase in the relative humidity, and that was basically similar for both of the orchards. So this is the young and then the mature. And then correlation with the wind speed. The wind speed alone did not give a really good correlation. And then, but what we observed was that there was higher wind speed in the mature orchard as compared to the young orchard. So we looked at the vapor pressure deficit. So what we observed as well here is that here there is a clear case whereby you see that at right about 2 kilopascals, there is a change in direction, but you don't see something similar to that on the mature orchard. Okay, let me finish up. So what we did was to do a stepwise regression so that we observe which weather variable plays a role comparing the young and the full berry. And then what we observed was that for the young orchard, the air temperature was the major contributor. And for the full bearing, it was the net radiation. Let me finish up. So in conclusion, it is observed that the net radiation, it's the most important variable when it comes to predicting the transpiration, as well as the air temperature and sometimes the wind speed, depending on how your rows are orientated. The other thing that was interesting was that at 25 degrees Celsius, that's when you see the pomegranate starts to regulate its stomatal control. And thank you to my funders. It was the Water Research Commission, the NRF, and the Agricultural Research Council for all of the support, and all the other collaborators and the farmers that contributed a lot in the study. Thank you.
Thank you very much. We have time for one brief question. There is any? Any question? Okay. So thank you.
Thank you.
Thank you very much. And now it's time for the ASHS business meeting. I call Piera Olani. And Giuseppe.
Le regole, rules, the rules.
Non so quanti sono, poi si prende qualche altro foto per votare una penna.
Non lo so, ok.
Dovete girare.
Sì. If you need to slow down and up, okay, you can do whatever you want. Professor, maybe then
You want to share?
Yes, and you can
Are you
find.
not here?
No, I have to go.
Okay.
You can find the video here. Okay, proposal to host Pomsym.
Yeah.
Okay, I left it open so you can. Okay, I just put. So you can find here. Okay.
So to proceed, okay, it's already there. So they click and it's gonna be up there already.
Proposal to host Pomsym
Yeah.
in 20 minutes. Can you go this one? Okay, and it's here. Okay?
Okay. So this, yeah. Okay.
Quindi bisogna vedere uno che ne posso parlare. Una presa. Se lo si avvicino quando so
So, good evening, colleagues. So, can we get people to come in so that we can start and finish as soon as possible? You can get people to come in. Can you ask people to come in? This is an important aspect of this symposium. Okay, maybe we can get started. I think people should come inside. But like I said, today we are having the ISHS business meeting, and this is the meeting in which we decide who will be the convener of the next symposium, and of course the destination of the symposium. We are going by the guidelines set by ISHS in terms of who is eligible and who is not eligible. The first thing that I know for sure is that you must be an ISHS registered member. That's for sure. And because it's so critical to the selection or the election process, we already have a system in place to verify. Stefano will help us with that because we know that membership can actually be updated. You might not be a member now, but then in a few hours' time, you are a member. So it is important to verify people's membership, then you can vote. And we'll make the vote anonymous or secret. So if not anonymous or secret, you don't need to be compelled to actually reveal your vote. So that's about the convener. The other part is the second election, in which we will decide whether I should continue as the chair of the working group. I'm putting forward my candidacy. I thought I've done a good job because one of the roles of the chair is to support the incoming convener and also support, just like I supported yesterday, and also to support whoever will host the next symposium. So we'll get to the second part, but let's get the first part done. I will ask Giuseppe to help briefly just skip through the guidelines because we want to go by the guidelines. The other thing that I wanted to mention here is that we are doing the election anyway, but it's not the final. The final approval, which Giuseppe will actually mention, actually lies with the president of ISHS and with the government body. We will put forward the outcome, and then they do the approval at the end of the day. So propose a contract. Yes, there will be a contract to sign and all that. Having said that, Giuseppe will read the regulation by the ISHS, and then that will convince people to put forward their candidacy or to withdraw their candidacy based on this regulation. And then we can proceed to call the potential candidate. Thank you very much.
Okay. Thank you, Olani. I will read the most important criteria for selecting the symposium convener and sites. Candidate convener must be a member of ISHS. Involvement of the potential convener and/or members of the institute in previous meetings of the working group. Scientific standing of the potential convener in horticulture and/or subject related to the working group. Evidence of organizational capacity of the convener and the supporting institution and/or PCO, professional conference organizer. Evidence of institutional and industrial support. Proposed location must be in a different country, preferably in a different continent than the previous meeting of the working group or the series. Likelihood of the proposed location for international participation. Important criteria are e.g. infrastructure, safety, ease of access, political stability, and so on. Potential of the proposed location for providing interesting scientific visit for the attendees related to the subject matter of the working group. General attractiveness of the potential venue and location to attendees and their accompanying person. These are the most important criteria that we have to consider for each proposal to be voted.
Great, thank you very much. So then I will move on to the next step. Having known this or understood this, do we have any question? Then I will call on potential interested parties who might want to put their candidacy forward. Any question? Can we move on to calling people now that might be interested? So if you are interested to, and you are capable to host the next, the next event in 2028, I believe, 2028. Can I see, or can you raise up your hands, please? We have 3 people, 4. Okay, together. Okay, we have 3 people. Can I ask you to come forward, please? Can you give them a round of applause? Can I ask you to come forward, please? Sit right there. Thank you very much. I know the 3 of you, but in case people did not know, for those who might not know you, can you introduce yourself, your name, the country you're coming from, and then your specialty area?
Hello to everyone, good evening. I'm pleased to be among you, and thank you for this excellent symposium so far. I am Dr. Sudirulai Amidu. I come from Cyprus department. I am a representative of my institution, Agricultural Research Institute of Cyprus. I work at the Fruit Trees Division and Viticulture for more than 15 years. I have many collaborations mainly with Mediterranean countries so far. I will be pleased to. Thank you very much. And I will be pleased to collaborate with across the European and Mediterranean borders. So far, for this now, I will present you later our candidacy. Thank you very much.
Okay.